بسم الله الرحمن الرحيم. الحمد لله والصلاة والسلام على أشرف خلق الله عليه وصحبه الكريم. أهلا بكم. Attending and participating in the 11th annual Forum for Medical Research titled COVID-19 Vaccines, Global Challenges and Prospects, under the auspices of His Royal Highness Prince Abdullah bin Bandar bin Abdulaziz, Minister of National Guard. This virtual forum is organized by وش إن الصوت كان واضح. يعطيكم العافية جميعا إن شاء الله. بداية موفقة. عليكم السلام ورحمة الله. طيب الله يسلمك. إذا بتبقى هنا دكتور بندر.
أعلنت المملكة استضافة قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة
We now begin at one time and with one goal. A journey on a bullet train in which we race against time towards developing a vaccine to save life and humanity. This journey includes 32 experts and 10 scientific sessions, in which we discuss 6 topics revolving around developing a successful vaccine against the COVID-19 virus. From Riyadh, Kingdom of Saudi Arabia, we welcome experts to whom the world looks with hope, and major companies that will lead the way in distributing vaccines around the world. From Health Affairs of Ministry of National Guard, King Abdullah International Medical Research Centre, KMARC, King Saud bin Abdulaziz University for Health Sciences, and the Saudi Secretariat of the Group of 20, G20. We say to everyone, let us proceed with one ambition and with one goal towards a meaningful life, and let us work together to protect the health of our fellow human beings and our future generations.
بسم الله الرحمن الرحيم. الحمد لله والصلاة والسلام على نبينا محمد وعلى آله وصحبه أفضل الصلاة وأتم التسليم. السلام عليكم ورحمة الله وبركاته، وأهلا ومرحبا بكم في حفل افتتاح المنتدى السنوي 11 للأبحاث الطبية، والذي ينعقد هذا العام بعنوان لقاحات كوفيد-19 التحديات العالمية والمستقبل، برعاية كريمة من صاحب السمو الملكي الأمير عبد الله بن بندر بن عبد العزيز وزير الحرس الوطني. باسمكم جميعا أتشرف بالترحيب بمعالي الدكتور بندر بن عبد المحسن القناوي، المدير العام التنفيذي للشؤون الصحية بوزارة الحرس الوطني، رئيس جامعة الملك سعود بن عبد العزيز للعلوم الصحية. وباسم اللجنة المنظمة نتشرف بحضوركم الكريم حفلنا هذا، والذي تنظمه الشؤون الصحية بوزارة الحرس الوطني ومركز الملك عبد الله العالمي للأبحاث الطبية وجامعة الملك سعود بن عبد العزيز للعلوم الصحية، بالتعاون مع الأمانة السعودية لمجموعة ال20. Distinguished participants, it is our pleasure to welcome all of you attending and participating in the 11th Annual Forum for Medical Research titled COVID-19 Vaccines, Global Challenges and Prospects, under the auspices of His Highness Prince Abdullah bin Bandar bin Abdul Aziz, Minister of National Guard. This virtual forum is organized by Ministry of National Guard Health Affairs, King Abdullah International Medical Research Center, KMARC, and King Saud bin Abdulaziz University for Health Sciences, in collaboration with G20 Saudi Secretariat. This global forum comes with a great collaboration and support from international organizations, including University of Oxford, Deep Knowledge Group, MRI Global, Kellogg College, Zoom, Bring Hope Foundation, CB and CBHI. In this year, the Kingdom of Saudi Arabia assumes the 2020 G20 presidency, creating a cooperative environment to introduce initiatives that fulfill the hopes of the people in the world. This global forum is one of the most important events in the field, aiming to bring together leaders in the field of COVID-19 vaccine development and production, health and academic institutes, businesses, and world organizations to discuss the latest update on COVID-19 vaccines. It is an honor to have a welcome address from His Excellency Dr. Bandar Al-Ganawi, Chief Executive Officer, Ministry of National Guard Health Affairs, President King Saud bin Abdulaziz University for Health Sciences.
بسم الله الرحمن الرحيم. الحمد لله رب العالمين، والصلاة والسلام على أشرف الأنبياء والمرسلين سيدنا محمد وعلى آله وصحبه وسلم. الأخوة الزملاء، زميلات، السادة المتحدثين الدوليين والمحليين، ضيوفنا الكرام، السلام عليكم ورحمة الله وبركاته. نرحب بكم جميعا في حفل إطلاق منتدى لقاحات كوفيد-19 التحديات العالمية والمستقبل، والذي تنظمه وزارة الحرس الوطني متمثلة بمركز الملك عبد الله العالمي للأبحاث الطبية الشؤون الصحية بالوزارة، برعاية كريمة من لدن صاحب السمو الملكي الأمير عبد الله بن بندر بن عبد العزيز وزير الحرس الوطني حفظه الله. لتأتي هذه الرعاية امتدادا لدعم سموه الكريم للشؤون الصحية وجامعة الملك سعود بن عبد العزيز للعلوم الصحية ومركز الملك عبد الله العالمي للأبحاث الطبية. ضيوفنا الأعزاء، يستعرض هذا المنتدى أهم المواضيع على الساحة العالمية، ويطرح محاور التحديات العالمية والمستقبلية في أهمية الوصول للقاحات تتصدى لجائحة كوفيد-19 بمشاركة كبيرة من كبار العلماء والمختصين في مجال اللقاحات الطبية وأهم الشركات المنتجة للقاحات في العالم. كما أن هذا المنتدى يأتي استكمالا لسلسلة النجاحات المتميزة في التصدي لجائحة كوفيد-19 رغم الظروف العالمية الحرجة التي نعيشها في هذا العام جراء تفشي هذا الفيروس، وما اتخذته حكومتنا الرشيدة من قرارات وما وفرته من دعم للسيطرة على تداعيات هذه الجائحة وما قامت به من دور عالمي في مكافحتها. ومن هذا المنبر شرفني أن أرفع لمقام سيدي خادم الحرمين الشريفين الملك سلمان بن عبد العزيز آل سعود حفظه الله، ولمقام سيدي صاحب السمو الملكي الأمير محمد بن سلمان بن عبد العزيز ولي العهد نائب رئيس مجلس الوزراء وزير الدفاع حفظه الله، جزيل الشكر والامتنان على ما يلقاه القطاع الصحي وكافة القطاعات في وطننا الغالي من دعم منقطع النظير وتسخير كافة الإمكانات لمكافحة هذه الجائحة العالمية. كما يسرني أن أرفع لصاحب السمو الملكي الأمير عبد الله بن بندر بن عبد العزيز وزير الحرس الوطني خالص الشكر والعرفان على دعمه للشؤون الصحية لتنظيم هذا المنتدى وتفضله بالرعاية الكريمة لتسليط الضوء على جهود المملكة المتميزة في التصدي لهذه الجائحة والإجراءات الاستباقية والاحترازية التي اتخذتها، حيث أصبحت هذه الجهود مثالا يحتذى به ومنهجا يتبع. كما أتوجه بالشكر لضيوفنا الكرام ورؤساء الجلسات والمتحدثين من داخل المملكة وخارجها على تلبية الدعوة ومشاركتنا فعاليات هذا التجمع العلمي المتميز. والشكر موصول للجان العلمية والتنظيمية على حسن تنظيم وإدارة هذا المنتدى بالصورة المشرفة كما عودونا دائما في المحافل العلمية الدولية والمحلية، داعيا الله سبحانه وتعالى أن يحقق لهذا التجمع العلمي البحثي العالمي الأهداف المأمولة بما يخدم إنتاج اللقاحات المناسبة والضرورية لمكافحة الأوبئة في جميع أنحاء العالم. والسلام عليكم ورحمة الله وبركاته. Now my speech in English. Excellencies, distinguished delegates, honorable guests, dear colleagues, ladies and gentlemen. From Riyadh, Saudi Arabia, I am honored and truly pleased to welcome you all to this day, to the KMARC 11th Annual Forum for Medical Research. This year, we concentrated on COVID-19 vaccine global challenges and prospects. We're absolutely delighted to host this unique virtual conference, which represent the most important event this year during this pandemic. Wherever you are, I hope one day that I will be able to welcome you personally in Riyadh, where you are always welcomed. I would like to thank every one of you for taking the time to join us. We thank His Royal Highness Prince Abdullah bin Bender, the Minister of the National Guard, for his strong support for this event, and the G20 Secretariat as one of our organizers. Since the initial onslaught of this COVID-19, the world continued to reel with the impact of this virus, where it took unprecedented way. COVID-19 has forced the world in an effort to rapidly develop a vaccine. The process that typically takes and require years are shortened for a few months. For the world to recover faster, it has to recover as one. In the year of Saudi Arabia G20's presidency, we're pleased to spearhead this event by bringing together leaders in the field of COVID-19 vaccine development and production from different health institutions, academic institutions, business, and world organizations to discuss the latest updates on COVID-19 vaccine development and possibly the availability of this vaccine in the near future. Let us, as a global partners, continue developing the COVID-19 vaccine. The world looks forward to the COVID-19 vaccine in order for us and for our lives to return to normal. This two-day global forum is structured to represent all aspects of the vaccine and development to ensure global participation from experts, decision makers, policy makers, agencies, healthcare workers, and the public in general. I am thrilled to welcome over 10,000 participants who registered for this forum from various countries around the world. The prominent speakers will shed light on the latest clinical data for the frontrunner vaccines. We also will hear about the latest plans for the access and distribution for these vaccines to various communities. There will be a discussion on the importance of vaccination to save lives based on scientific evidence and principles. I would particularly like to thank the organizing committee, the advisory board, for the collaboration, sponsors, and the speakers for indeed an important event that all of us are attending today. And was as well to extend my appreciation for all of our attendees. I thank you for participating in this important conference and wish you all the best and hope that you will gain a valuable learning through this activity. Once more, welcome to Riyadh. Please enjoy this exceptional event and timely forum. Thank you very much.
Proudly, we can say that our organization successfully meeting the goals and achieving the government mission in advanced health sciences and researches. This is the annual KMARC forum that comes with the related topics to our health system. And in this year, the focus is on COVID-19 vaccines. Next is a forum introduction by Dr. Ahmed Al-Askar, Executive Director, King Abdullah International Medical Research Center, President of 11th KMARC Annual Forum for Medical Research.
بسم الله والصلاة والسلام على رسول الله. السلام عليكم ورحمة الله وبركاته. يسعدني ويشرفني أن أرحب بكم في المنتدى السنوي 11 للأبحاث الطبية، واسمحوا لي أن ألقي كلمتي باللغة الإنجليزية، اللغة العلمية لهذا المؤتمر. It's truly a great honor and privilege to welcome you to the 11th Annual Forum for Medical Research, the premier national forum for medical R&D and innovation in the Kingdom of Saudi Arabia. This year's edition, entitled COVID-19 Vaccines, Global Challenges and Prospects. It will come at no surprise to you, with the strong focus around the current COVID-19 global pandemic, with a very special emphasis on the much needed vaccines for this infectious disease. This very important scientific global gathering is being held under the auspices of His Royal Highness Prince Abdullah bin Abdulaziz, Prince Abdullah bin Bandar bin Abdulaziz, the Minister of the National Guard of the Kingdom of Saudi Arabia, with the strong support and commitment of His Excellency Dr. Bandar Ganawi, Chief Executive Officer, Ministry of National Guard Health Affairs, and in presence of elite scientists, corporate executives, healthcare professionals, academic experts, and representatives from various organizations around the world. We are so proud to host this outstanding event through our center, King Abdullah International Medical Research Center. KMARC, while a young biomedical research and development and innovation enterprise, yet have succeeded at KMARC to establish a robust R&D agenda with a number of strategic disease areas, including infectious diseases, as a cornerstone of our R&D agenda. This has ultimately led us to collaborate and participate along so many other leading academic and industrial research entities worldwide in the fight against COVID-19 global pandemic. Last year, we have successfully completed a trial for yet another coronavirus, the MERS-CoV. It was a trial for a vaccine candidate for MERS-CoV that was conducted in camels and was developed in collaboration with the Jenner Institute of Oxford University. Subsequently, earlier this year, we completed the phase 1 clinical trial of this vaccine in human. This had enabled us to further develop and strengthen our R&D program for emerging pathogens. Since the beginning of COVID-19 pandemic, we have launched an intensive multifaceted R&D program using an arsenal of various technology platforms, including the whole genome sequencing for the virus, for screening of druggable targets using bioinformatics and drug discovery platforms. As a result, we ended up with at least 4 interventional clinical trials that are led nationally by KMARC. As a result of this extraordinary concerted effort, we now have more than 150 COVID-19 research projects at KMARC, generating an excess of 110 publications and 8 patents applications so far. And research still ongoing, we remain open and welcoming collaborative opportunities with all local stakeholders and international partners sharing similar vision and goals. My dear colleagues from around the globe, this gathering, which is your gathering during these difficult times, will no doubt help guide us in answering, or at least attempting to answer, so many queries that are still standing and would hopefully assure the public and shed more light on the expectation from all the global scientific efforts aiming to uncovering solutions for this pandemic. Having said that, I must commend the unprecedented global efforts and the numerous achievements so far in such a short duration of less than a year, to have accumulated such wealth of knowledge and led to numerous developments of diagnostics, therapeutics, and preventive measures, including the vaccines. We are certainly all interested and very eager to learn more about all the facets of COVID-19 R&D. However, this event will focus primarily during these 2 days on COVID vaccines. Indeed, it's a great opportunity that we meet today under the G20 summit that is hosted and led by the Kingdom of Saudi Arabia, to share with the world such noble efforts and to express our sincere gratitude to all scientific institutions and teams participating in this forum. My very special thanks and gratitude to the wonderful team members of the organizing committee and to all participating academic, governments, and industrial partners. Thank you, and I wish you a very enjoyable forum.
Our world is engaged to a serious race to create a vaccine for COVID-19, with many obstacles and factors that delaying the vaccine's production. All of these challenges will be discussed in our forum. Now, it is our pleasure to introduce Ms. Margherita Colangelo, the co-founder and manager director of the Deep Knowledge Group, member of the forum's International Organizing Committee.
Right now. We'd like to welcome everyone to the Global Forum. We have over 10,000 people from 94 countries attending the forum today. We're very happy that you're all joining us, and we hope that you enjoy the forum. We're entering the 11th month of the pandemic, and we're all very interested in the vaccines that we've read so much about. There are 321 vaccines in development, 9 of which are in phase 3 trials. Over the next 2 days, we will have the opportunity to hear directly from 32 experts who will give us updates on the top vaccines. We will cover several types of vaccines and will discuss how they differ from each other. We will also hear from humanitarians about distributing the vaccines to people in need. This global forum will enlighten all of us and help us gain a better understanding of these vaccines. I would like to thank King Abdullah International Medical Research Center for hosting this event. I would especially like to thank His Highness Minister of National Guard, Royal Highness Prince Abdullah bin Bandar bin Abdul Aziz. I would also like to thank Dr. Al-Knawi, Dr. Azkar, Dr. Boujalal, and Dr. Almajed for envisioning a truly international forum and for graciously welcoming all of us from all around the world. Please enjoy the forum.
Thanks for this valuable welcome address. In this event, COVID-19 vaccine global challenges and prospects, we're expecting to review related researches and all advances in a proposed vaccines by the original developers. Next is the address of Dr. Muhammad Boujalal, Symposium Chairman, King Abdullah International Medical Research Center.
Thank you. Bismillah ar-Rahman ar-Rahim. COVID-19 vaccines, global challenge and prospects is the theme of the 11 KMARC Annual Medical Research Forum. It is a special honor to have the privilege to chair this important and highly anticipated forum. First, I pass my salutations and gratitude to His Royal Highness Prince Abdullah bin Bandar bin Abdul Aziz, Minister of the National Guard, for the patronage of this important forum. To His Excellency Dr. Bandar al-Ganawi and Dr. Ahmed al-Askar for giving us a chance and the opportunity to organize this meeting. We thank our partners who made this forum possible: University of Oxford and College, Deep Knowledge Group, Amara Global, and Bring Hope Humanitarian Foundations. We thank all the national and international organizing committee members who have worked tirelessly to make this forum possible. We thank the prominent speakers who are the leading scientists in COVID-19 vaccines from academia and industry, for without them, we would not have hoped to have this vaccine ready by now. In the history of mankind, only two events so far have shut down the earth: is the flood at the time of Noah that happened 6,000 years ago, and the COVID-19 pandemic. Even the World War II that happened in the 20th century did not bring the world a standstill as COVID-19 has did. The Prophet Muhammad, peace be on him, said, "There is no disease that God has created except that God, He also has created its treatment." Noah saved the humanity by the knowledge available at that time in building the ship that took him 900 years. Today, it is our prominent scientists that are saving the humanity and restoring normal life to the earth through the science and knowledge in creating these vaccines, not in 900 years, but in 9 months. We thank and we want to salute them, as well as we thank them for presenting in this forum and assuring us that these vaccines are safe and efficacious. We also want to thank the World organizations, CP, Gates Foundation, Bring Home Foundations, and others who are making all the effort and distribute these vaccines equally around the world, regardless of the color, the race, and the GDP of the countries. We thank our sponsor who have made this forum possible, especially Zoom, who made their platform available around the world to make all our attendees around the world to attend for free. We thank you all for attending this forum, and we wish you a knowledgeable forum and good learning. Thank you very much.
Now we reach the end of this session. Thank you for listening. We will proceed with the keynote presentation in 7 minutes. Thank you again. Wish you all the best.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound. Where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
المملكة استضافة قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية.
بسم الله الرحمن الرحيم. Assalamu alaikum wa rahmatullahi wa barakatuh. Good afternoon, everybody. It is my great pleasure to be here today to be moderating the keynote speakers by Professor Bill Blondren. My name is Majid Al-Graibi. I am the chairman of Infectious Disease Research Department at King Abdullah International Medical Research Center. First of all, on behalf of the organizers, I would like to extend my warm welcome to all of the guests to this forum. And I would like to extend my warm welcome to all the organizers, the Ministry of National Guard, Health Affairs, the Saudi Arabian Secretary G20, and King Abdullah International Medical Research Center, and King Saud bin Abdulaziz University of Health Sciences, for organizing this outstanding event to exchange professional and scientific experience and highlighting the global challenges and perspective of COVID-19 vaccine. I am honored to welcome Prof. Bali Blondren as the keynote speaker in this event. Professor Bali, he is a professor at the Stanford University School of Medicine and a member of the Institute for Immunology, Transplantations and Infections and the Department of Pathology, Microbiology and Immunity at Stanford University. He is adjunct professor at Emory University and Yerkes National Pyramid Centers and director of NHI Centers for System Vaccinology at Emory University in Atlanta. Today, Dr. Blondren's speech is focused on understanding the mechanism by which the innate immunity system regulates adaptive immunity and harnessing such mechanism in designing of a novel vaccine. More recently, his lab pioneered the use of system biological approach to predict the efficiency of vaccines and decrease a new molecular correlate of protections against infectious disease. Dr. Blondren's research is published in frontline journals such as Nature, Science, Cell, Natural Medicine, and Nature Immunology. Furthermore, Dr. Blondren is the recipient of numerous grants from the National Institutes of Health and from the Bill and Melinda Gates Foundation. Serves on many editorial boards and is the recipient of 2 Current Merit Awards from the National Institute of Health. Dr. Blondren serves on many advisory boards, including that of Keystone Symposia and on the External Immunology Network of GSK. He is listed on Thames Reuters list of high-cited researchers, which recognize the world's most influential researchers of the past decades, demonstrated by the productions of multiple high-cited papers that ranks in the top 1% by citations. Here, I would like to welcome again Professor Blondren, and the floor is yours.
Thank you, Dr. Al-Garoubi, for your very kind introduction. Your Excellency, President Bandar Al-Gnawi, Dr. Alaska, Dr. Colangelo, Dr. Bujalal, Dr. Ahmedjed, I thank you for your invitation to participate in this international forum. It is my distinct honor to be here with you today. Distinguished guests and colleagues, ladies and gentlemen, Assalamu alaikum. Today, I would like to talk with you about an emerging new field called systems vaccinology and the impact it is having on the advancement of new vaccines. Now, can you all see my slides? Okay, here we are in the midst of this global crisis. Yet, I think it is important for us to remember that this is not the first pandemic that's afflicted humankind, nor will it be the last. In fact, if you look at the history of interaction between microbes and humans, it reads like an epic saga. So you can see as far back as 2,000 years ago, there was the plague of Justinian, and then the Black Death somewhere around the Middle Ages, around 1400s or 1300s, killed 200 million people, practically half the population of Europe at that time. And fast forward to more recent times, just in the last 100 years, the 1918 pandemic influenza that really wiped out a significant fraction of the population. So if you now come to present day time, here we are in the midst of this COVID-19 crisis, 1.2 million deaths and 48 million cases and counting. So I would like to talk with you about warp speed immunology. Why do I say warp speed immunology? Well, in the few short months since the emergence of this virus, our advancement in understanding the nature of the human immune response to this virus has progressed in stunning speed. Let me share with you some recent insights that my colleagues and I have obtained. Now, a few months ago, around March or so, we very much wanted to understand the way in which our human immune system was interacting with this virus. We must remember that until recently, this was essentially a black box. We knew virtually nothing about what this virus was doing in the human immune system. So the approach we undertook is shown in this slide, where we applied a number of different technologies indicated in this diagram, by which we could analyze the cells and the genes and the proteins and the metabolites in the body and how these were being triggered by the viral infection. And the questions that we wanted to address are shown here. What is the nature of the immune response to SARS-CoV-2? Is there an immune signature that can predict disease severity? And of course, you understand that infection with this virus can result in diverse clinical outcomes indicated in the slide. Some patients, unfortunately, become critically ill or develop severe symptoms, whereas the majority of people develop only mild or moderate symptoms. And so the question was, what is the immune response like in these different clinical states? And also, what is the immune response in asymptomatic individuals? So these were the questions that we and a number of colleagues around the world were grappling with. And let me share with you some recent data. This was a paper that my colleagues and I published in the journal Science 2 months ago. This was a collaborative effort with many different colleagues at Stanford University, but also with the laboratory of Malik Piris and Nadine Rupail. And what we set out to do was to use a systems-based approach to analyze the host immune response to this virus. So what we discovered is indicated in this cartoon here. We found that early in infection, when I say early, within a few days of symptom onset, there was an initial burst of cytokines. Cytokines are hormones produced by the immune system. And in particular, this cytokine called type 1 IFN, type 1 interferon, which is extremely potent at fighting viral infections. So there was an initial burst of this type 1 interferon from cells in the lung that then turned on interferon response genes in other cells, other immune cells in the blood. However, this initial burst of type 1 interferon was very short-lived because as the patients progressed to more severe disease later on in infection, what happened was, in fact, this burst of interferons was attenuated. So they no longer produced interferon, so they were no longer able to control the virus, but rather what happened was that there was an excessive production of cytokines such as LIGHT, NRAGE, and oncostatin M. These are molecules that were produced in high quantities and that had previously been shown to be associated with inflammation in the lung, raising the possibility that these molecules were playing a role in mediating lung disease in the context of COVID. The other surprising finding that we observed was that we could detect bacterial debris in the blood. So we could detect DNA from bacteria and endotoxin from bacteria. Now you realize that in a healthy individual that these bacterial debris are never detected in the blood. It's only being described in the context of people suffering from sepsis-like disease. And so the question was, where are these bacterial debris coming from? And so we suspect that this is coming from the microbiota, very likely in the lung, but possibly maybe also in the gut. And this is an area that we're investigating currently. The other surprising finding that we saw in patients suffering with severe COVID was that the innate immune system in the blood was suppressed. So cells of the immune system that respond to this virus or any other virus, in fact, were very suppressed in the blood. This was a surprise because this was quite the opposite of what had been described in the lung and what we were also seeing, in the sense that there was massive inflammation going on in the lung. There was excessive production of these pro-inflammatory cytokines in the lung, and yet the immune system elsewhere was suppressed, the innate immune system. And so the question is, why is this so? And this is, again, a topic that is under intense investigation. We suspect that this spatial dichotomy in the immune response, in other words, that there's excessive inflammation in the lung, but suppressed innate immunity elsewhere, might be a homeostatic mechanism that's evolved to prevent excessive damage, systemic damage during the course of a tissue infection, such as COVID infection. All right, let us now talk about warp speed vaccines. Of course, you understand that this is a term that's commonly used to describe the incredibly rapid pace at which COVID vaccine developments is happening. But I'd like to step back and remind you that vaccines are incredibly powerful public health tools. And if you look at the history of vaccines, really, if you look at average life expectancy since the dawn of time, so this is 3 million years ago, what's remarkable is that it's hovered at around 20 or 30 years of age. But then somewhere in 1796, a remarkable event happened, and that was the famous experiment in human immunology done by Edward Jenner when he inoculated this young boy with cowpox and showed that this resulted in enhanced resistance to smallpox. Now, if you look at life expectancy shortly afterwards, you see that there's this tremendous increase in life expectancy. Now, of course, this is not solely due to vaccination. There have been stunning improvements in public health and sanitation and clean water and so on, but vaccines have contributed tremendously to enhance life expectancy. And the legacy of Jenner, as you know, lives on. Smallpox was eradicated in 1980, 40 years ago, and some 20 plus licensed vaccines have saved hundreds of millions of lives. So vaccines are incredibly powerful public health tools. So where are we in the COVID vaccine landscape? We're going to hear many exciting talks from my colleagues in the seminars and talks that will follow, but I just want to give you a broad perspective of where we are. So the COVID vaccine landscape could be summarized in this diagram. There are several hundred plus candidates that are being tested in preclinical models. Several, 40 odd, have already been tested in humans in phase 1, phase 2 trials, and I believe there are some 11 that are being assessed in phase 3 trials, and these are indicated here. What's remarkable is that this has progressed in warp speed. In other words, from here to here has happened in a matter of months. You have to remember that typically this process of going from a vaccine concept to testing in phase 3 trials can take years, sometimes 10 years, perhaps even longer, up to 15 years. So what's happened is truly a triumph in vaccinology with COVID-19 vaccines. Now, one point to remember is that the vast majority of these vaccines are predicated on the idea that inducing neutralizing antibodies against COVID will be efficacious in mediating protection. And certainly this is a very sound idea, because if you look at any number of other vaccines that have been licensed, and I'm showing you just a handful here, they've all been developed on the principle that vaccines can induce an antibody response that can fight infection, that can prevent or fight infection. And so this is really based on what we call serological correlates of protection. So for example, it's a single value of some antibody titer, ELISA titer, or microneutralization titer, a threshold that is defined as being important, or what you need for protection. So for example, for influenza, you need hemagglutinin titers of 1 in 40. And for Haemophilus influenza B, you need 0.15 microgram per mil of ELISA binding titer. But yet, it is important to remember that our human immune system is very complex. It is much more complex than simply antibody titers. You see, I show you this example of T cells and B cells. And the Lasker Prize last year was given to my friends Max Cooper and Jacques Miller for their discovery of T and B cells. And you realize that B cells make antibodies, they become plasma cells and they make antibodies. T cells can help the B cells make antibodies, but they can also act as killer cells that can kill virally infected cells. I mention this to you because as we think about COVID vaccines, it is important to think of how we might engage the multiple arms of the host immune system. So how do vaccines work? Well, vaccines work through different mechanisms. Firstly, when the vaccine enters the body, there's a very rapid response within the first few minutes to days after vaccination, which is what we call the innate immune response. This is an evolutionarily ancient system of host defense that is highly conserved in all creatures on the planet, from frogs to fruit flies to humans. This is followed by the adaptive immune response, which is what we've talked about, antibodies and T cells, which is evolutionarily more recent than the innate immune response system. And this takes days and can last up to several decades. And then a property of this adaptive immune response is that if the immune system encounters the vaccine or the pathogen at some point later on, maybe several decades later, it responds in a heightened fashion, a much faster response and a more pronounced response. And so, as I mentioned, the adaptive immune response is mediated by B cells that make these antibody molecules and T cells that can kill virally infected cells. Now, why is this important? This is important because it turns out that these two arms of the immune system can collaborate to provide a synergistic protection against a pathogen. So I want to show you one example of this. This is a study that we published in Nature Medicine earlier this year, in which we did an experiment in non-human primates to evaluate different types of vaccines in their capacity to protect against simian human immunodeficiency virus, so a monkey HIV virus. And the groups are shown here. We had a control group in which we only injected an adjuvant. An adjuvant is just a substance that is normally added to a vaccine, but it by itself doesn't really mediate protection. That is the current thinking. In the second group, the red group of animals, we immunized them with a protein from the HIV virus, the HIV envelope protein, together with an adjuvant. In this case, this vaccine, the red vaccine, induced only an antibody response. And then in the third group of animals, we had viral vectors, much like what you're going to hear about at the symposium, that express the HIV antigen, but also, and these viral vectors were very efficient at inducing a very strong T cell response, but also, like in the red group, we had the same HIV antigen with adjuvant that could induce the antibody response. So you see, the blue group was a vaccine that could stimulate both the T cells and the B cells, but the red group, only the B cell response. And what did we find? What we found was something quite remarkable. When we looked at what is the quantity of antibody you need for protection against HIV infection in monkeys, we could define a threshold, about a titer of 300 there. Any animal that had a level of antibody above that threshold was protected. What about the animals that didn't have reached that threshold? Well, many of them were infected, shown here. But you see, there was a group of animals that were still protected, even though they hadn't reached that threshold. Why is that so? Well, it turns out that this group that were protected were practically all animals that had strong T cell responses. So in other words, T cells induced by the vaccine have reduced the threshold of neutralizing antibody you need for durable protection. And I do mean durable protection, because when we re-challenged these monkeys that had survived the initial infection, 6 months later, even at that time, the monkeys that had received the T cell and the antibody vaccine survived much better. So I believe this is important lessons as we think about how best to design the next generation of COVID vaccines. We should be looking certainly at antibodies, neutralizing antibodies, which is the primary means by which vaccines protect. But I believe we should also be keeping an eye on learning how to induce strong T-cell responses. So I've told you that the immune system is comprised of multiple arms, T cells, antibodies, and the innate. How do we evaluate this in the context of vaccines? I want to tell you about the new frontier in vaccines called systems vaccinology. It is effectively using the tools of systems biology, the so-called tools in the advent of the omics revolution, where you can measure the behavior of genes and metabolites and cytokines and the epigenome in humans in response to a vaccine. And this is an area that my colleagues and I have been working on for the last several years. Our initial work on this concerned this vaccine called the yellow fever vaccine, which many of you recognize is a vaccine that has been given to 600 million people globally. It's one of the most successful vaccines. It is 99.9% efficacious, and a single immunization induces antibodies and protection for several decades, for up to 30 years, and a single immunization induces robust T-cell immunity. So we thought about 10 years ago, this was a remarkable vaccine, a gold standard vaccine. How does it work? And it turned out there was very little understanding of how exactly this vaccine was stimulating the immune system. And I always think that this is the sort of aspirational goal that any vaccine manufacturer should have as they develop vaccines against COVID and other such pathogens. So the experiment we did was the human immunology experiment. This was the paper that was published in 2008, where we vaccinated people with this yellow fever vaccine. We collected blood samples before vaccination and at different times after vaccination. And then what we were able to do is to use genomics to analyze the change in the expression of genes in blood cells in response to this vaccine. We were able to see within the first few days, within the first 7 days or so, there was a very strong expression of genes induced by the vaccine that correlated with the subsequent immune response, the T-cell response or the antibody response. Using machine learning techniques, we were able to define sets of genes, we call them ABC or BFG or ADE, groups of genes that could predict the strength of the antibody response several months later or the strength of the T-cell response several weeks later. So this was the first example of using the systems biology approach to analyzing the immune response to vaccination and to use machine learning to predict the response to vaccines. And we felt that this was very important because it would allow a means to rapidly analyze whether a certain vaccine was going to induce a strong response or not. Since that time, what we've done, and when I say we, it's not just our lab now, there are many labs across the world using this approach, is to use not only to look at the genes, but also the epigenome and the proteome and the metabolome and the cell populations to perform an integrative analysis of multi-omics data to see how the immune system responds to a vaccine. And so this was a study that we published about 3 years ago in which we used this approach to define early signatures that could predict the response to the shingles vaccine in humans. We've also extended this approach to the seasonal influenza vaccine, and I'm just showing you an example here where you could do systems analysis of response in humans who received the seasonal influenza vaccine by analyzing the change in the expression of genes early after vaccination. You could define these gene signatures that then you could apply into machine learning techniques to come up with signatures that predict the antibody response that happens 1 month later on. And you can then design a PCR-based chip in which you can actually apply this and test this predictive signature in several trials. Now, of course, you might say 1 month is okay, but we're very interested in the durability of the vaccine responses. And this is a topic that all of us are interested in as we all anxiously await the first generation of COVID vaccines. How long lasting is their protection going to be? And this is something that we have addressed using the systems approach. So I'm showing you an example here from a paper that we published back in 2015. This is with the seasonal flu vaccine. This is the antibody response to seasonal flu. You can see these subjects here respond well at 1 month, but then at 6 months or 180 days, their response is diminished below the threshold needed for protection. However, there are some subjects who do seem to maintain their antibody titers above that threshold. And so the question was, can you predict at baseline or day 1 or day 3 or day 7 what the durability of this response was going to be? And indeed, that was what we set out to do. I don't have time to go through the details, but we could apply the tools of systems biology to define a signature in the blood at day 1 or day 3 or day 7 that we could use to predict the subsequent antibody response to this vaccine. Since that time, we have extended this and to look at other vaccines to see if the signature can predict the durability of other vaccines, and the results are very encouraging. So we feel is an important area that is currently being investigated. And then finally, we can apply systems approaches in clinical trials to look at efficacy, not just how strong an immune response a vaccine is going to have, but also how well is it going to respond. And so this is shown here, for example, where you can, this is with the GSK's RTSS malaria vaccine that is currently being developed. And you can immunize healthy humans with the RTSS vaccine at day 0, day 28, day 56, and then collect blood samples at all these different time points. What you can do with malaria is that there's a challenge model where you can infect people deliberately with Plasmodium falciparum. This is a person in the trial. He is being exposed to mosquitoes in this jar that have been infected with Plasmodium falciparum. As it turns out, 50% of the people developed disease with this vaccine. The question was, could we predict that by looking at the changes in the genes being induced by this vaccine at early time points? And indeed, we were able to define a signature that could predict vaccine outcome. So let me, in the last part of my talk, I want to say, okay, no doubt the systems approach is generating a lot of data. And Sidney Brenner, one of the greatest biologists who ever lived in the last century, said about systems biology in general that we are drowning in a sea of data and thirsting for knowledge. He then went on to say systems biology in general is low input, high throughput, no output biology. Okay, so the question is, what mechanistic insights can this kind of approach yield? And I want to give you 2 examples. One concerns our second genome, also known as the microbiome. And what we had seen in a study that we did with the seasonal flu vaccine was that the genes that were being induced by vaccination, amongst the genes, there was this gene called toll-like receptor 5, TLR5, which is a receptor of bacterial flagellum. This was induced within a few days after vaccination, and it correlated with the antibody response that happened at 1 month. And we were puzzled by this because, of course, flu is a virus. Why should a viral vaccine be inducing the gene encoding toll-like receptor 5? So we then investigated this, and that this is where we are. You see, this is the expression of toll-like receptor 5, 3 days after vaccination. This is the antibody response at 30 days. There was a beautiful correlation. We then went to a mouse model where we could immunize mice that were genetically deficient in this receptor, TLR5 knockout mice, and show that the antibody response to flu vaccine in those mice was greatly diminished. How could that be so? We thought, well, if it's recognizing bacterial flagellum, perhaps the microbiota is important. So we treated the mice with antibiotics, and we can see that there was greatly diminished antibody responses, showing that the microbiota was critically important in inducing antibody responses to flu vaccine. So that was the first example to show that the microbiota was important. Of course, that was in mice. We were interested in humans. So we asked, is the microbiota important in humans in vaccine immunity? Should that be something we should be thinking about as we develop COVID vaccines against different populations in different parts of the world? So together with Nadine Rupail at Emory University, we did a human trial in which healthy humans were given a cocktail of neomycin, vancomycin, metronidazole for 5 days to ablate their microbiome. They were then given the flu vaccine, and we analyzed the immune response at various time points, all the way up to 1 year. And the results of that study were published just a few months ago in Cell, where we showed that indeed the gut microbiome was critically important in mediating innate and adaptive response to influenza vaccine, but only for people who had very low baseline titers. If there was strong memory built up, that could withstand even the most severe dysbiosis of the microbiome. So, ladies and gentlemen, I hope I've convinced you that this field, this emerging field of systems vaccinology, allows you to probe the immune system and how it is responding to viral infections in great detail. So you could start by using these omics technologies to analyze the innate and adaptive response, working together with bioinformatics specialists and computational scientists. You could define signatures that predict how well a person is going to respond to a vaccine. But it's not just signatures. You could devise exciting new hypotheses about exactly how the vaccine is working, to test those hypotheses in animal models that then feeds back into designing better vaccines. So it's an iterative process. And the example I gave you was with the microbiota that started off with the defining the TLR5 in the seasonal flu vaccine study. And in the last few minutes, I want to tell you another example that's resulted from systems vaccinology, and that concerns this concept of immune memory. We all know about immune memory. We all know that immune memory, the seat of immune memory, is in what we call memory B cells or memory T cells, and this we've known for decades. However, recent work suggests that the innate immune system that I mentioned, the evolutionarily ancient immune system that is highly conserved in all creatures, may also have a form of immune memory. And we believe this is coming from the work of my colleague Mihai Netia and others, that we believe that this innate memory is mediated through what we call epigenetic programming. Now, what is epigenetic programming? This is exactly what this is. Here, our genes are encoded in the chromosome, as you know, and our chromosome is like a ball of spaghetti that's wrapped up tightly. It's wrapped up tightly around these orange circles called histones. When the chromosome is wrapped up, the DNA is inaccessible, so the genes cannot be expressed. However, certain stimuli can result in unwinding of this ball of spaghetti so that the chromatin becomes accessible, like here. So now the genes can now be expressed and proteins can be made. So what we discovered was that what we did was to analyze the epigenome in the context of influenza vaccination. This is using this technique called epitoph. For the non-specialists, don't worry about this. This is a fancy technique whereby we can look at the single cell level at all the different cells in the immune system, at all the different histone or epigenetic markers to see which genes are tightly wrapped up and which genes can be expressed. And what we discovered was something quite stunning. We discovered that epigenomically, you can define different types of immune cells in a drop of blood, but more importantly, after vaccination, in this case, we used the seasonal influenza vaccine. In fact, there were dramatic changes in the epigenome in these innate cells, monocytes, myeloid cells, plasmacytoid dendritic cells. And you might say that's not surprising because the innate system is responding, but in fact, this lasted for several weeks, sometimes months. And this is shown here. Up to 6 months, there were these changes in the epigenome of these myeloid cells. What does that matter? Should we be worried about that? And yes, it turns out that if you isolate those cells and you ask, can those cells respond to TLR stimulation or microbial stimulation in tissue culture? In fact, they have a diminished capacity to produce pro-inflammatory cytokines in response to this TLR stimulation with either bacterial stimuli or viral stimuli. So this was surprising. So 30 days after vaccination with seasonal flu vaccine, the cells in the blood, the innate cells, are unable to respond effectively in response to stimulation. And this innate refractoriness or impairment, it's really the nadir is at around 30 days, but in many subjects it can last for up to 6 months. So it's quite a long time after vaccination. So to look at this in more detail, we used what we call this single cell ataxia or ataxia that really gives you a deep look at the regions of the chromatin. And we could see that this region, this AP1 loci, again, for the non-specialists, don't worry about this, but basically it is to show that the epigenome is being modified after flu vaccination. This was quite a surprise to us because some previous work by Mihai Netia and colleagues had suggested that vaccination with BCG, in fact, has the opposite effect. It programs the immune system epigenetically to opening up its chromatin loci so that it can respond more vigorously with other stimuli. So we're still trying to figure this dichotomy, but the model that we have is that this innate immune system, this epigenetic reprogramming, is contextual. When you have the BCG vaccine or live attenuated vaccines like yellow fever, strong vaccines that induce a strong immune response, or BCG, then you have opening up of the chromatin loci, more IL-6, more pro-inflammatory cytokines. But when you have vaccines that do not have adjuvants like the seasonal flu vaccines or other non-adjuvanted vaccines, perhaps you have a closing up. But in fact, what we see is that certain adjuvanted vaccines can, yes, result in closing up of this chromatin loci, but they can also open up other loci involving antiviral resistance. And I summarized this in this cartoon where, for example, influenza vaccines that are given with certain adjuvants can actually cause epigenetic reprogramming to induce closing of this certain chromatin loci that results in decreased pro-inflammatory cytokines, but opening up with other chromatin loci that may lead to enhanced antiviral resistance. So this may have some implications in terms of developing new adjuvants that could program the innate immune system to have enhanced resistance to a wide range of different viruses, so epigenetic adjuvants. So the last two slides, let me conclude. I've talked to you about what we've learned about the human immune response to COVID infection. I've mentioned to you that there are these exciting new advances in systems vaccinology that's providing a new aperture into understanding how vaccines work and defining correlates of vaccine immunogenicity and protection. So I come back to where we started off with vaccinology BC. BC meaning not before Christ, but before Corona. This, as you know, is a very protracted process that can take several years, sometimes 10 years or more. And it's certainly very expensive, $500 million to $1 billion, it is estimated, to bring a vaccine from the research phase all the way up to licensure. Now, vaccinology in the time of COVID-19 has been a remarkable triumph. I mean, I'm in awe at what the vaccine manufacturers in industry, government, and academia have done. It is remarkable, and it's truly inspiring what's happened in a matter of months to bring so many promising candidates to phase 3 clinical trials. And we all hope that many of these will turn out to be safe and effective vaccines. But as we look towards the future, 2-3 years down the line, as we get a better handle on COVID-19, how should we view vaccinology, landscape of vaccinology? Should we go back to business as usual here? Or is there anything we can learn from the crisis that we have gone through? I submit to you that vaccinology after Corona will benefit enormously from integrating systems vaccinology in clinical trials. And these are examples of how this could be done. For example, in phase 2 or 3 trials, one could perform retrospective nested case control studies to take samples from people who were protected and people who were not, and analyze if there are signatures early on that could predict the outcome and what these signatures would inform about mechanisms of immunity. I also mentioned established human challenge models like the one in malaria. There are discussions afoot to perhaps design a human challenge model for COVID-19, huge ethical issues that one needs to work through, but that's a possibility. And then vaccines with known correlates. Let us say we define a certain threshold of neutralizing antibodies, so 300, that seems important for protection. Well, can we predict the durability of that? I showed you the example of how systems vaccinology can indeed have a signature a few days after vaccination that could predict durability. Now, wouldn't that be useful? You don't have to wait for 1 year or 2 years to say this is what the titer is going to be. You would be able to predict that in the first few days or weeks. So with that, let me thank the number of people who have helped and collaborated. This is only a partial list. The human immunology work to understand the human immune response to COVID-19 was led by Prabhu Arunachalam, Farian Wimers, and Thomas Hagen and Ukraine Feng. This was a collaboration, as I said, with Professor Malik Piris's lab and with Nadeem Rupail's lab at Emory and the many other people shown here. So I thank you for your attention.
Thank you, Dr. Thank you, Professor Belli, for this great presentations. We'll like to open the floor for questions from the audience. If you have any questions, you can send it through the chat at Zoom. Until we receive questions, Professor Belli, people are wondering all the time after the people infected by COVID, are they immune after the disease or not? Let me give you that question because a lot of people is wondering about having immunity after infections, and how can the vaccine later on be effective to boost the immunity?
Yes, thank you. This is an extremely important question. And of course, there is a basis of vaccination that in the history of vaccines, we all look at natural infection and ask the question, does natural infection induce protective immunity? And of course, there are many examples of natural infection inducing lifelong immunity, like measles. You know, some of us have had measles when we were children. We will never again get measles because our immune system remembers them for a lifetime. Chickenpox, the same thing. And in fact, many live viral vaccines, such as the measles vaccine or chickenpox vaccine, do precisely that. Now, what about COVID-19? What do we know about natural immunity? Well, it's still early days, but there are some clues that we can have. The first set of clues comes from animal challenge models. There are studies in which scientists have infected non-human primates with SARS-CoV-2, and then after a month, reinfected the same animals with SARS-CoV-2 and showed significant protection. It's not sterilizing immunity, but the quantity of virus is down 10,000-fold. So it's really significant protection. But it is only 1 month. So the question is, what happens longer term? Then the second set of evidence comes from human challenge models, not with SARS-CoV-2, but with the common cold virus. Some of these common cold viruses are also coronaviruses, like OC49. This was done in England sometime in the 1980s and 1990s, where in the hospital setting, humans were deliberately infected with the common cold virus, and then about 6 months later or a year later, these same humans were given a second dose of the common cold virus. And there, the indications were there was protection in some of the people, but this seemed to last for a few months, I would say. So again, showing that there is significant protection for some period of time. Not clear for how long, but for some period of time. And then finally, of course, it is with analyzing the antibody titers in SARS-CoV-2 infected people. There are many, many reports that are now coming out. Some earlier reports suggest that the antibody titers wane rapidly in the first few months. But I have to remind our friends that all antibody titers wane. Even with the world's best vaccine, what goes up must come down. The question is, how slowly does it come down? You know, the half-life of antibody response to measles infection is 3,000 years. But the half-life of antibody response to COVID-19, we don't know because the virus has only been with us for a few months. So clearly this is an important question. The data is emerging. The data, some other studies suggest that there could be persistent antibodies that last for some months, but you know, some months is not years. So the question is, we have to be open. We have to do this in a very rigorous way and analyze longitudinal samples to see how long-lasting natural immunity is.
Thank you for answering these questions. Also, we have questions through the Zoom. What type of cells we are including and how strong could be high effective T cells response might trigger as high humoral T and B cells response blurring immunity.
Yeah, so was the question about what type of T cell responses and
yes,
yes,
the humoral T cells and B cells.
Yeah, well, I think the nature of the T cell response to COVID-19 infection and vaccination is a topic of great interest. We know for sure that in infected people there is quite a robust T cell response. And we also know that this may be quite durable. But the key question is what impact, if any, do the T cells have in controlling the virus or in contributing to immunopathology? And as the questioner asks, what kind of T cell response? I didn't go into this, but T cell responses come in different flavors. There are T helper type 1, T helper type 2, T helper type 17, and many different flavors based on the cytokines they produce. It's not yet clear what kind of a T cell response. Generally, it is believed that the Th1 response is helpful for controlling viral infections, and there's some suggestion that maybe a Th2 response is not desirable, although I'm not sure what the data for that is. So I think, again, it's early days. We're not quite sure what kind of a T cell response, but I would say by any, I think having a strong cytotoxic killer T cell response is absolutely an important and desirable goal. As I said, they can work hand in hand with the neutralizing antibody response to confer protection.
That's amazing. It seems that we come to the end of this presentations. I would like to thank everybody for attending the sessions, and I would like to give a special thanks to Professor Bill Brundry for his great keynote presentations, and I will recommend everybody to go and to see his recent publications, which is kind of very interesting to read about the immunity and COVID-19. Thank you so much, and we look forward to seeing you in the next session. Thank you so much.
Thank you.
Bye, Dr. Bali.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound. Where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
المملكة استضافة قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound, where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
مملكة استضافة قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
I'm Ahmed Salban, a senior immunologist, vaccinologist at the General Institute and fellow of clinical medicine and Kellogg College at the University of Oxford. I would like to start by sending my warmest greetings and welcoming from Oxford to the conference organizers, sponsors, bright speakers, and the audience from all over the world. It's a great honor and pleasure to welcome you all. Safe and efficient COVID vaccine can save millions of lives. The global collaborations and mutual assistance beyond borders are key to tackle this health crisis and facilitate equitable access and distribution of these vaccines to protect people in all countries. Scientists from all
Thank you very much for the introduction and also the opportunity to speak to you this morning.
Thank you very much.
So I'm going to start by just telling you a little bit about the technology that we use. We have used this to make many different vaccines in the past before using it in COVID-19. So ChAdOx1 is a non-replicating simian adenoviral vector vaccine, and in this case it expresses the spike protein of the novel coronavirus. So the adenoviral vector is non-replicating because the E1 gene has been removed. So that means that we manufacture it in a cell line which supplies the E1, but when we use it to vaccinate either a person or an animal, the virus can't replicate inside the first cell it reaches. It can only express the antigen that we've added into it, and it does that at a very high level, but it can't spread into any other cells after that first cell has been infected and the antigen has been expressed. So it's very safe to use, even in people who have a compromised immune system. We use a simian adenovirus rather than a human adenovirus to start from, because this avoids the issues with pre-existing immunity to human adenoviruses. It's known that if you use a common human adenovirus, some people will have neutralizing antibodies to it already before you immunize them, and that reduces the ability of the adenovirus to infect cells and express the antigen, and it reduces the immunogenicity of the vaccine in those people. So we avoid that problem by using an adenovirus which is genetically very similar but doesn't circulate amongst humans, so there are no pre-existing immune responses to it. And because the vaccine antigen is encoded in the genome of the adenovirus, rather than being part of the surface of the virus, it means that the structure of the virion is always the same, whichever the antigen is that we're encoding. And so the manufacturing is the same in every case. So it's a true platform technology. If we can work out how to manufacture one of our viral vector vaccines at large scale, we can apply that same manufacturing process to any other one using the ChAdOx1 construct.
We know it induces strong B and T cell responses after a single vaccination. They start to arise within a week of vaccination. But as I will also show you, if we give a second vaccination with the same viral vector, we can increase those immune responses. And before we started using this in our clinical trials against COVID-19 in April of this year, we'd already conducted 12 phase 1 studies of ChAdOx1 encoding other antigens, including in particular MERS coronavirus spike protein, which is obviously very relevant to the development of another anti-coronavirus vaccine. And we don't have any vaccines against human coronaviruses. Early work on SARS didn't get very far. There were problems with enhanced disease after a challenge of vaccinated animals. And MERS vaccine development is still at a relatively early stage, and there aren't any attempts to make vaccines against the 4 seasonal human coronaviruses. However, there are veterinary vaccines against coronaviruses that infect either cows or chickens, and these are very successful vaccines. So the prospects of making a vaccine against a coronavirus are probably quite good. However, there has been this problem of enhanced disease after challenge, which was seen in SARS vaccine development in some animal models, and it's also been seen in another veterinary coronavirus, which is feline infectious peritonitis. And here, a recombinant live virus vaccine induced antibody-dependent enhancement of infection after the vaccinated animals had been challenged. So this is a phenomenon that we have to be aware of and take into account when we're planning our new COVID vaccines. And we need to do preclinical studies before getting into the clinical trials to guard against the fact that we might potentially see enhanced disease after challenge. So what happened in the early SARS vaccine development was that vaccinated animals had a Th2 skewed immune response, resulting in eosinophil infiltration of the lungs, and in other studies, antibody-dependent enhancement of infection, where non-neutralizing antibodies resulted in enhanced entry of the virus and a greater degree of severity of the infection. So when we plan our SARS-CoV-2 preclinical studies, we need to look for the induction of neutralizing antibodies as well as non-neutralizing antibodies. We need to understand whether the immune response is Th1 or Th2. And remember, it's the Th2 skewed response that was associated with eosinophil infiltration and a bad outcome. And we need to look for any evidence of enhanced disease after challenge of infected animals, and we can look for that by a number of different methods. The clinical scores of the animals, the virus titers in the lungs, the radiographic study of the lungs, and then finally histopathology after necropsy, comparing to a control group of non-vaccinated animals. So we planned 4 preclinical studies in the early part of this year. The first one, which was conducted at NIH, was published. We had this information before we started our clinical trial, and in this we vaccinated non-human primates, rhesus macaques, with either 1 or 2 doses of our vaccine, ChAdOx1 nCoV-19, and we measured neutralizing antibodies as well as ELISA titers, and we looked for the T-cell response by ELISpot. The monkeys were then challenged with a very high dose challenge, intratracheal, which puts the virus straight into the lungs, as well as challenged in the upper respiratory tract through the oral, intranasal and ocular routes. So this is a very thorough challenge. And what we saw there was that the clinical signs were reduced in the monkeys who were vaccinated. There was protection against pneumonia in the lungs. The vaccinated animals had a significantly better outcome than the non-vaccinated animals, and we certainly weren't seeing any evidence of enhanced disease. And we went on to do 3 more studies in non-human primates, again at Porton Down, where we used a different method of examining the lungs after virus challenge, doing CT scans, which is the technique that's being used to look at lungs in humans with COVID-19. And again, looked at the virus in the bowel and looked at histopathology of the lungs after challenge. And we also had 2 ferret studies at Porton Down and at CSIRO in Australia. And in none of these did we see any evidence of enhanced disease. And I'm very pleased to report that in all the other studies that have been done of candidate vaccines going into clinical trials, we're not seeing enhanced disease. So this really does seem to be a theoretical concern, but it was one that was important to address thoroughly before getting into clinical development of the vaccine. So we managed to start our clinical trials 104 days after we got the sequence. This graph shows you the increase in the number of cases being detected in the world. And when we decided to go ahead and start making the vaccine, the outbreak was still at a very early stage, and we had no idea how far it was going to spread. But we decided that if we were going to do anything, we needed to start straight away. And so we started on some preclinical work. We got straight into GMP manufacturing in our own facility at the university, generated the initial preclinical immunogenicity results, and were then ready with our GMP vaccine by the 23rd of April to start vaccinating in our clinical trial. So we published the first clinical trial data in July in the Lancet, and this was of a phase 1 trial of 1,077 participants, healthy adults aged 18 to 55 years, who are randomized to receive either ChAdOx1 nCoV19, which is now known as AZD1222 since being licensed to AstraZeneca, or the control group received a meningitis vaccine. And we used a comparator vaccine rather than a saline placebo because we wanted the recipients to be truly blinded to the vaccine they have received. And when you give people a saline placebo, the reactogenicity of that is very low, and it was likely that our intelligent population of volunteers would have worked out which vaccination they had received, and we didn't want them to be able to do that. We also had a small group of people who had 2 doses of the vaccine given 4 weeks apart. The vaccine was tolerated. The reactogenicity was as it normally is for this type of viral vector vaccine given at this dose. We found that giving paracetamol at the time of vaccination for the first 24 hours would reduce some of those symptoms without having any effect on the immunogenicity of the vaccine. There were no serious adverse events, and reactogenicity was lower after the second dose of the vaccine compared to the first. We started to look at the immune responses, and here we're looking at the IgG responses by ELISA titer. This is the group with the dots shown in red of the vaccinees who received the nCoV19 vaccine, and you can see by 14 days after vaccination we're seeing an onset of antibody responses which increased somewhat by day 28 and are still present at day 56. But if we gave 2 doses of the vaccine, we saw an increase after the second dose. And we're comparing this here to a group of convalescent plasma samples, people who are PCR positive for COVID-19. The ones in light green are mild cases, and in dark green, severe cases who tend to have higher antibody titers. And on the red stars are the serum samples that were then taken into neutralizing antibody assays, which I'll show you on the next slide. And you can see that these ones are the ones at the higher end of the convalescent sample group. We did see that some people were were seropositive prior to vaccination. A small percentage were, and the vaccine was safe in these participants as well, and the vaccination increased their immune response. Now we need to measure neutralizing antibodies, and there are many different ways of doing this, and we're still waiting for the ability to compare neutralizing antibodies by different studies, and that will require different studies to use the same method for measuring neutralizing antibodies. This is a very stringent IC100 assay, where we look at the ability to completely neutralize the virus. And this is what we see after 1 dose of the vaccine at 28 and 56 days compared to the PCR positive samples that were on the previous slide. And we see that if we give a second dose of the vaccine at 4 weeks, we then increase these neutralizing antibody titers. We also see higher neutralizing antibody titers in those symptomatic patients than the asymptomatic healthcare workers who were seropositive when screened for vaccination. There are different assays for neutralization. That was an IC100 assay. This is a PRNT IC50 assay, which will give us a different readout on the y-axis. Again, we're seeing the induction of neutralizing antibodies 28 days after a single vaccination. We can also use a microneutralization assay, which is a more rapid assay, again an IC50 readout here. But with the microneutralization assay, we can also report as an IC80 or as an IC90 readout. And depending on which of those you choose to use, you will read off a different number from the y-axis. So when comparing neutralization assays between studies, you need to be aware of whether it's measuring IC50, 80, 90, or 100, because all of those different methods will give you a different readout. possible to use a pseudovirus to look at neutralization assays, and we did that too. Again, this is an IC50 assay, and here we're comparing the response after a single dose to the response to 2 doses against a group of convalescent plasma samples, shown here with a very wide spread in those samples, again with the patients who had the severe disease tending to have the higher neutralizing antibody titers as well as the high IgG binding titers. We looked at T-cell responses, and as we would expect with an adenovirus vector vaccine, we see rapid onset of T-cell responses. It's an ELISpot assay measuring both CD4 and CD8 responses. We see that it increases to a peak at day 14, is maintained at day 56, but the T-cell response is not increased by giving a second dose of the vaccine at 4 weeks, whereas the neutralizing and the binding antibody responses are. From the phase 1 study, we went into a phase 2 in 2 different age groups because it's really important to look at the performance of this vaccine in older adults, and we will be able to report some data on these phase 2 trials shortly. We then commenced a phase 3 trial in the UK at the very end of May, beginning with the 18 to 55-year-olds, and then as we had safety data from the phase 2 trial, we added in the older age groups so that the study became a phase 3 in adults of 18 years and over. We also started studies in Brazil and South Africa. It's very important to look at the performance of the vaccine for both safety, immunogenicity, and also efficacy in different populations around the world, because both ethnicity and the exposure to different microorganisms in different parts of the world can have an effect on vaccine immunogenicity and therefore on efficacy, and we need to understand that. So we are working with partners in Brazil and South Africa who are very experienced in running clinical trials to look at the performance of the vaccine in studies there as well. AstraZeneca took on the license to the vaccine and have been working extremely hard on large-scale manufacturing scale-up. They're also sponsoring the phase 3 trial in the US, which is 30,000 adults, and again in adults over the age of 18 years. So we have multiple phase 3 trials running now as we come to the end of the year, and as are many other vaccine developers expecting to see some efficacy readout before too much longer. And in these trials, the majority of the participants will receive 2 doses because we don't yet know the correlates of protection against SARS-CoV-2, and we know that 2 doses give us a higher antibody titer than 1, so we're aiming for the best results by giving 2 doses of the vaccine. AstraZeneca have an exclusive license to this vaccine, but they are working with a large network of partners with collaborations and sub-licenses to arrange for large-scale manufacturing and distribution of the vaccine all around the world. They have manufacturing ongoing in multiple different countries with the aim of being able to supply different parts of the world. And that includes the Serum Institute of India, who have extremely large-scale capacity for vaccine manufacturing and will be able to supply vaccines to Gavi for use in low- and middle-income countries. AstraZeneca are sponsoring a number of the clinical trials as well, with the US trial in progress. Japan has actually initiated, and there are plans for a trial in Russia also. Obviously, other vaccines are in advanced development as well, and we're going to hear more about those, so I won't say any more just now. But we'd just like to move on to what we need to achieve for vaccine licensure, because it's not just the efficacy result. We do need an efficacy result to be determined in 1 or more phase 3 trials, but that we then also need a vaccine safety and immunogenicity database from the relevant populations in which we're hoping to license the vaccine. And to begin with, for our trial, that will be adults over the age of 18 years. We've also initiated studies in people living with HIV, both in South Africa and in the UK, to look at the safety, immunogenicity of the vaccine in that subgroup of the population as well, so that they may in future be added to a vaccine license. And we are conducting reproductive toxicology studies, which will be required before we could conduct any clinical trials in pregnant women, which are another important group to be able to protect against infection with this virus. As yet, we haven't conducted any clinical studies in children. It's now known that the risk to children from SARS-CoV-2 is considerably lower than it is in adults, and we will wait until we have an efficacy signal from the phase 3 trials before deciding to initiate any pediatric vaccinations. So that's something to come a bit later on. But once we have the safety and immunogenicity database, from the clinical trials, we can then apply to a regulator for emergency use, and the regulators will make their decision about whether to use this vaccine or not. That will take some time. So the question everybody always wants to know is, when will the vaccine be ready for rollout? We have to achieve all of these things before the vaccine can be used on a wide scale, and it's not possible to predict the exact timing for any of them. To speed up the regulatory approval, clinical package in order to have as rapid a licensure procedure as possible, but that's still not going to be an overnight process. So there are very, very many people involved. We have a huge group from the Jenner Institute and Oxford Vaccine Group and our collaborators all around the world working on this, as well as AstraZeneca. So I'm not going to attempt to name all of them. I'm just showing you a few of the different types of the people that we're working with that have contributed to this study. And I'll finish there. Thank you.
It's very nice. I think and the pandemic, the control. So
DNA assay, and a very similar picture that there were very low levels of neutralizing titers at baseline and in placebos. And following vaccination, a single-shot vaccine raised neutralizing titers in the vast majority of individuals, with the titers shown here. And similar titers were seen in the older adults over 65 in this very small data set so far. We also looked at T-cell responses, and as you would expect for an adenovirus vaccine, this vaccine raised CD4 T-cell responses shown here in the vast majority of individuals, both in the younger group and the older group. These were entirely Th1 responses, and essentially no Th2 responses were observed. The vaccine also raised CD8 T-cell responses in at least the majority of individuals. So the phase 3 study is a randomized double-blind placebo-controlled trial for efficacy and safety. The trial is a target enrollment of 60,000 individuals. This started on September 21st. It's a 1-to-1 vaccine placebo randomization. This is testing a single-shot vaccine of 5 times 10 to the 10th viral particles. And another feature of this vaccine is that there's no requirement for a frozen cold chain, and it's opening in the United States and South Africa and multiple countries in Latin America. There will be a second phase 3 study that will start in the next couple of weeks that will test a 2-shot version of this vaccine. So we're hoping this vaccine will give a readout both in a single-shot format as well as in a 2-shot format. So I'll just close with a few perspectives that are more general. That vaccine development is proceeding faster than for any other pathogen in history. Safety and public trust are absolutely critical. Our belief is that multiple vaccines should be pursued in parallel, since it's not clear at this point in time which will be the safest, most protective, and ultimately most deployable. We think that preclinical models can provide useful insights into vaccine potency, efficacy, immune correlates, as well as vaccine safety, as well as pathogenesis. And finally, we have to realize as a global community with 7 billion people in the world, we really need multiple vaccines to be successful. I'll stop there. There's really too many people to thank. These are the individuals who contributed to the I-26 non-human primate study. And of course, a large group of clinical researchers led by J&J, BIDMC, the NIH co-VPN networks, and others for the clinical development program. And this is a picture of our group. Thank you. I'm happy to take any questions when the time is appropriate for that.
Perfect. Thank you so much, Professor Dan, for the interesting results and amazing talk. We will keep the discussion and the questions for the last session, of the end of this session. I just would like to highlight some issues that we experienced with the sound with some of the talks. So all the talks will be recorded and will be uploaded on the conference website and YouTube channel. So if you missed something or you experience some difficulties to hear some talks, be sorry for that, and we will upload the talks later. Now we can move to our next speaker, Dr. Dennis Longo, the Deputy Director for Science in Gamaleya National Research Center of Epidemiology and Microbiology, and corresponding member of the Russian Academy of Science, Doctor of Biological Science. Dr. Longo is the head of the department team in the Russia coronavirus vaccine, Sputnik 5. The first Russian vaccine against COVID-19 was developed under the supervision of Gamaleya National Center Epidemiology and Microbiology Center. This center is one of the leading research institutes. It was founded in 1891 and had a good experience with developing different vaccines against different diseases, including MERS and HIV and recently SARS-CoV-2 virus. Welcome, Dr. Longo. Please, if you can share.
Colleagues, distinguished participants of the conference, I would like to use this opportunity to thank you for giving us an opportunity to present the results of our studies on such a high level. So my presentation will be split into 2 parts. The first part, I will briefly talk about what is happening in the development of our vaccine. And the second part of my presentation, the scientific part of it, will be delivered by one of the members of the developers of the vaccine in part of the testing of the vaccine in the preclinical and clinical trials, that is Dr. Dolzhikova. So Dr. Dolzhikova will provide a full detailed presentation. I also would like to use this opportunity to express specific respect to Dr. Dan Barak. In fact, the whole number of publications prepared and published by Professor Barak to a large degree formed and shaped the adenoviral studies that has been conducted by the Gamaleya Research Center. I also would like to emphasize that thanks to the work conducted by Professor Dan Barak and also a number of colleagues who have been working with adenovirus type 5, a huge sets of data have been accumulated regarding the safety of the recombinant adenovirus of serotypes 26 and 5. That is exactly what has enabled Russian Federation to apply a special governmental decree. And please do not switch the slides at the moment. No need to switch them yet. Thank you. So that is something that enabled Russian Federation to apply a special governmental decree issued by the government. And according to that decree, if there is a candidate vaccine and we see that there is sufficient database on safety accumulated, then such a product or a vaccine can be issued in civil circulation in the time of pandemic under special conditions. So that is why the Russian regulator used this legal opportunity and registered our vaccine on condition, or the so-called provisional registration, and I'll explain what it means. That means that those conditions require that the Gamaleya Center is obliged to conduct large-scale clinical trials to prove the efficacy of the vaccine in parallel to civil circulation of the vaccine. So that means that at the moment our vaccine is simultaneously being conducted in civil circulation, where the group risks are being vaccinated, and by that I mean teachers, medical workers, social workers, and so on. And in parallel to that, we're also conducting our clinical trials. So at the moment, we've vaccinated more than 10,000 patients in civil circulation. And on the clinical trials, we already vaccinated around 20,000 volunteers with the first component of our vaccine, that is adenovirus 26. And around 12,000 volunteers have been vaccinated with both components of our vaccine. So as for production capacities in the prospect, I'd like to say the following. At the moment, we're at the stage of scalability of our production in Russia. And in that process, we're using full production sites. So we expect that we'll be able to produce millions of doses of the vaccine by the end of the year. And those production volumes would be primarily used to vaccinate the population of Russia. In order to further translate and transfer technology abroad, we are working in close partnership with the Russian Direct Investment Fund. And so far, we are planning to engage a number of production facilities in India, Brazil, Korea, and China. And in the nearest future, We are to launch or have already launched our clinical trials abroad. Primarily, those are the clinical trials that have already been launched in Belarus, and almost all the patients have been vaccinated already. And we're also planning to launch clinical trials in the United Arab Emirates and India. So here I would stop with my introductory remarks on the key areas of work, and I would like to give the floor over to Dr. Dolzhikova, who will do our scientific presentation. And I also would like to apologize, probably I will not be able to stay with you until the end of the presentation. However, my colleague Dr. Dolzhikova is fully competent to answer any questions that you might have later. Thank you very much for your attention. And now I'd like to give the floor over to Inna. Good afternoon, dear colleagues. Can you please turn back presentation at the very beginning? And the next slide, please. Thank you. So, for the first time in modern history, the humanity is facing coronavirus infection of such a scale. So, as for the stage of the 4th of November 2019, we see that the pandemic has covered more than 200 countries. And the number of people infected is more than 46 million people. More than 1 billion people have been diseased. And we see that all around the world, a whole number of measures are being taken by various countries in order to prevent the global catastrophe. And one of the efficient means to combat pandemics is vaccination. So during the previous coronavirus infections of SARS and MERS have demonstrated that in order to induce protective cellular response, it is important to develop humoral and cell immunity. When developing a vaccine, it is important to bear in mind that protective immunity has to be developed in a short period of time. Up to 1 month, particularly in the situation of a pandemic. It is also necessary to make sure that humoral and cell-induced immunity is performed. In order to achieve those objectives, one of the most attractive options would be the vaccine based on recombinant adenoviral vectors that enable creation of protective immune response already after 1 or 2 immunizations. The most promising vectors are recombinant adenovirus. Such vectors have been used for quite a long time, and their high levels of safety are confirmed by a huge amount of clinical trials, various preventive and therapeutical drugs. Therefore, when choosing a platform for delivery of antigens, adenoviral vectors seem to be the most reliable source. One of the promising antigens to be included in the vaccine is the glycoprotein. It is necessary for interaction with the receptor on the surface of the cell, which launches a cascade of reaction that results in internalization of the virus. Blocking of that type of interaction enables us to prevent internalization and decrease the replication of the virus. In doing so, the adenoviral particles that carry the gene of full-fledged glycoprotein of the SARS-CoV-2 virus is the most suitable way to immunize in order to induce protective immune response. Next slide, please. Can we go to the next slide? Yeah, thank you. In order to induce long-lasting immune response, it is necessary to conduct 2-stage vaccination. However, in case of use of recombinant adenoviral vectors, it is important to bear in mind that after the primary immunization, the immune response is induced not only on the targeted antigen, but also to the components of the vector, which in its turn can decrease the efficacy of the boost immunization if it is being conducted with the same adenoviral vector. This problem can be resolved with the use of approach of heterological vaccination. So in that case, for the first and second immunization, we use various recombinant vectors. And in that case, the immune response formed to the vector part after the first immunization will not decrease the efficacy of the boost vaccination. Next slide, please. We have developed a vaccine for prevention of COVID-19 based on recombinant adenoviral vectors. This vaccine is comprised of two components. In the component 1, we have recombinant vectors based on adenovirus, human adenovirus of the serotype 26. And the component 2 is a recombinant particles based on human adenovirus of serotype 5. Both of those vectors carry a full-fledged gene of S-glycoprotein of SARS-CoV-2 virus. The vaccination is conducted in 2 stages. So at first with the component 1, and afterwards in 21 days with component 2. This vaccine has been developed in 2 forms, namely the liquid form and lyophilized form. The storage conditions for those 2 forms vary, and you can see this information on the slide. Next slide, please. The preclinical studies of the vaccine include protectivity assays, immunogenicity, and safety assays on various animals. Today, we would like to draw your attention to the studies of protectivity. Protectivity assays have been conducted on several types of animals. Those were the Syrian hamsters with induced immunodeficiency, transgenic mice with HAC2 of human, and 2 types of primates. Those were common marmosets and rhesus macaque. During the assays of the vaccine and the Syrian hamsters were managed to demonstrate that the survival rates among vaccinated animals amounted to 100%. And at the same time, the survival rates in the control group amounted to 0%, and by the 12th day of the trial, all the animals died. On the 8th day after infection, a number of animals from each group, we examined their lungs. And we've tested macroscopical and histological analysis. And we've also analyzed the viral load in those tissues. And we managed to demonstrate that the lungs of the control animals have multiple impact. And the histological analysis demonstrate inflammation. And the viral load amounted to more than 10 logarithm of TCID 50 per gram of tissue. At the same time, we see that the lungs of vaccinated animals on the 8th day after infection looked healthy and have not demonstrated any signs of the impact of the infection. and the virus in the lungs of vaccinated animals was not detected. The research of the vaccine based on the transgenic mice has also demonstrated that the vaccine provides protection for 100% of transgenic mice from the lethal infection. At the same time, the survival rate in the control group was 0%. The research conducted among primates demonstrated that in the lungs of the control animals after infection, we see the spots of pneumonia and multiple infiltrates in the lungs, and that is demonstrated by the histologic assay. At the same time, in the lungs of the vaccinated animals after infection, we have not witnessed any injuries or any impact. And the lungs of the vaccinated animals after infections were not different from the lungs of the control animals that were not infected. The infection dose in all the trials are demonstrated on the slide on the left. And here it is important to pay attention to the fact that in all of the studies that we've been conducting, no AD effect has been detected. Next slide, please. So the generated results of the preclinical trials enabled us to get a permission to start clinical trials that have been launched on the 18th of June. The study was split into 2 stages and included 76 healthy volunteers of both sexes. We simultaneously analyzed safety and immunogenicity of the liquid and lyophilized forms of our vaccine. At the first stage of trials, we've analyzed the safety and immunogenicity of individual components of the vaccine. Each of the groups included 9 volunteers. On the 5th day after administration of individual components of the vaccine to all volunteers, we have conducted preliminary report on safety, and based on that report, the decision was made to move on to the 2nd stage of clinical trials. At the 2nd stage of this study, the volunteers have been administered with 2 components of the vaccine in the interval of 21 days. At the 2nd stage, we've analyzed safety and immunogenicity of the vaccine, and each group included 20 volunteers. In doing so, both of the components of the vaccine under this clinical trials have been administered to 40 volunteers. Next slide, please. When analyzing safety, we managed to demonstrate that after vaccination, the most frequent adverse effects included hypothermia, headache, asthenia, muscular and joint pain, changes to laboratory indicators of blood, and pain at the place of injection. However, all of the registered adverse effects were generic for the vaccines and cannot be considered as unexpected. It is also important to pay attention to the fact that the majority of the registered adverse effects were light, and less than 5% of all the registered adverse effects had an average degree of severity. In this study, we have not registered not a single serious adverse effect. In doing so, we managed to demonstrate that the vaccine has a good profile of safety. Next slide, please. The analysis of immunogenicity was conducted on several parameters of humoral and cellular immune response. Study of humoral immune response was conducted on both stages of our study. At the first stage, after administration of individual components of the vaccine, the volunteers in the specified time intervals would submit the samples of their blood serum to perform antigen-specific IgG and neutralizing antibodies. As for the volunteers of the second stage of the study, we've also gathered the blood samples in the identified time intervals, and we have analyzed the antigen-specific IgG and neutralizing antibodies. Next slide, please. And based on the results of the clinical trials at the first stage, we managed to demonstrate that administration of individual components of the vaccine enables induction of humoral immune response with 100% of volunteers. So on the 21st day after administration of one component of the vaccine, the level of seroconversion amounted to 100%. It is important to pay attention to the fact that the level of antibodies of IgG on the 28th day after vaccination with separate components of the vaccine were no different from the level of the antibodies of the same volunteers on the 21st day. When analyzing the viral neutralizing antibodies, we demonstrated that the volunteers after one-time vaccination provide viral neutralizing antibodies. However, the level of seroconversion was relatively low. On the 28th day after administration of component 1, the level of seroconversion of viral neutralizing antibodies amounted to 60%, and for component 2, around 78%. Next slide, please. At the second stage of our study, we demonstrated that administration of component 2 provides for an effective boost of immune response that was induced after administration of the first component. So on the 28th day from the start of monitoring, the volunteers demonstrated of the antigen-specific IgG was considerably higher than on the 21st day. And the increase of level of the glycoprotein-specific antibodies continued until the 42nd day. When analyzing neutralizing antibodies, we have demonstrated that administration of the boost vaccination induces neutralizing antibodies with 100% of volunteers. So on the 28th day, the level of seroconversion for neutralizing antibodies amounted to 95%. And on the 42nd day since the start of monitoring, it amounted to 100%. In doing so, we have demonstrated that our vaccine is highly immunogenic and induces clear humoral immune response. Next slide, please. We have also conducted analysis of cell-mediated immune response after vaccination. And cell-mediated immune response was analyzed based on antigen-specific proliferation of CD4 and CD8, and also on the antigen-specific expression of IFN gamma mononucleonic cells of peripheral blood. And we managed to demonstrate that vaccination enables creation of glycoprotein-specific cellular immune response with 100% of volunteers. Next slide, please. So based on the results of conducted preclinical and clinical trials, the vaccine vaccine has been registered on 11th of August 2020 under special condition. So that was a provisional registration under the decree of the government of the Russian Federation. So this provisional registration requires conduction of a large-scale clinical trial. However, it also allows the vaccine to be used in civil circulation under strict pharmacovigilance. Currently, we are conducting post-registration clinical trials that will include 40,000 volunteers. Clinical trial is conducted under constant monitoring of the condition of the volunteers. And next slide, please. So under the conducted post-registration clinical trial, we apply the so-called digital countries or digital loop that enables monitoring of safety and quality of the product at all stages of its life cycle. So that includes administration to the volunteers under clinical trials and also during civil circulation. This digital countries includes a shared register of all vaccinated patients, the system of monitoring of movement of each particular dose of the vaccine, and also a system for gathering of all registered adverse effects that are also being registered by the vaccinated patients with the use of a special mobile app. So as of today, the first component of the vaccine has been administered to around 20,000 volunteers. And both components of the vaccine have been administered to more than 12,500 volunteers. Thank you for your attention.
Thank you very much. That's a very interesting talk, and thank you for sharing these results with us. Now we can start the discussion session. There's 10 questions, and our audience are very interested to pass on to you. We have some questions that will be directed to all our current speakers, and some of them will be specifically to some speakers based on related questions to their vaccines. Can I ask all the respected speakers please to open their cameras? So, since we are, is everyone hearing you? Our discussion panel. Professor Sarah, Professor Dan, and Dr. Enna.
Ahmed, your sound is very...
Colleagues, apologies. It looks like there is some troubles with the sound on the Russian side. Can you please help us? Or is there a way to turn the volume on? We do not hear anything at all. Thank you.
Do you hear me better?
No, not really. And actually, if you just take a look at the website, at the chart here, we can see that a lot of audience are having problem with the volume. And this is just something that happened to us at the moment too. We can't hear you.
Is that also in team?
I can hear you. It doesn't sound clear, but I can hear you. This is Dan Brewer.
Okay, perfect. So, since we are speaking about non-applicable viral vector vaccines, so the first questions that will come to people's mind is that the pre-existing vector immunity. I'm sure there's different hypotheses and theory about how you can minimize that. So I would like to start by directing the question to Professor Sarah Gilbert that maybe an Oxford vaccine they try to minimize the pre-existing immunity against the viral vector, but how you can overcome that when you need to boost if that's required.
So I think the question was, that we minimised the chances for anti-vector immunity before giving the first vaccination, but then if we vaccinate with the same vector again, and we're giving it at a 4-week interval, will that be a problem? Well, I would agree that the highest immune responses possible are probably going to be generated with a heterologous vaccination using 2 different vectors. And both in Oxford and also at Janssen vaccines, a lot of work has been done with adenovirus priming and MVA boosting. That results in very strong immune responses because they're different viral vectors and there's no interference with anti-vector immunity. However, it does require the manufacture and the testing of 2 quite different vaccines, and this adds to the complexity of the challenge of trying to vaccinate very large numbers of people. So the approach that we have taken is to simplify that and use the same vector twice over. And we showed that the second vaccination does increase the immune responses. Interestingly, it doesn't appear to increase the anti-vector immune responses. We've shown that we get a very high level of spike antigen expressed in human cells that have been transduced with our viral vector, and very low levels of the adenoviral proteins themselves. And so, although we can detect anti-vector immunity being induced with the first vaccination, it appears to have a limited impact on the second vaccination, and also we don't increase the anti-vector immunity with the second vaccination. It's obviously something we're going to monitor for the long-term immune response and see how that changes over time. It may be that we don't need to give 2 doses of the vaccine. As I said before, we don't yet understand what level of neutralizing antibodies or T-cell response we need to achieve to get protection. And it would be really wonderful if it turns out that at least in younger adults, 1 dose is protective, because that would mean we'd get a faster onset of protective immunity and we would have more doses available to vaccinate the world. But until we have a better understanding, we'll continue using 2 doses of the vaccine, and the anti-vector immunity doesn't really seem to be posing too much of a problem. I'll stop there.
Thank you very much. I direct the same question to Professor Dan.
Sure, absolutely. So I think our thoughts are really quite similar to Dr. Gilbert in that the most potent vaccines would probably be heterologous prime boosts with 2 different platforms completely, such as adenovirus with a pox virus, adenovirus with a protein, or maybe a DNA or RNA with an adenovirus, for example. In my opinion, that would probably be the most potent vaccine. 2 different adenoviruses would probably be second most potent, and a single adenovirus given more than once would probably be next most potent, and then a single shot vaccine would be less potent. So I think it's a balance between complexity and simplicity, and we know that both are very, very important for a global vaccine. So I think that it's good that there's several different approaches being tried. And in particular, we've noticed that 2 shots of Ad26 give a tremendous boost of immune responses, very similar to a heterologous advector. But I think all the approaches need to be tried. So that's why in our program we looked at a single shot advector because it might be enough, and a 2-shot homologous vaccine, which would likely be substantially more potent and still requires manufacture of a vector. But we also appreciate that other heterologous platforms, particularly when there's 2 different virus classes, will likely intrinsically still be more potent.
Dr. Ayman, please give us your comments about the same question.
Dear colleagues, what we have demonstrated is that we see that one-time vaccination provides us with an opportunity to induce immune response with the volunteers. However, at the same time, when the second shot of vaccine is being administered, we can increase the immune response by multiple times. We therefore believe that based on the clinical trials we've generated and the post-clinical trials result that we've got, the most effective way to do that is to do this prime boost vaccination.
My second question is about basically studying the non-neutralizing antibodies. Of course, all the viral vector vaccines will aim mainly to induce strong T cell response, but you still can get a good titer of antibodies. Did you measure any titer for the non-neutralizing antibodies and how that can be having a negative or positive impact on the vaccine immunity? Professor Sparks.
Yes, I think all the vaccine studies have measured non-neutralizing antibodies as well as neutralizing antibodies. And we're now starting to do some more detailed work to look at other roles which may allow those antibodies to have a protective effect, and looking at the interaction of the antibodies against the spike protein with other cells that make up parts of the immune system. So it's quite a complex story, and it's something that we still have under review at the moment. But I think that we will be concentrating on neutralizing antibodies as the most obvious correlative protection. We're also measuring T cell responses, of course, because the adenoviral vector vaccines are very good at inducing T cell responses. And with all of the data in the very large numbers of people vaccinated in clinical trials, tens of thousands now being vaccinated with each of these vaccines, that may well give us the opportunity to dissect out the role of humoral immunity versus cellular immunity. But both are induced together by vaccination, and it's probably going to take quite large studies to really tease apart those differences. It's not something that we've been able to do with our preclinical studies, which have been fairly small, but maybe Dr. Baruch has got some better indications from his studies, which have been larger preclinical studies in the non-human primates.
Well, thank you, Dr. Gilbert. In our animal studies, in our monkey studies, we looked quite closely at neutralizing antibodies and non-neutralizing antibodies with Galit Alter and other colleagues. And consistently across 4 or 5 large non-human primate studies, including the published ones and some non-published ones, we see neutralizing antibodies as a strong correlative protection. But we also see that other non-neutralizing antibody functions also correlate strongly with protection. In particular, antibody-dependent NK cell activation and antibody-dependent cellular phagocytosis. It's difficult to dissect the differences between these antibody functions because many of the antibody functions actually correlate with each other as well. But ostensibly, the data would suggest that neutralizing antibodies are probably the most important correlative protection, but that other antibody functions may also contribute. So I wouldn't discount the potential of other antibody functions to contribute, as well as Sarah said, as well as T cell functions, because much of virus is located within cells. And while neutralizing antibodies will likely be able to neutralize free virus, then it is likely that other antibody functions and T cell functions are important for eliminating the first few cells that might get infected. So our view is that neutralizing antibodies are probably the most important, but that other antibody functions as well as T cell functions likely also contribute. And currently, in some unpublished data, we're doing adoptive transfer studies and CD8 depletion studies in the non-human primate model to get a better sense as the immune determinants of protection.
Have you done any related studies or measurements of China?
I apologize. If that question was directed to me, then I was unable to hear it.
I'm sorry, just I'm directing the same question to Dr. Anwar.
Okay.
Thank you for the question. In our analysis, we've also analyzed the virus neutralizing antibodies with the volunteers at the stage of clinical trials. And we managed to demonstrate that on average, the VNA level of the vaccinated volunteers was higher than the level of neutralizing antibodies of the reconvalescent patient 1 month after recovering from the COVID-19. So we also analyzed the cellular-induced immunity. Since today we cannot clearly state which of the two indicators of the immune response can be considered as a surrogate marker of protectivity. However, on average, we do see that administration of the vaccine provides for induction of a very clear immune response.
Perfect. The next question is directed to also all speakers. It's maybe hard to get a certain answer from now, but based on the preliminary data, would your vaccines will work in the age population, which is the most vulnerable group? Dr. Sarah.
Well, it's really important to assess the vaccine in older adults, and that's something that we've been doing recently. We started with a trial of 1,000 subjects between the ages of 56 and 69, and then 56 to 69, and then over 70 years, 1,000 subjects in each group, and that formed part of our phase 3 study. So we've been looking at the all of the immune responses in those volunteers, and we hope to report on that very soon.
Perfect. Thank you very much. Do you have any data, Dr. Dan?
I think really the same perspective that testing the vaccine in elderly as well as individuals with comorbidities is critically important because those will be people we need to protect. And so our clinical trials are making a special emphasis of enrolling elderly, racial, ethnic minorities, as well as people with comorbidities. Another population is pediatrics, and our clinical trials will start enrolling pediatric populations very shortly as well, which is also a very important population given the back-to-school issue. So we think that all those populations are very important to protect and therefore need to be included in the clinical trials.
Thank you very much. Dr. Anna, did you include it in your study, this age population? And did you have any preliminary data that suggests that your vaccine is working well in the age population?
As for the clinical study that we're conducting at the moment, according to the protocol, we include there volunteers aged 60 years and older, and they comprise 1/5 of the total amount of volunteers engaged in the clinical study. So in this clinical study, we are going to vaccinate 8,000 volunteers at the age of 60 and older. And based on the results of this clinical study, of course, we'll make conclusions on the safety, immunogenicity, and efficacy of the vaccine at this age cohort.
Perfect. Thank you very much. So maybe that's have been done in animals, or because it's also hard to answer this question at the moment because of the short time since all the clinical trials started. But do you have any idea maybe about the longevity or the duration of the immune response against your vaccine? Sarah, it's maybe for humans still running, but is there any preclinical data support that vaccine can stay for a certain period, long or short, and you might need a booster vaccinations or not?
Well, with our vaccine, we don't actually have preclinical data on durability of response because the animals that were vaccinated were then challenged 4 weeks after they were vaccinated, and then the study ended a couple of weeks after that. What we do have is clinical data from our MERS vaccine study, MERS coronavirus, which expresses the spike protein of that coronavirus. And we saw that that also induced both antibody responses, neutralizing and non-neutralizing anti-T-cell response in the subjects that we immunized. And a year after vaccination, they all had strong immune responses, which were still present. So we certainly expect with adenoviral vector vaccines to be able to see responses present at a year after vaccination. And obviously, as part of our clinical trials, we'll continue to follow up our volunteers to look at the persistence of those immune responses. But it's not data that we have yet.
Thank you very much. And do you have any preclinical data that supports certain longevity or durability of the immune response?
I apologize. I had trouble hearing the question. I'll assume that question was directed to me. I apologize if it wasn't. We are also looking at durability in both monkeys and in humans. Naturally, the one-year durability studies will naturally take longer to complete. So we do not have long-term durability data yet in either monkeys or humans with our Ad26 COVID-19 vaccine. We do have long-term durability data with the Ad26 Zika vaccine and our Ad26 HIV vaccine that looks very good in humans as well as monkeys. But we'll have to just wait and see. There can be antigen differences, so I think we have to just wait and see to measure the durability empirically in both animals and in humans.
Thank you very much. Dr. Alma, do you have any comments on the same question about the durability and longevity of your vaccine?
Yes, already today we do know that among primates, the immune response lasts for no less than 6 months. It achieves a maximums on the 3rd month after vaccination, and at 6 months, the humoral response is not going down. The level of IgG does not change from the 3rd to the 6th months. And we also know that the neutralizing antibodies are also preserved at the same level, and that is quite a high level. That is what applies to the COVID-19 vaccine. But we have also developed a vaccine against the MERS, where we've used the full-fledged glycoprotein of MERS-CoV virus. And in that clinical trial, we managed to demonstrate that the immune response among primates lasts for no less than 1.5 years. At the same time, that is not just antigen-specific IgG, but also our virus neutralizing antibodies. And based on these data, we can expect and hope that the vaccine against COVID-19 will also provide an immune response that will last no less than that.
Wonderful. Thank you very much. Dr. Sarah, this is a directed question about Oxford vaccine. And it's asking about did you see like differences in the immune response depending on the geographical locations of the participants in different locations for the clinical trials?
I think the question was, are we seeing different immune responses in participants in different locations for the clinical trials? That's not a question I can answer yet. We still have a lot of work to do to get all of the samples from the different clinical trial locations around the world to core labs, which will run the same assay on samples coming out of all of those different trial sites. So we will report on those studies when we have the data, but it's not something I can tell you about today. We have looked at immune responses to other adenovector vaccines in different populations in the past, particularly malaria vaccines in Africa. We do tend to see a slightly lower immune response in African populations, so it's something we need to look at carefully with the COVID vaccine in the different geographical locations for our clinical trial sites.
very important questions that, of course, the people ask about. Since all the vaccines or the viral vector vaccines based on one single antigen from the virus, which is the spike protein, how you can feel confident about any mutation that could happen or the need of reformulating your vaccines in case any mutation that happened particularly in that spike protein, and how it's conserved sequence protein that could basically, so what's the chance of being mutated or changed, then you might need to reformulate your vaccine. I'll start with Dr. Sarah, if you can give your comments on that.
Well, so far we haven't seen any drop in the ability of antibodies induced by vaccination to neutralize the mutant spike proteins that are now in circulation. Remember that the spike protein needs to maintain the same conformation to bind to the receptor on the human cells. If it mutates away from that, it won't be able to bind. It will be a less effective virus, and so will be selected against. Obviously, we have to keep an eye on mutations that arise, and it would be possible with this type of technology to insert a different spike protein sequence if necessary, but as yet we haven't seen any indication that that is going to be necessary.
That's great. Thank you very much. Professor Dan, if you heard the same questions, I will appreciate giving your own comment on that.
Yes, thank you for the question. I think it's a very important question. As Dr. Gilbert said, so far, the mutations that have arisen do not appear to impact neutralization. And so I think we're optimistic that the current vaccines that we and my colleagues on this panel and others in the world are developing will be effective for the current circulating viruses. However, I think we need to remain vigilant. There is a theoretical possibility that as vaccines get rolled out in populations, they will provide an additional new immune selection pressure, and therefore there's at least a theoretical possibility that as vaccines are rolled out, there could be immune selection pressure for viruses that would specifically evade part or all of the neutralizing antibody responses induced by these vaccines. And since virtually all the vaccines that are currently in trials today are based on the original sequence from Wuhan, then it's a theoretical possibility that there could be cross-neutralization or cross-resistance with other vaccines. I'm not saying that's going to happen. In fact, I'm optimistic that it's a virus that doesn't mutate very quickly. However, I do think that the scientific community needs to remain vigilant and to continue to sequence viruses and screen for possible resistance. But as of now, current viruses, we believe, should be sensitive to the neutralizing titers raised by these vaccines.
That's really great. Thank you very much. Dr. Enna, do you have any comments to add?
Yes, I'd like to say the following. We have analyzed the serum of vaccinated animals in the neutralizing reaction with a number of various isolates that have been identified at the various points and detected on the territory of Russia. Those isolates have several replacements and amino acid replacement in the sequencing of glycoprotein. However, the fact of replacement of that did not impact the level of virus neutralizing antibodies in the serum, apart from the blood of the vaccinated animals. We also see that the level of mutational activity at the glycoprotein is not very high, and as a result, we expect that the vaccine should work even if some new mutations come up. Well, that's our expectation at least.
So I would like to ask the last question, and that will be kind of a combined question, which is, of course, the most interesting question that will be asked by public. And that's asking about when the vaccine will be ready for approval, because these two stages, and then it will be ready to be available for public. I know it is really hard to give an estimation, but I would ask at least for the best case scenario. Professor Sarr, please give us your view.
Well, yes, you're right that it's impossible to know when these vaccines are going to be ready to be rolled out. And of course, when we do come to vaccine rollout, it's not going to be the entire population that gets the vaccine offered to them on the same day. So there has to be a process of prioritization within countries to decide who to give the vaccine to first. And the two top target groups for prioritization are either healthcare workers who are at risk of infection because of their job, and the older part of the population who have been shown to have much higher risk of severe disease and death from this virus infection. So we're all very interested to look at the detail of the immune response in older people to work out if it's actually appropriate to use these vaccines in older adults, if we expect them to be protective, as it is in younger people. And then that last piece of the information will help us decide on the final prioritization of the vaccine rollout, which in itself is going to take some months. Even after we have an efficacy result and regulatory approval, getting the vaccine out to immunize large parts of the population is going to take some time. So I'm afraid it's not possible to give you a simple answer, but I would say that we would expect to be starting this process by the end of the year in terms of getting the efficacy result and then moving forward to apply for regulatory approval. That depends on transmission of the virus in all the locations of our trial sites, but it has been high recently, so that may lead us to a result within the next couple of months.
Thank you very much. Professor Dan, the same question.
Well, thank you for the question. It's an incredibly important question, of course, the question on everyone's mind, including on my mind. I think we have to be very careful at giving calendar dates. There have been calendar dates proposed, and those have now been revised by multiple groups. So I'm hesitant to give a calendar date because, as you know, the phase 3 trials are endpoint driven. And so they're endpoint driven rather than timeline driven. So it's very difficult to predict the exact calendar date that they may end. And also when they end, and if they do show safety and clinical efficacy, then there will still be a time period for regulatory review and approval, as well as the actual rollout, assuming that manufacturing is not a factor. So there's a lot of uncertainties, and so any exact prediction is really more of an opinion rather than anything to count on. I think that for our program, we started the phase 3 trials a bit after some of the other groups, and so naturally we would expect efficacy data from our vaccine program to be several months after some of the other groups in the field. So if everything goes well, we're hoping to see efficacy data by the end of this calendar year, possibly the beginning of 2021. But at this point, I would really hesitate being any more specific than that.
Great, thank you so much. Final comment from Dr. Anna about the availability of the vaccine. When do you think that could happen?
Well, like we explained, the Russian vaccine has already been registered on a provisional condition. As for the regular registration, it will only be made available after the completion of a large-scale clinical trial that includes 40,000 volunteers. As for the preliminary results of those clinical trials, hopefully they will be generated somewhere in the beginning of 2021. However, the final results and the reports on the efficacy and safety of the vaccine will only be made available upon completion of this large-scale clinical trial.
Thank you very much. Thank you very much for all our respected speakers, Professor Sarah Gilbert, Professor Dan Arouche, Dr. Anna. I really appreciate your time, and on behalf of the organizers and our audience, we know how you are busy, and we really appreciate the time you gave to us to share this valuable information. So I'd like to thank you, and thanks for our audience. Thank you very much.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound. Where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
المملكة استضافت قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound. Where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
المملكة استضافت قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
Welcome to the RNA-based vaccine track. Today we have Dr. Robin Shattuck from Imperial College London and Dr. Barney Graham from the NIH in the United States. And the first person who's going to speak is Professor Robin Shattuck. He's from the Department of Infectious Diseases at Imperial College, and he's leading the COVID vaccine effort at Imperial College.
Thank you very much. And it's a great pleasure and privilege to be invited to talk to you today. I'm going to give you an update of where we are with developing a COVID-19 self-amplifying RNA vaccine. So we had already been working on this manufacturing platform with the view to its utility in outbreak situations. And one of the advantages, along with other RNA approaches, is that it can be manufactured on a relatively small footprint, and therefore it has the potential for rapid production capacity, but also distributed manufacturing, which may be important as the world looks towards new ways of responding to pandemics faster in the future, given the portability of this type of technology. We work with self-amplifying RNA. It's slightly different from conventional RNA that you'll hear about in the second presentation. Self-amplifying RNA is based on an alpha virus backbone that encodes the non-structural proteins of the alpha virus required for copying the viral genome, but replaces all the structural proteins of the alpha virus with the gene of interest, in this case being the glycoprotein of SARS-CoV-2. When this RNA is introduced into cells, into the cytoplasm, it initially goes through an amplification process where it amplifies copies of the whole genome, and then it goes through a process where the polymerase enzymes are self-cleaved and switches to predominantly making the subgenomic RNA, which encodes the gene of interest, here being the surface glycoprotein of SARS-CoV-2. And the potential advantage means that one can use lower doses than conventional RNA in order to achieve the same level of immunogenicity. And this second slide really highlights the difference. Conventional RNA on the left-hand side, self-amplifying RNA on the right-hand side. They both need to be appropriately capped and have appropriate 5' and 3' UTRs and poly-A tails. And they both need a formulation approach to introduce the RNA into the cytoplasm of the cells. But there's a direct relationship between mRNA and the amount of protein expression, whereas for self-amplifying RNA, because of this amplification process, you get more output of protein expression per molecule of RNA introduced into the cell. So the way the vaccine is used, conventionally, we're looking at an intramuscular delivery for this vaccine platform. We are also just starting to look to explore whether the same technology can be used mucosally, and evidence suggests it can be introduced mucosally, at least into animals. With conventional intramuscular immunization, we get self-amplification in the muscle, high levels of expression, and potent immune response. The process for manufacturing the vaccine is relatively straightforward, and it has a high yield in terms of product, producing about 1 million doses per liter. And the technology and the scalability is highlighted in this publication, also shown on the slide if people want to look at that in more detail. For delivery of the RNA, we have been collaborating with a company in Canada called Acuitas to use a lipid nanoparticle that has been optimized for delivery. This encapsulates the RNA into a lipid nanoparticle that's then taken up by target cells, and the RNA is released into the cytoplasm, where it can go into its cycle of amplification and expression. Like many in the field, we have been using a pre-fusion stabilized version of the spike glycoprotein. It's full length, so it utilizes the transmembrane and cytoplasmic domains, and this means it's expressed in a native-like format on the surface of cells, and this hopefully will be a good target for induction of neutralizing antibodies in humans. So the technology allowed us to move very fast in the design sequence and to move into preclinical trials. We started as an academic group with no manufacturing capability, and were able to develop the process and get it into a clinical trial from a standing start in January to starting clinical evaluation in June. So outside of a pharmaceutical setting, I think that shows that one's able to make extremely rapid progress with this type of technology. We have undertaken preclinical studies for immunogenicity, and this was recently published in Nature Communications, where we looked at a range of doses and assessed the immune response. Shown on the left-hand graph, this is now looking at binding antibody responses. vaccine given at 0 and 4 weeks. You can see here the response to the vaccine given as a DNA vaccine with electroporation. And now then the dose response with the self-amplifying RNA going from a 10 microgram dose down to a 0.01 microgram dose. And comparing to a small subsection of recovered COVID-19 patients, you can see that even with 2 doses of the vaccine, we're inducing a response that's significantly higher than that seen in recovered subjects. We've also assessed neutralizing antibody responses. This shows, again, a dose response in terms of the induction of neutralizing antibody responses. And there's a wide number of viruses assessed in the paper if anyone wants to look at the online version. So our target dose for humans is between 1 and 10 micrograms, and we're in the process of evaluating that. We also saw in these early animal experiments that induces a good cellular response. So this gives us confidence that induces both neutralizing antibodies and cellular responses. The proof of the pudding, as it were, will be to see whether these are recapitulated in clinical trials. We also assessed the ratio of IgG1 to IgG2 in these mouse models as a marker of Th1 to Th2 phenotype. And you can see in this left-hand graph showing the ratio of these two isotypes, indicating that it is inducing a strong Th1 response, which is encouraging in terms of having the right phenotype of response for a COVID-19 vaccine and typical of other RNA platforms that are being developed. So we've moved this from a design of concept through into a formulated product. We are now in a range of different challenge studies. We've undertaken the relevant toxicology studies that we need to do to move into clinical trials. Those were perhaps more extensive than some of the platforms that had already been into the clinic. And so that gave us a little bit of a time penalty, but we were able to get into clinical evaluation in the summer of this year. So where are we in that evaluation? Well, we're in the final stages of selecting the dose. Now, obviously, our clinical trials are still ongoing, so I'm unable to give you the detail of the studies, but we're seeing a clear dose-response effect in terms of immunogenicity and a good safety profile. We hope that if that immunogenicity profile looks good enough, that we will move into efficacy trials in the beginning of next year, potentially both in the UK and internationally, subject to having the right funding to move it forward. The target dose is between 1 and 10 micrograms. It's a 2-dose vaccine. So far, we've seen a very good reactogenicity profile. The vaccine itself can be used as a standalone 2-shot vaccine, but also has the potential for boosting other candidates that may be less suitable for boosting campaigns, particularly those types of approaches like the use of viral vectors that have less utility the more times you use them. And so we are not there yet. A lot depends on the data that we will accrue by the end of the year to determine whether we move into efficacy trials with this first generation approach of self-amplifying RNA. We are hopeful that that will progress, but we are also developing this technology very actively because we think it has high utility for other infectious threats that have pandemic potential for the future. The production approach has been developed. It's currently a frozen formulation, and it needs to be kept at -70, but we are working actively on a 2 to 8 formulation, and so far we've accrued 3 months stability at 2 to 8. That hopefully will allow us to transition from having an exclusive low temperature distribution pathway to moving towards a more refrigerated pathway that would be advantageous for pandemic use and use in situations where freezing of the vaccine is less accessible. We have set up a spin-out entity to commercialize the vaccine called Vax Equity Global Health. This is based on social business principles, and we are dedicated to making the vaccine available if it is effective at the cost of goods plus a small percentage to keep the vaccine updated and the manufacturing capacity maintained. That gives us some advantages and disadvantages. The disadvantage is that we don't have funds to ensure manufacturing capacity up front and reserve that. The advantage means we have flexibility to work with different manufacturers around the world, and also to have easy relationships with the likes of organizations like COVAX, should we require to do that, and should there be interest in supporting the vaccine dependent on efficacy data. So as we think about the future, we are very much wedded towards trying to establish a distributed approach to vaccine manufacture. This is just as an example. We don't have the partners finalized in those different parts of the world, and we may work with different countries. This is just to suggest that a platform approach that can be placed in many different centers of the world will enhance distribution of the vaccine, and we believe ultimately equitable access of vaccines for the future. So thank you very much for your attention, and I will leave it there and hopefully take your questions when we get to the question session. Thank you.
Thank you very much, Professor Shattuck. And now we're going to hear a presentation from Dr. Barney Graham. And Dr. Graham is Deputy Director of Vaccine Research of the Vaccine Research Center at the National Institute of Allergies and Infectious Diseases at the NIH. And the NIH is co-developing an RNA vaccine with Moderna. And Professor Graham is leading the vaccine effort at the NIH. Thank you, and welcome, Dr. Graham. Thank you. I think you're on mute, Dr. Graham.
Thank you very much. Good morning and good evening. I'm going to talk today about our program, not just for the coronavirus, but an approach to pandemic preparedness that we think could be relevant even into the future. I work at the Vaccine Research Center at NIH on the Bethesda campus, and this was founded about 20 years ago to develop an HIV vaccine. Although we haven't been successful with this effort yet, it has driven the technology development that has been applied to a number of other problems, including influenza and other unmet medical needs, including emerging infectious diseases shown at the bottom. We can do this because the center not only has the basic research facility, but also has about 100 engineers north of in Gaithersburg that do process development, another 100 or so engineers that do pilot plant manufacturing, a self-contained clinic, and a laboratory that does GLP analysis of our clinical samples. It's done and managed by this group of principal investigators and program heads led by John Muscola, shown here. And over the years, we have developed a number of different delivery approaches for vaccines. I'm going to talk today about the nucleic acids, especially mRNA, but we've also done vector-based approaches. We've made VLPs for chikungunya. We've made protein-based approaches, especially for RSV that I'll mention, and nanoparticle-based approaches for influenza. And we make a number of different monoclonal antibodies and distribute them around the world including the MAV-114 that went into East Congo in 2018 and was shown to be effective at treating Ebola virus disease. So our attempts this decade to address emerging infections with rapid response have had a mixed outcome. And in 2014, in West Africa, even with a vaccine on the shelf prepared to go into phase 1 studies, by the time we got into this phase 3 trial based in Liberia on the red line, the outbreak was largely over. So even though we were able to enter phase 3 testing within about 8 months, we missed the major part of the outbreak and did not get an answer with this adenovirus-based vaccine. Similar thing happened with Zika in 2016 when we made a DNA vaccine from sequence to phase 1 within about 100 days, and then got a phase 3 trial started within about less than a year into a phase 2B, and still failed to get an answer because the outbreak had already waned. And so even though we can go relatively fast, we are not going fast enough. And let's see here. We're not going fast enough. And so we think we have to do better preparedness prospectively. And we wrote about this in 2018, a concept of prototype pathogen approach for pandemic preparedness, in which we would divide the 25 virus families that infect humans into 4 major groups based on entry mechanisms and their type of fusion mechanisms. So class 1 fusion proteins, class 2 non-enveloped, etc. and provide centralized functions that would allow you to do all these activities needed to do both the basic research, solving structures, making reagents for at least prototypes in each of these families, and also establish the mechanisms to rapidly manufacture. So we have been pushing this concept and think it's quite important, and especially now. And not only do we want to go fast, but we want to be precise. And so if you have a rapid response without a precision antigen design, we think it has not as good a chance of working. And a lot of our thinking on this has been based on our experience with respiratory syncytial virus. I'm showing you here that F-glycoprotein on the surface of the RSV that we were able to solve in 2013. Jason McClellan, who was a postdoc in Peter Kwong's lab at the time. And during that process, we discovered these apical epitopes shown in red that are highly neutralization sensitive. Antibodies to these sites are 100 to 1,000 times more potent than the licensed antibody palivizumab would be for this site. But we know now that this molecule rapidly flips into a post-fusion conformation. And in that case, the red epitope is lost. And we know from the last 30 years of trials repeatedly showing that this molecule can only boost neutralizing activity about 2 to 3-fold. Whereas if you can stabilize the pre-fusion conformation with the C-terminal trimerization domain, internal disulfides, and cavity-filling mutations, this molecule can now boost neutralizing activity by 16-fold or more. So we have proof of concept for a structure-based vaccine design. And the reason it's so important, I'm showing you in this movie, where you can see how dynamic this molecule is. And so all class 1 fusion proteins are rearranged in order to make membrane fusion so the viral nucleic capsid can get inside the host cell. You want to target this initial molecule, not the final post-fusion molecule. Class 1 fusion proteins are present on many of the envelope viruses of concern, including the families here, the pneumoviruses, RSV, paramyxoviruses, and even coronavirus spike is analogous, and even Though they don't have the same kind of domain structure, they have a very similar overall organization with endoproteolytic cleavage exposing hydrophobic fusion peptides. This is what enters the host cell, and this heptad repeat pulls into this heptad repeat, which is anchored in the viral membrane, pulling the membranes together and allowing fusion. So the strategy for maintaining pre-fusion is the same in all these molecules. So finding solving the atomic structure revealed this new target of vulnerability. We have clinical proof of concept for this approach, preserving neutralization-sensitive epitopes, and the pre-fusion conformation can be generalized across other virus families, as I'm showing here. So, for instance, in the paramyxovirus family, which is also a very large family with a very large zoonotic reservoir, we've looked at not just the parainfluenza viruses that would be over here, but we've looked at Nipah as the prototype for this family, and also measles and mumps viruses. And the paramyxoviruses are very similar to the pneumovirus, like RSV. It has a trimeric fusion, class 1 fusion protein. It also has an attachment protein that's either in a tetramer or a dimer of dimers. And we know that these fusion proteins assume a pre-fusion state that flips into the post-fusion state. And here is the loosely organized attachment protein that, depending on the virus family, is either called HN for hemagglutinin, H for hemagglutinin, or G. And we've looked and found ways to stabilize the fusion protein of Nipah, measles, and mumps, as shown here, and have made antigens that either have pre-F alone, the hexameric G, or a molecule in which you combine the pre-fusion F with a trimerization domain that then extends into expressing 3 copies of the G or HN protein. What we found is that this molecule, what we call the pre-FG chimera, is the most immunogenic. It can generate the highest level of neutralizing activity and can be delivered by either protein or mRNA. So we think this general concept could be used for future paramyxovirus outbreaks, and you can even make heterotypic constructs, for instance, using the fusion protein of measles and the G protein of mumps, or vice versa. You can generate immunity to more than one virus at a time. So looking at paramyxoviruses and Nipah as a prototype, we know again that pre-F is better than wild type or post-F, that stabilizing mutations can be generalized across paramyxoviruses, particularly within these henipaviruses, and that these pre-FG chimeras, even if they're heterologous chimeras, can elicit neutralizing activity against each component. So on to the coronaviruses, which is the topic of the day, of the year. We know that we've had 4 endemic coronaviruses circulating for many years, hundreds of years, since the alpha viruses entered the human population. And now we also have 2 endemic beta coronaviruses, OC43 and HKU1. And over these last 2 decades, we've had 3 more entries of beta coronaviruses into the human population. So it's likely that we will have more. And so back in 2012 and 13, when MERS was erupting in the Middle East and some other locations, and we were also at the time solving this pre-fusion structure of the RSVF protein, we decided it was important to solve the structure of the spike protein of coronaviruses, which was not known at the time. And we tried and failed a few times with MERS and SARS. In collaboration with Jason McClellan again and Andrew Ward at Scripps, once we started working with this endemic coronavirus HKU1, which was inherently more stable, we were able to solve this spike structure. And learned a number of interesting things. It's dynamic in different ways, and this is the receptor binding domain flipping up. It has to flip up in order to reach its receptor. In the case of MERS, that would be a human DPP4. In the case of SARS, it would be the ACE2 receptor. But this protein has to be dynamic or it won't function. And so what we learned from that, once we had the structure and could see the amino acid side chains, we could find and identify stabilizing mutations. And the one that worked best was between the heptad repeat and the central helix, shown here. If you change these two amino acids to prolines, you can prevent the rearrangement of these helices into the heptad repeat and largely stabilize the molecule. But fortunately, it turned out that putting the two prolines into this position, even in MERS and SARS, stabilized those proteins and then allowed those structures to be solved. And not only did it preserve the neutralization sensitive epitopes at the apex of this molecule, but especially important for gene-based delivery approaches for vaccines, stabilizing the protein, as Dr. Shattuck said, that 2 proline stabilization per spike increases expression levels up to 50-fold over wild type in MERS. So it's allowed us to solve not only HKU1, but the MERS and SARS spikes. It's allowed the solution of other beta coronaviruses that are endemic, or alpha coronavirus endemic viruses, veterinary viruses, and even potentially emerging viruses out of bats. So that is would be the approach then for a new coronavirus outbreak, and we wanted to combine that with rapid manufacturing platforms. And because of the work we did with Moderna in 2016 on Zika, we had been working with Moderna on a prototype pathogen approach for both paramyxoviruses and coronaviruses. And here I'm just showing you a cartoon of how these mRNA molecules have to be lipid encapsulated to get them into the cell, and they're then translated. And in our case, we express a full-length spike protein that's stabilized with the 2p mutation, and it's expressed on the surface of the transduced cell. That should elicit antibodies to the authentic conformation of the spike protein. In the case of the Moderna type of mRNA, it is more conventional than Dr. Shattuck's approach of using the alpha virus amplicon, but it is a modified nucleotide RNA. It uses a 1-methyluridine instead of just normal nucleotides, and because of that, it changes the way the cell recognizes the RNA and triggers these different response systems that generate interferon responses. And so it reduces the amount of TLR7 signaling, reduces the amount of TLR3 signaling, and purification processes to eliminate double-stranded RNA also eliminate some of the other intracytoplasmic signaling pathways. So this modified nucleotide RNA is thought to reduce the amount of overall innate immune response and allow longer-term protein expression. So these different ways of delivering RNA, either as an amplicon or as a modified nucleotide or as a normal nucleotide, have different levels of protein expression or innate immune response, and they all may have roles in different settings. So this is some of the data we generated before this outbreak using the MERS prototype and the mRNA from Moderna. It's gone through mice. The mRNA for the Nipah virus has gone through mice and ferrets. But in these DPP4 transgenic mice from Ralph Barrick's lab at UNC, we could show that 0.1 or 1 microgram of RNA delivered on a 0 3-week schedule could prevent infection of the lung, and even at 0.01 could reduce virus load by about a log. And this completely prevented disease in this lethal infection. And even at the breakthrough dose, the partial immunity resulted in partial protection and didn't have any evidence of the kind of enhanced pathology that some people have worried about that had been seen in prior SARS-1 models and had been seen in the 1960s with a whole inactivated RSV vaccine. So the proline mutations stabilize S spike in the pre-fusion conformation. That makes them more immunogenic. We think this is a general solution for beta coronaviruses, and mRNA is a suitable delivery approach, and it can be rapidly manufactured. So when this new SARS virus was reported as the cause of COVID-19 around the 7th of January, we started making plans with our collaborators, not just Moderna, but also the 4 academic labs that we've been working with over these last 3 or 4 years to make a rapid response to this new problem. And this is the virus infecting a ciliated epithelial cell. And I'm just highlighting this part of the frame. And you see huge numbers of viruses coming out of this cell. And just to highlight, this is what the virus looks like. It's a small spherical particle, and it has these large knobs, and this is the spike protein, and this is the target for all of our vaccines. And as Robin mentioned, because we could build immediately using that 2P mutation a stabilized protein, we were able to rapidly make the protein in collaboration with Jason McClellan, got the structure by around January 31st, and that then set the course for the rest of the project, because we think high quality protein is the beginning for everything. And we used this protein knowing that it was in the right conformation because we could see it for diagnostic development with the CDC. We used it as a B-cell probe in collaboration with AbCellera. We found an antibody called 555 that has now been manufactured and put into clinical trials by Lilly, and used it also in this vaccine approach with Moderna. But the stabilized version of spike is also being used in these other approaches that are heading toward phase 3 trials in the US, including the other mRNA vaccine, the adenovirus vaccine from Janssen, and the 2 subunit vaccines being made by Novavax and GSK. So it was really because of this prior work on both isolating antibodies, mapping and neutralizing sensitive epitopes and structure, stabilization and vaccine evaluation that allowed us to go from the sequence being reported on around the 10th of January to a phase 1 clinical trial in 65 days and a phase 3 trial within 6.5 months. And likewise, because the byproduct of this is making protein that can be used as probes, we sent probes and cells to AbCellera around the middle of February, and within 5 months of that, Lilly was in phase 3 evaluation and has already shown that early treatment of people with symptomatic coronavirus antibody treatment can prevent a progression to more severe disease. So the human clinical trial, the phase 1 trial, has been reported now. We've done this in different aged adults at different dose levels. And I'm just showing you some of the antibody data here. The spike, full-length spike trimer on the left, and the receptor binding domain antibody response on the right. And these columns here are convalescent sera from about 42 subjects, 6 of which have severe disease. And you see that the antibody response to the full spike or to the receptor binding domain is well above the convalescent sera responses at all dose levels. Likewise, for neutralizing activity, the pseudovirus result is about 3 to 4 times higher than the geometric mean titer of of the convalescent sera, and the PRNT, the classical neutralization assay at ID80, is about 5 times higher than the average convalescent sera. And looking at an ID50 of another live virus nude assay, you can show now that in the older age groups, 56 to 70 or greater than 71, that you're still getting high-level neutralization activity that's comparable to what you see in the younger adults. So we now have evidence that this mRNA is immunogenic. It works in both mice and monkeys and humans with good results that are higher than convalescent serum results. We've shown protection in mice and in monkeys in both the upper and lower airways. And we've got safety data based on T-cell responses. And in the case of mice, monkeys, and humans, this is very Th1 biased response and relatively no Th2 cytokines, which is one of the signals that was shown in the early RSV enhanced disease studies. And these have all now been published and can be looked up. And now we've shown that the response in elderly subjects is also robust and also Th1 biased. So I just want to briefly review that it started quickly because of the prior basic and translational research and a plan for using prototype pathogens to prepare for the next pandemic. We think it's important to not only go fast, but to use precision in antigen design. And I think this shows the importance of pre-established public-private partnerships, not only with Moderna, but also with AbCellera. The phase 3 trial is now enrolled. It finished enrollment on October 22nd, and more than 20,000 people have had their second dose. And we're now just waiting on endpoints to accumulate. These studies were done under the Operation Warp Speed with the Coronavirus Prevention Network, and the phase 1 study was done in collaboration with DMID, another division within NIAID. This is in a trial, and we think that a global approach is needed for pandemic preparedness that's in a more systematic way. And I just want to thank my PI collaborators, Jason McClellan, who's been a long-term collaborator on structure, Ms. Mikia Corbett, who led our small team of coronavirus researchers over the last few years, and then our academic collaborators at Vanderbilt, UNC, Mark Dennison, and Ralph Berry. And I'll stop there. Thank you very much.
Thank you very much, Dr. Graham. So now Professor Shattuck is going to come back. Welcome back. So now we're going to have a short discussion. And let's start out, if Professor Shattuck, if you could tell us a little bit about the differences between the RNA vaccines and the other types of vaccines, and specifically the advantages of the RNA vaccines.
I mean, some of the advantages of the RNA-based vaccines is speed of production and the fact that they're made by a fully synthetic process, which means in terms of the release of the vaccines, it makes it easier. It also makes it a technology that can be adopted by many manufacturers in different parts of the world. It also allows for combination vaccines to be made very readily because you're just combining different strains of different RNAs into the same vaccine. And it means if the virus, particularly COVID-19, was to change, there's no evidence that it's changing at the moment in a non-immune population. But if immune pressure, once the vaccine's rolled out, they'd cause changes, the vaccines could be adapted very readily. The disadvantage right now is that they are all dependent on a frozen cold chain. I think that's likely to change fairly quickly in the coming months to years. But at the moment, that's probably the major disadvantage in the approach against some of the more conventional technologies.
So even if it, as you said during your presentation, that in a few months you hope to have your vaccine stable at refrigerated temperatures.
For a period of a few months. So we're building up our stability data, but it does mean that you could have a central hub with frozen product. But if it was needing to be distributed to more remote settings, it could then be viable at 2 to 8 for a period of a few months.
Do you think it could ever be stable at room temperature?
Well, we already know from the field more generally that it's possible to freeze dry these RNA vaccines. So, longer term, it would be possible to stockpile vaccines and make freeze dried products that would have stability potentially at room temperature. They're not available right now because everybody has been moving as fast as possible to develop these vaccines. And so I think this is something we'll see in later phases of development and rollout.
It's going to be really interesting to see that. Dr. Graham, can you tell us some of the advantages of the RNA vaccines?
I'll just add a couple of things to what Dr. Shattuck has already said. First of all, the Moderna vaccine is stable for a long time at -20 or -30 degrees. So, and it's stable at 4 degrees for at least 2 weeks. So as he said, this makes it feasible for a central hub to deliver vaccine to practicing clinics and keep that for a couple of weeks at room temperature. So, and there are plans to lyophilize this. So I think that eventually this could be made kept at room temperature. The other big advantage, and the reason we chose a gene-based delivery approach, especially one that is not affected by pre-existing immunity, is that our experience with RSV is that if you prime with the gene-based vector and elicit CD8 T cells and type 1 CD4 T cells, that you can avoid this type of enhanced disease immune profile that we really wanted to avoid. So one of the major reasons we chose RNA is to get that type of T cell profile.
During your presentation, Dr. Graham, you talked about how you started developing the vaccine on January 10th, and within 65 days, you were already starting phase 1 trials. And then you said that prior relationships were very important. Can you tell us a little bit more about your experience developing vaccines that helped make this go so quickly?
The U.S. federal government does not have a mechanism for submitting a biological license agreement to the FDA, commercializing vaccines. And so if we don't have commercial industrial partners, any of the work we do would just stay in the medical journals. It wouldn't ever get into a volunteer's arms. So for us, it's crucial that our publicly funded laboratories have an industrial partner if we're trying to make products. It's not necessary for the basic research, but if you're trying to do product development, as we are, it's critical. And so in this case, we've been working with Moderna intensely for about 3 years on both the paramyxovirus and the coronavirus projects. And we actually were planning to go in with a demonstration project for Nipah. But on the 7th of January, when the coronavirus, it was recognized as beta coronavirus, the CEO of Moderna told me, as soon as you send us the sequence, we'll start making the product, because we had learned to trust each other. Or we trusted the data we'd already generated. And so he made a very high-risk decision, a brave decision to start GMP manufacturing without any additional experimentation. So this is where the relationship is important, and the prior work is also important.
And Professor Shattuck, do you find the same, that you already had prior relationships and so it was much easier to move quickly?
I mean, we did have prior relationships. I mean, I think we were more constrained in that because this was new technology, we had to do more preclinical toxicology before we could move into human trials. I suspect, in future iterations, we won't have to do that, so we could be as quick. And I think we were probably more challenged financially to move this product forward, given it was originally coming out of an academic setting. So again, we are, we've established a spin-out company in order to make sure that that part moves faster in the future.
And how much do you anticipate your vaccine will cost per dose?
So that's quite difficult to put an exact figure on it because it depends on scale. We anticipate it will be between $10 to $20 per dose. That could come down dramatically with orders of scale. And so it's hard to predict without knowing the scale and the volume that might be required.
Dr. Graham, you're in a different situation because the United States government paid for the vaccine for everyone in the United States, correct?
Right. One of the ways this has gone fast is that everything has been done in parallel. You know, most of these steps are usually taken in series or serially because people are more interested in saving money than in saving time. In this case, all these steps have been done together, and a lot of money has been put at risk. So the U.S. government has already prepaid for vaccine that may not work. And they've already committed dollars to buy vaccine in case it works. And so this kind of putting dollars at risk is what allowed us to go so quickly without putting any kind of safety issue at risk. And I really feel like we have not skipped any of the important safety steps in this development. And so buying these products ahead of time means that if they work and as they are scaled up, they should be available to the U.S. population. I know Moderna, at least, is also negotiating deals with other countries for purchases of doses. So that's what's happening right now, and we expect that by the end of this year, there'll be several million doses, and by April or so of next year, there'll be up to 100 million doses, and then it's expected that you could make 500 to 1 billion doses a year of this kind of product. Of course, that's not enough for the whole world of 7 billion people, and so that's why it's so important that multiple products work. We really need more than one vaccine to get through this problem.
Mm-hmm. Professor Shattuck, you're planning to seek approval for your vaccine next summer. Is that correct?
That's correct. If everything goes well, if we have the right profile and we ensure the right funding to do large-scale efficacy trials, we anticipate we may get ready for approval by next summer.
And Dr. Graham, yours is going to be you're going to seek approval next month. Is that the plan?
This is all under negotiation, but Moderna has announced publicly that they plan to submit an emergency use authorization application to the FDA the last week of November. Mm-hmm. And whether that is the mechanism for using a vaccine, if it turns out to be safe and effective, is still being negotiated. It's also possible that this would be used under something called expanded access, which means it's still a research product, not a approved licensed product. And then it's also possible the regulatory agencies will just say, we'll just finish the full licensure package, the BLA, and get us a full package so we can just approve it for use. So I think a lot of these regulatory mechanisms and approval processes or authorizations are still being under negotiation.
Professor Shattuck, do RNA vaccines in general, are they more beneficial for certain demographics, like, for example, for the elderly or for pregnant women? Or do you anticipate that there'll be certain groups where it's more beneficial?
I mean, right now we don't know. We certainly are seeing data that suggests that they work well in older populations. but there needs to be much more work to look at a wider range of populations with different underlying comorbidities, and also in younger populations. I think what will be very key is how well these are tolerated in different at-risk groups. There will obviously be a very different risk-benefit ratio between those who are vulnerable to severe disease, particularly elderly populations, and younger age groups, which predominantly aren't at risk of disease, but by getting the vaccine will prevent the virus being transmitted to others. And there they may require a vaccine that's more tolerable because they're getting less benefit from it themselves. So I think it will be very interesting to see how it unfolds, and I suspect we'll see different vaccine candidates are more appropriate or more acceptable for different groups as we move forward.
So in the trials, do you test the vaccine on people over 70, and do you test it on people who are 80 and 90?
So you've heard from Dr. Graham already that they've looked at an older population. We're in the process of looking at an older population to check that our vaccine is as safe and as potent in that type of age group. And so those are very important studies to do.
Yeah, I'm really concerned about the elderly. So I'm really interested in how that's going to be as effective.
I mean, one of the things that I think is, again, an advantage of the RNA technology is the ability to give repeat booster shots. And certainly we know for some vaccines in certain groups where they don't make as good an immune response as healthy individuals, they may require additional vaccinations. And RNA can do that because you don't get any immune response against the RNA itself. So if you needed 3 shots to ensure somebody in a vulnerable group was really well protected, that's possible. If you need an annual booster, that's possible. So there's a lot of flexibility with the technology.
So if somebody gets the RNA shot, they get it 1 month, and then you wait a month and get your second dose the next month. So when is someone protected at what point is someone protected? It would be probably 6 weeks after the initial dose, or?
That's what we would anticipate. But I mean, of course, that's the $60 million question is what level of immunity is required for protection? And are we talking about protection from developing disease or protection against infection and transmission? We don't know those answers until the data is revealed. And so that's why we need all these different vaccine candidates to ensure the success at preventing disease, but ideally success at preventing transmission.
And Dr. Graham, so are you imagining that someone would be protected for a year and then they would get a booster shot, or potentially longer? Or do we not?
The real answer to that question is ask me in a year.
Okay. I'll ask you next year.
But for our vaccine, we're
definitely will protect us for a year.
We're going to start. We're going to start counting cases 2 weeks after the second dose. But Pfizer, who has a very similar vaccine, it also uses a modified uridine nucleotide, and it also is using the full-length spike, and it's giving its doses at 0 to 3 weeks and starting to count cases after 1 week. So it's counting cases much earlier, and we do see immune response. We actually see 100% antibody response after the first dose. So we don't really know what is going to happen. We know in mice, if we give a single 10 microgram dose, that at 7 weeks we have fully protected mice with a 7-week challenge. And so 1 dose could be protected, but this is one of the things. We don't know how long the immunity will last, and we don't know how much immunity you need to be protected. And those are the things we really have to wait on these trials to find out.
Do you know how long it would take to manufacture the vaccine? The manufacturing process?
Once you have the supply chain secured for all the lipids, all the nucleotides you need, the manufacturing process itself is very quick turnaround. It's just a matter of scale. And so, the thing is, if these things work, people are going to be working, making these 24 hours a day around the clock with multiple different machines and facilities. And so it's going to be a continuous flow of making these kinds of products.
Can RNA vaccines be manufactured quicker than other types of vaccines?
That's the other reason we chose RNA, is we think this is the fastest. As I said, we can make our own DNA at the VRC, and for Zika, we made it in about 100 days. In this case, from the time we gave the sequence to Moderna to the time they delivered a GMP product was 41 days. It's quite remarkable. It was the only time in my life that I've seen the manufacturing outpace the clinical permissions and IRB and all the other things you have to do. So it's remarkable how quickly this kind of synthetic process can go. It's not like the process where you require cells to make vectors or proteins and things like that. That's one of the reasons why it's attractive for response. Now, these other products that use vectors and proteins, we also need them to be working. Because, like I say, I don't know that the supply chain or the manufacturing scale is enough for the whole world if you just have an mRNA.
Mm-hmm. So we have time for one last question. So I'll let you, if you just want to make some final comments, because I still have a few more questions. But Professor Shattuck, in closing, would you like to make just a final comment?
I think the promise of this technology is enormous, not just for COVID-19, but for future pandemics. It also makes realistic that one might be able to make vaccines against things that have not been commercially attractive because they only have a relatively small demand, and you can make smaller batches at probably an affordable cost. We are keen on the self-amplifying approach because we are very focused on global health, and we think that we will increasingly be able to drive the dose down. So if we can drive the dose down, it means that the product will go a lot further. And in terms of pandemic response, that may well be very important.
Mm-hmm. And Dr. Graham, do you have any final comments?
Well, the point I hope people remember is how important it is to know things in advance. And the only way to know that is by supporting basic research. So if we hadn't known the spike structure and some of these other things before, we would have been months later. If this had been a Bunya virus instead of a coronavirus, it would have taken months and months longer, or an arena virus. If this had not been a coronavirus, we would not have gotten off this kind of start. And for a world economy that's probably had 11 or more trillion dollars extracted out of it, you would think that a few billion extra dollars a year to study these other 25 virus families that can infect humans would be a very attractive investment. So to me, it means that we need to do better at supporting our basic research and being better prepared.
Mm-hmm. Well, thank you both for joining us today, and thank you so much for all your hard work on the vaccine. We're all rooting for you. Okay. Thank you so much.
Thank you.
We didn't HKU1 story.
We have 1 minute left if you want to do it.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound, where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
المملكة استضافة قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound, where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
المملكة استضافة قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
And I'm the moderator for this session. And in this session, the speakers will discuss their efforts in the development of a protein-based vaccine, and a DNA-based vaccine. The first presenter will be Dr. Lai, who is a professor at the Institute of Microbiology, the Chinese Academy of Sciences. And his talk is entitled Development of Beta Coronavirus Vaccines Against COVID-19, MERS, and SARS. And the second speaker will be Dr. Kate Broderick. She's a Senior Vice President in Research Development at Inovio Pharmaceuticals. And her talk is entitled INO-4800, a DNA-based vaccine against COVID-19. We'll have the speakers present first, and then we'll have questions afterwards that you could type questions into the Q&A box if you would like us to answer any specific questions. And welcome to the presenters. Dr. Dai will be the first speaker.
Hello, good morning, good afternoon, and good night, everyone. So today, I think I would like to give you an introduction about the protein subunit COVID-19 vaccine. And this vaccine is developed from our institute with Zhifei Biologic together. So, we are facing unprecedented threats by coronavirus infections. 7 of them are infected humans, but 4 of them are only cause the mild cold-like symptoms. Only 3 of them are life-threatening. They are the SARS-CoV-2 and SARS-CoV and MERS. And also the coronavirus contains many from the animal that cause the animal pathogenesis. So we urgently needed to developing the COVID-19 vaccines. So far, several speakers have introduced different platforms. We summarize it now as 7 platforms, including the inactivated one, which has several candidates in clinical trials in China, and also live attenuated ones, although there's no one so far in clinical trials, but several in the preclinical studies because of its long development process. And also some groups using only part of the virus, especially for the S protein. So this is we using we call the protein subunit vaccine, and we are focused on today will give you a more introduction about this platform. And some platform has has introduced before is viral vectors. Many groups are using the adenovirus vectors for vaccine, and also the DNA. I think later Dr. Cates will give you the details, and also the mRNA. There's two nicely talks just now showing the potential of this platform, and the last one, the viral vector, virus-like particles, which do not contain the virus genetic materials and mimicking the virus. And so far, there are also some vaccine candidates based on this tech platform. So to start for the coronavirus vaccine, we need to choose a target as just before. Several speakers have introduced the S protein or S2P S protein mutants as the antigen targets, and also many of group using the receptor binding domain. So we're choosing the receptor binding domain. As you see, the S protein contains the receptor RBD region, which is responsible for a bond to its host receptor. For MERS, it binds to DPP4, and for the SARS and SARS-CoV-2, it binds to the ACE2. So RBD is a major target for neutralizing antibody. So it's a attractive target for COVID-19 vaccine. So another reason we choose the RBD as the antigen targets because to avoid the potential ADE risk, because the antibody band enhancement has been described for many viruses like dengue, and also in coronavirus, like in feline coronavirus, has really severe AD effects for the vaccine. And also in animal model, the SARS-CoV has reported such effects. Preferred strategies to reduce the AD risk is to achieve the immunofocusing, by which you can see in the right panel, the RBD region mainly induced potent neutralizing antibody, and also for the other part of the S protein like NTD, S2, they also reported some weakling or some relatively weak neutralizing antibody. But this antibody sometimes their benefits they extend the neutralizing activity, but also some non-neutralizing weak neutralizing antibody which can bind the virus but not enough to neutralizing the virus, but can enhance the virus entry. So we focus on the RBD, but RBD is a small size of a protein. So for that, it's usually less immunogenic compared with the full-length S protein. So at the start of the project a few years ago, we started the MERS coronavirus vaccine development. When we doing the producing the RBD protein for basic research, we found interesting that we always get equilibration of RBD monomer and dimer, and for vaccine we don't know which one is better, so we immunize the mice for both forms of RBD, and significantly the RBD dimer shows a dramatically higher binding antibody induce much higher RBD specific IgG and neutralizing antibodies. So we challenged mice with MERS and find the dimer has significantly reduced virus loads in lung and also dramatically relieved the lung injury in the mice. So, or then we choose the RBD dimer as a better imaging as compared with the RBD monomer. So then we determine the crystal structure of the RBD dimer, and you can see it's like a bilateral long structure and fully exposing the receptor binding motif, which is responsible for the receptor docking. And also this dimer is linked by the C-terminal free cysteine. So that's why we see equilibration of a monomer dimer, because sometimes they don't has a stable homogeneous dimer formation because they has a equilibration of the monomer dimer. So this is maybe a drawback for the downstream of vaccine production. So we have another round of structure-based design, because the N-terminal and C-terminal are density invisible. So we get another round of mutagenesis just by connecting the C-terminal and N-terminal of this dimer together, because the flexible N-C terminals makes them flexible to connect it together and do not interrupt the dimer formation. So by this way, we get a quite stable and homogeneous RBD dimer, and we compare this dimer with the sulfate bonds linkage dimer and finds the RBD single chain dimer or the tandem repeat dimer has a similar, if not higher, immunogenicity, inducing a high level for neutralizing antibody. So we, when we finish this MERS part, we'll summarize this data, and then in the 2020 in January, the SARS-CoV-2 comes. So we have a quick response. To quick response this pathogenesis, we ask whether this strategy can be used to for the other beta coronavirus vaccine development. So we using the concept for SARS-CoV-2 and also the SARS-CoV. And by analyze the sequence, we adopted the same strategy, started from the same starting position, but stopped it before the free cysteine. And we found there's a very conserved free cysteine for all the RBD from the beta coronavirus. So we stopped it cotton 1 cotton before, and we got the stable SARS-CoV-2 RBD dimer, and we immunized the mice, compare with the monomer, and find the RBD dimer has dramatically increased in the specific IgG and neutralizing antibody. And also for the real 2 vaccination, introduced the high level for neutralizing antibody against the live SARS-CoV-2 virus. So we also tested the strategy for the SARS, and also find SARS, the RBD dimer, tandem repeated RBD dimer has much higher immunogenicity and introducing higher neutralizing antibody compared with the monomer. So we during the same time we collaborate with the Zhifei biologic company, a leading company, vaccine company in China, and so we generated the stable cell line in CHO cell, and we finally we have a very high antigen yields. For the MERS, we have more than gram per liter, and for the SARS-CoV-2, we can purify 0.67 gram per liter. So, which means this high imaging yields will be scalable, productive, and can meet the huge global demand for the SARS-CoV-2 vaccine. Then we testing the protective efficacy in the mice, ACE2 transgenic mice, and 2 doses of vaccination using high RBD-specific IgG and neutralizing antibody. And after challenge with real virus, SARS-CoV-2 virus, we found in the virus loads in lung has dramatically decreased for around 4 logs. And to examine the pathology in the lung, we found in the placebo group that there was a severe interstitial pneumonia, but by contrast, the vaccine group has dramatically relieved the lung injury, only show very slightly lung change. Okay, so the overview is that we developed such this strategy and proved this concept for the RBD dimer, and it shows protective efficacy for the MERS and SARS-CoV-2, and also can be applied to counter the future emerging threats by the other beta coronavirus infections. And also we translated quickly with GFA to generating high yields of antigens, and has a pilot scale production and entering into the clinical trials. So in the June 19, this vaccine has proved by China FDA to enter the phase 1 clinical trials. And in July 10th, the vaccine has entered the phase 2 clinical trials in China. And this picture shows the first injection of the phase 1 clinical trials in China. And just a few days before the phase 1 and phase 2 clinical trial data is blends unsealed. So we don't have finished analyzing the data so far. We just give you a summary of a preliminary of the phase 1 and phase 2 clinical trial data. So we enrolled 15 volunteers in the phase 1 trial and 900 volunteers in phase 2 trials. And in both trials, vaccine shows safe and tolerates in all individuals. And so far, there's no vaccine-associated severe adverse events in both phase 1 and phase 2 trials. For the neutralizing antibody, seroconversion rate is 100% in phase 1 and 96.6% percentage in the phase 2. So it's so far it's high seroconservation. And for the neutralizing antibody titer, we're using the CPE method. Yes, some of you using the PRNT, but we use the CPE, and that is in the phase 1 is around 94.5 GMT titer, and for the phase 2 we have 102.5 GMT titer. And all the detailed data we are now in the summarizing and analyzing it, and we assume to get more detailed analysis data for publish. And the phase 3 trial we have already applied for several countries and expected to start it at November this week, maybe the end of this month. Okay, thank you for your attention. So I'm willing to answer your questions later. So
Julia, you're on mute.
Okay.
Thank you very much. That was very interesting and informative. I
Thank
have some
you.
questions, and there's a few in the chat too. We'll answer them after Dr. Broderick gives her talk about the DNA-based vaccine against COVID-19.
Let me just pull it up. Just let me know, Julia, when you can see the screen.
Okay.
Seems to be taking some time here.
Leon Penn, maybe you want to stop. Oh, there it comes. Okay, it's just got a little lag because this is a global conference, so it's actually doing fairly well all around the world, but it takes a little bit of time sometimes to load. Now we can see it.
Great. So thank you so much, Julia, for your introduction today, and also thank you very much to the organisers for the kind invitation to be here today. I really do appreciate that. I'm actually having some problems with my computer here. Sorry, just give me just 2 minutes. Julia, for some reason, I can't see it on my computer. Oh gosh.
You can't see it on the Teams interface?
I'm sorry.
To expand that again.
Oh gosh. I'm just going to stop. I am just going to. Oh, here we go. I'm sorry. And can you see now?
Yes.
Okay, great. So I'm sorry, I'm still having some problems, but I'm going to get started anyway. So my name is Kate Broderick. I'm the Senior Vice President of Research and Development at Inovio Pharmaceuticals. We're a US-based pharmaceutical company with offices both in Pennsylvania and in San Diego, which is where I'm based. We specialize in making DNA medicine approaches for infectious diseases, for vaccines, and also immunotherapies for oncology targets. But obviously today the topic on everybody's mind is, of course, COVID-19. And so I'll be talking today about our vaccine INO 4800, which is a DNA-based vaccine against COVID-19. So this slide really just gives you a little overview of our technology platform. And so I should say it has really two major components, one of which is the optimized DNA vaccine portion. So we take the sequence of the bacteria, virus, cancer cell that we're interested in, and we optimize that sequence for a variety of different reasons, of course, expression being one of them. Then we insert that into our backbone DNA, which really holds the DNA in place. And DNA in general is an incredibly robust molecule, one that's incredibly stable, so that has a lot of benefits that I'll touch on recently. Also one that's really been proven to be extremely safe over many, many years of research. But we combine that with our proprietary delivery technology. So in the case of the COVID-19 vaccine, we're actually delivering this to the skin rather than to the muscle, which is, of course, what standardly occurs for many vaccines. So we also have we've been working in this field for a long time now, and so we've treated over 2,000 subjects with our technology platform, so combining the DNA with the electroporation, and so have a really very robust clinical safety profile. And we were also the first entity to show clinical efficacy with combining our DNA with our electroporation platform, and that was for our precancerous cervical dysplasia vaccine, which is now in 2 concurrent phase 3 trials. So I'm going to talk through really some of the elements that make DNA platforms, specifically our optimized DNA platform, so appropriate really to address the issues that the globe, frankly, is currently experiencing with the COVID-19 pandemic outbreak. One, of course, is efficacy. So we all know that of the 2 key components for an appropriate vaccine, an effective vaccine, of course, is absolutely key, and a safe vaccine, of course, is also very, very key. And we really have multiple demonstrations of efficacy for other targets. We've been working very rigorously in the infectious disease space for many years now. And in fact, our learnings on MERS, another coronavirus, really positioned us very well to be able to move so quickly when the pandemic reared its head back in January of this year. We've also worked extensively on Lassa, influenza, Ebola, and Zika, and have shown efficacy in animal models in all of those different disease targets. Another area that is really key, and it might be sobering for some people to hear, but as of today, we don't actually know what are the protective immune responses required for protection from COVID-19. I mean, we certainly have experience with other coronaviruses and, of course, other infectious diseases, but we don't actually specifically know what's required for COVID-19. So from that perspective, I think it's really very important that we think of a very broad immune response, and that's really a major differentiator of DNA vaccine approaches, is that we're able to generate not just those all-important neutralization antibodies, but also those T cell responses, which we believe are absolutely crucial to really controlling the virus spread, especially in the lower respiratory tract. So that's something that really is very important, and I think until we really lock down those specific mechanisms of protection, it is really very important to be looking at broad immune responses. Another particularly admirable characteristic of DNA is the stability of the product. So as we think about potentially vaccinating every single person on the planet, it really is quite important that we think of the logistics of moving vaccine all over the world and storing and stockpiling vaccine all over the world so that everyone who needs it can get vaccinated. And as such, having a stability profile that doesn't involve frozen cold chain is really quite key. And thankfully, DNA has that. In actual fact, we're stable at room temperature for a year, and actually at accelerated increased temperatures of 37 for over 2 months. So when you think of kind of moving vaccine product around in aeroplanes and in other modalities, not having to have that frozen or even in our case refrigerated is a huge benefit. Of course, safety is absolutely key here. No vaccine should proceed unless it's proven, of course, to be safe. And really, we have a very strong historical safety profile with DNA, and that continues to be reflected in the data that we're generating with our COVID-19 vaccine. And lastly, we're very able to scale up the process of manufacture. When we're thinking of potentially 8 billion doses of vaccine, it's really quite key to be able to say that we have enough vaccine to be relevant. You might have the most efficacious, the safest vaccine on the planet, but if you can only make a certain number of doses, then that stops being relevant to a global pandemic. And we really feel that DNA lends itself to that scalable manufacture. Oops. So now moving on, as I mentioned, our technology really has a variety of different elements to it. We have these precisely designed DNA plasmids. And just to give you an example, we use an algorithm to come up with our with the sequence for our DNA vaccines, and we use the full-length spike for COVID-19. And in actual fact, from receiving the sequence of the virus from the Chinese authorities back on January 10th to having a fully designed vaccine on paper using our algorithm took us 3 hours. And that was really quite a key catalyst for us to be able to kind of press start and get moving on the effort towards a vaccine for COVID-19. We combine that with our smart proprietary delivery device. So this is a device that delivers our vaccine to the skin of our subjects or our patients. The skin really is an incredibly immunogenic organ, and is also allows us really to deliver the vaccine using incredibly short needles, which of course is very beneficial when we think of needle stick injuries, and also the very real needle phobia which is suffered by many people around the world. And really it's the marriage of those two elements together that allow us to generate the really robust in vivo responses that we see both in relevant animal models and also, of course, in subjects in the clinic. So this cartoon really just kind of outlines the steps that go into our process for our platform. So, of course, and if we use COVID-19 as an example here, we identify the causative agent. And so we were incredibly grateful for the Chinese authorities for working so fast to sequence what we now know as the SARS-CoV-2 virus. And really using that sequence, we inserted that into our proprietary algorithm. We chose the spike, full-length spike, and really we did that on the historical data that we had from our extensive work on MERS. We optimized that sequence, we inserted that into our backbone and manufactured enough of the vaccine to allow us really to immediately start testing that in animal models. And really then, the race was on then to develop the appropriate supporting data then to get into the clinic as fast as possible, which we did. So moving on, and I apologise because I'm still having some IT issues on my side. But so really that was kind of a general statement about our platform in general, but really to give you some very specifics about the COVID-19 program. So firstly, when you have a global pandemic, I think it's very key to be part of a global effort and a global consortium. And that really has absolutely been very, very key to our ability to move very rapidly in this space. And as such, I'd really like to thank the people, the supporters, the funders who have really allowed us to move as rapidly as we were able. Two of those have been, working in the infectious disease global health space for many, many years now. That's CEPI and, of course, the Gates Foundation. And they were very early supporters of our effort. We're also incredibly grateful to the US government and the Department of Defense for their ongoing support of our program, the Wistar Institute, AdVaccine Beijing, IVI in Korea, and our other ongoing collaborators in Australia, at PHE in England, who have been absolutely fantastic collaborators, allowing us to move very, very rapidly in what is a very complex situation that we're experiencing today. So really, as I mentioned very early on, one thing that was very key for us was our ability to build on our prior experience and leverage our experience in developing a MERS vaccine. And we had a huge amount of success in that area. We had performed a phase 1 trial in the US and a phase 2 trial in Korea, both of which showed really excellent protection from challenge in primate studies, 100% protection, but also had generated very nice clinical responses in our clinical trials. And so that really emboldened us to knowing that our platform worked very well for another coronavirus. And so we felt very confident about the activities for SARS-CoV-2. We also of course, had a huge historical database in regards to other infectious disease targets. So we were very confident in regards to the safety profile and also the robust immune response profile that we were likely to see in the clinic. And we felt that profile that we had historically generated would be something that we would be able to use to address the need for an EUA or an expanded use moving forward. So we're generating at the moment a dual strategy, both for emergency use authorization, but also for traditional approval pathways through generating very strong preclinical package, which we did very early on and have shown protection in a variety of different animal models, from the mouse COVID-19 model all the way up to the primate model, which we've shown. Interestingly, we've been able to show duration of protection, and that's really very, very key. So as I mentioned earlier on, you might have a very safe vaccine, you might have a very effective vaccine, but if the duration of that protection only lasts for a few months, its relevance in a global pandemic is less. So we were able to show that we had just under 4 months of protective efficacy in a primate model, and we're currently about to start the challenge for the 12-month time point too. So really, as we think of what we could kind of in our heads imagine as a perfect vaccine, you would really be looking for that duration of protection as well. So we feel very confident that DNA is going to allow us to do that. We've also completed our phase 1 clinical trial for our COVID-19 and are looking to rapidly advance into our phase 2-3 program in the near future. So, as I mentioned, we based our design of our SARS-CoV-2 vaccine on our experience with MERS. So we used the full-length spike, and we did that with the confidence that we had not seen any evidence of vaccine-enhanced disease in our MERS program. So we felt very confident that that would be replicated for our SARS-CoV-2 program, and that's certainly been the case so far, with no evidence in any of our preclinical or clinical models. We, of course, went through all the standard steps of preclinical development, where we showed in vitro expression work, and then also extensive preclinical work, which is published in Nature Communications. And this is really just a timeline really showing what we at Inovio have been doing for the last 11 months. And really, I'm sure that's very reflective of all the developers that have been working in this space. It is, from a personal standpoint, I can't decide whether it feels like 11 minutes or 11 years, but it certainly doesn't feel like 11 months. It's been really quite quite a year that I certainly personally have never experienced in my professional career, and hopefully never will again. It's been really, really quite a remarkable experience and quite a humbling experience as we all work towards that that much needed vaccine for this awful pandemic. So this is really the target product profile that we have generated for our INO 4800 SARS-CoV-2 vaccine product. And really, we've done this very much using the WHO blueprint as a template. Again, I would say that we are looking at, of course, different targets depending on whether we're looking at an emergency use authorization or for long-term use. But essentially, the profile really remains the same, with that remarkable stability profile, the use in an adult population, although of course we'll be looking at pediatric populations in time, and also looking to use it in an elderly population as well, which is absolutely key, as we know, of course, they are disproportionately affected by this vaccine. So we started our phase 1 trial in April at 2 different sites, our AMR site and also our site at UPenn. That was 40 healthy volunteers. We were able to use sites that we'd previously used for other trials, and so that was really helped us be very, very speedy. And we actually went from designing our construct to treating our first subject in 83 days, which was really remarkably fast for us. So we're still analysing that data, but we're very excited with the safety and immunogenicity profiles that we've seen so far. And really here are the summaries there. Our safety profile was safe and well tolerated. The most common adverse events were grade 1 in severity, and they were generally injection site reactions at the skin. And on the immune response side, 100% of our participants demonstrated an immunological response. And we were able to, of course, as I mentioned, provide protection in both the mouse and the NHP model. So overall, our objectives were to show that this was a safe model, and I realise that I'm up on time. Suffice to say that we are very excited about moving into our phase 2/3 trial in the very near future, and I thank you very much for your time. Julia, do I need to stop sharing, or is that?
Yeah, I think you could stop sharing.
Sure.
Mm-hmm. Thank you. And then we'll get everybody up on camera, and I think we'll go into a question session. So we did get some questions coming in. Let's just confirm with IT that we can start the questions. Okay, yes, we can start the questions. There was a question that came in about how long were you predicting that the neutralizing antibodies will be effective for after using either of these types of vaccines? But that also brings up the question of defining neutralizing antibody for this audience. I'm not sure that that's it's a pretty scientific term. So if maybe each of you, starting with Kate, could talk about, I think, Kate, you did talk a little bit about how long you think you'll get an immune response to last in a person after the vaccination. But could you explain the types of immunity that you're looking for? And then also, the second question that came in related to that is, if the virus mutates, would that immunity then still be effective?
Yes, certainly. Thank you, Julia, and thank you for the question. So maybe I'll start backwards, if you don't mind, in regards to if the virus mutates. So I think that's obviously a real concern when we think of viral pandemics, just because viruses are incredibly tricky little organisms that, their whole process is to ensure that they stick around. And so one of the ways that they do that is through mutations. However, and of course, a perfect example of that is influenza, and that's why we need to get different annual flu shots every year, because it's so incredibly effective at mutating itself. Thankfully, touch wood, we haven't seen those levels of mutations for SARS-CoV-2, and that's something that we should definitely be grateful for. However, there have been some variants, and so in different parts of the world there are different variants of the virus circulating. However, certainly I can only speak for our vaccine, but we've actually been able to show that our vaccine is able to neutralise all the circulating variants of SARS-CoV-2 at the moment. And that's really kind of one of the characteristics of the DNA is through our optimization process, we can try and make it a much more broad protective response, as opposed to very specific to a single variant. And of course, Lee Pan will be able to talk about his vaccine in regards to that as well. But coming back to your first question, Julia, in regards to what do we need really as a protective immune response, and that really comes back to the fact, as sobering as it is, we don't actually know what that is at the moment. I think infectious disease in general, people kind of have the dogma of neutralizing antibodies, which of course are, I'm sure, vital components. But I think we should keep open minds at the moment that since we don't know, a broad immune response encompassing both the humoral and those T cells, those kind of killer cells that are able to really actually kill the virus, might well be very, very crucial in providing protection against this disease. And I'll pass it over to Dr. Dai to answer as well from his perspective.
Okay. So in my opinion, for the protection correlates, there is lots of paper says that shows the neutralizing antibodies protective correlates in animal model, and also adaptive passive transform of serum, convalescent serum, or a monoclonal antibody can treat the patient, coronavirus patients. So neutralizing antibody, I think, is definitely very important for the protection, but also from the recovered patients, that's finds that the T cell responses correlates with the minor of the diseases. So and also T cell responses may contribute for the long term for the immune memory for the long term of your vaccine induced immunity. So I think the best way is to induce both arms of responses. And so when the antibody is not enough, maybe T cell can contribute more for the protection.
Mm-hmm. Okay, I have a question that I had thought of before. Is a lot of groups are using MERS as sort of the standard for developing the vaccine, because that's what we know, and that MERS and then the original SARS were coronaviruses, and that SARS coronavirus 2 is very similar to SARS. But do you have an idea of why these viruses are behaving so differently, and if the same approach should be used for them? I guess the question is, for like, if you're choosing the same approach to make a vaccine, how do you know that it will work against COVID-19?
Okay, maybe I can I can share my opinion. There are really difference for the MERS and SARS and maybe the other beta coronaviruses because first they use different receptors. For SARS and SARS-CoV-2, they use in SE2, but for MERS, they use in DPP4. And also for some other coronavirus, they're using different receptors. But so far, most of them using the C-terminal as the RBD, but in some cases, they're using the N-terminal as their receptor binding. So maybe they're not a universal strategy. You have to case by case. But we are seeking for universal strategies for all these coronavirus or even all the coronaviruses. So we can choose in for example conserved targets and so far this research is really poor identified. It's really small amount for research on that. And also some conserved epitopes and T cell maybe contribute for the cross reactivity that you can, for example, like influenza, you are seeking for universal vaccine. So maybe for coronavirus, universal vaccines is a sort. Yeah.
Mm-hmm. I think, Julia, when you want to kind of use all your learnings you have for general coronavirus research. But, I think there were some very distinct differences about MERS, SARS, and SARS-CoV-2. I think specifically when we think of the fatality rates of those outbreaks, thank goodness, the fatality rates for MERS and SARS were much, much higher than anything that we're seeing for SARS-CoV-2. So that's something I think we can be extremely grateful for. But really, I think from my perspective, one of the key differences was, and really was one of the reasons why we were able to contain the MERS and the SARS outbreaks the way we were, was that people were only shedding the virus when they were symptomatic. So it made it much, much easier to, easier in air quotes, of course, but it made it much more containable, I think, because obviously those people were put in isolation wards in clinical settings, and so the spread of the virus was able to be much, much faster contained. Of course, that's absolutely not the case with SARS-CoV-2, where obviously we're very well aware that there are many asymptomatic spreaders, and that indeed you can spread the virus prior to even being symptomatic at all. And so really the epidemiology of those viruses, albeit that they're all coronaviruses, are really quite distinct. And so I think there are things that we can learn, and I think there are things that are really very, very different between the three diseases.
Yeah. Okay, another question that was brought up earlier is, another two-part question. One, we're learning that collaboration and already having set up some relationships can really help speed along these vaccines. And then the second part of the question was about how quickly do you think you'll be able to get FDA approval or get to the next phases of the clinical trial? And it might be good if you could just explain the different phases real quickly of the clinical trial. Either of you could start with that. I think you covered it in your slide, but maybe to summarize.
Dr. Dai, do you want to go first?
So maybe I missed the first question. So it's what?
Oh, the first question is, how important do you think the collaborations that you already had set in place help speed up the development of your vaccine?
Okay. Yes, we actually in our hands we have a cooperation for MERS before. Actually, as I show in slides that we started in MERS in 2005 for the vaccine development, and we proved the concept, and then we set up the cooperation with the company a few years before and set up the system. So, which when the SARS-CoV-2 comes, we can quickly and reactive. So we don't even prove the concept. We just set up the stable cell line just to bet it works. So I think for future, then definitely we'll have the other coronavirus because they are already adapted so good for the animal and will come to the human. So there are some emergence. So I think we should have maybe a technique storage technique that can prepare for that one. So for example, we set up the collaboration now, the system or the cell line, which can quickly reactive to the future. So.
Yeah, that seems important.
And I think when it comes to a global pandemic, really the only solution is a global collaboration. And I would say that as awful as the situation has been for the last 11 months, the collaboration that I've seen in the scientific and the clinical community has been absolutely remarkable. And, you know, when we think about potentially vaccinating every person on the globe, there is no way this will be able to be achieved if we don't have global collaborations from logistical perspectives, from manufacturing perspectives, from regulatory perspectives. These are going to be absolutely key. And I do hope that people can the public maybe can see how the fact that really we are all trying to work together as developers, as nations, as hospitals, as clinical sites to really work towards that goal. And I have every faith that we will have multiple effective vaccines by the end of this effort.
Okay. There's a question that came in for Dr. Broderick. Will your vaccine be a single shot or will it need a boost? And then the second question is a really old question about DNA, delivering DNA to a person, and are there any chances that it could cause like malignancy or cancer? I think that would be assuming that the DNA got incorporated somehow into one of the cells. So I guess the question is about your delivery system, and then whether you need a boost, and then the long-term safety.
Yep, absolutely. Let me start with you're absolutely right, Julia. That's kind of a historical question related to nucleic acid vaccines. But, let me be very clear about that. There has been multiple demonstrations that, integration of DNA vaccines is not a concern. To the point where now, from our perspective in the US, the FDA does not ask for any biointegration studies prior to starting. There's very much the WHO also accepts that integration into the genome is not a concern. So, in actuality, I think DNA vaccines are an incredibly safe vaccine modality. Coming back to the second question about the boostability, yes, our profile for this vaccine will be 2 shots, 4 weeks apart. So you get your shot on day 0, and then you come back a month later to get your boost. And that's one of the very nice elements about DNA, is that in actual fact, you can sort of boost ad nauseum. You can boost really infinitively. And that's in fact the case of what we do with our cancer patients. We don't have any anti-vector issues, so you can, keep giving DNA essentially forever if you need to. And as we look at this potentially as being something where we're going to require annual shots, that's really quite a nice characteristic to have.
Mm-hmm. Okay, I think we only have a few minutes left. Just one more question that seems to be coming out as a theme for the conference is the cold chain, and I think that the DNA one was already answered, but how about the cold chain for the receptor binding domain dimer? Will that be a -80, or do you expect it to be able to be distributed around the world?
Yeah. So far we are we're using 2 to 8 centigrade. So it's just 2 to 8, yeah. So no need to frozen cold chain. So it's so far it's stable for a long time.
Okay, is it in a fluid then? Like it'll come in a liquid that's ready to be injected?
Yeah, it's
and
in liquid.
Refrigerated.
No, not refrigerated. It's in liquid.
Yeah. Okay.
And in the 4 degree, 4 degree refrigerate.
And did you say whether your vaccine would need a boost?
Yeah, it definitely needs because the protein subunit 1 needs to boost. So we tried 1 boost and 2 boosts, and both shows induction for neutralizing antibody. It depends on we still to determine with using one boost or 2 boost. So maybe can test in the next phase 3.
Okay. And did you guys have any closing statement you wanted to make about your talk or the questions?
I guess I would like to maybe offer sort of a glimmer of hope that certainly this has been a rough, terrible year really for everybody, and my heart goes out to anybody who's lost anyone with this pandemic. But I do believe that all the developers are working day and night towards this, this needed vaccine, and that I have no doubt that we will get there, and there will be multiple effective vaccines for COVID-19.
Great. Dr. Dai, do you have anything you want to say?
Yes, I really appreciate everybody's here to join us the meeting and to discuss the issues. I think in the future I really want the vaccine can be. I think it really needs the global really needs a lots of amount of vaccine. So that's why we have so many developers using different platform. So we calculate if you have 2/3 of the population to get vaccinated to get herd immunity. So you really all the company to produce the COVID-19 cannot take so far. So I just hope that more and more a company can get their vaccine approved and to join into this supply and to end this pandemic soon, and to back to the normalcy of before.
I agree. Thank you very much. And then we could all get together in one room too for the next conference. All right. Thank you. That was it for our time.
Okay. Nice to meet you.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound. Where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
المملكة المملكة استضافة قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound. Where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
المملكة استضافة قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
Good morning, good afternoon, good evening, or good day. It is my pleasure to moderate this session. My name is Manal Al-Amri. I'm the Chairman of Innovation and Entrepreneurship at King Abdullah International Medical Research Center. I'm also the Head of Developmental Medicine. This session will focus on other vaccines, such as the vaccine developed by Moderna, Johnson & Johnson, and Baylor India vaccine. Our speakers for this session are Dr. Tal Zaks from Moderna, Dr. Catherine Taylor from Johnson & Johnson, and Prof. Maria Elena Potasi from Baylor College. I would like to introduce now our first speaker, Dr. Tal Zaks. Dr. Zaks is the Chief Medical Officer. He oversees clinical development and regulatory affairs across Moderna. Prior to joining Moderna, Dr. Zaks was the Senior VP and Head of the Global Oncology at Sanofi, where he was responsible for all aspects of oncology, drug discovery, and development and commercialization. Dr. Zaks begins his industry career at GlaxoSmithKline in the Genetics Research Group, where he built the Oncology Translational Medicine team and led the translational research. In addition to his industry work, Dr. Zaks is an associate professor of medicine at University of Pennsylvania, UPenn. Dr. Zaks holds a medical degree and a PhD degree and conducted postdoctoral research at the U.S. National Institute of Health. He completed his clinical training in internal medicine at Temple University Hospital, followed by a fellowship in medical oncology at the University of Pennsylvania. Dr. Zaks, the floor is yours.
Thank you so much, Manal, and thanks for having me. Assalamu alaikum to everybody. It's a real pleasure to be here with you. Let me just see that I can share my presentation. This worked a minute ago, but for some reason I don't see. Can you see my presentation?
Not yet.
I apologize for this. For some reason, the little button to share has gone. Give me a second and I'll try to upload it. I'm sorry, this is the first time this has happened. Manel, I suggest that we switch the order and I will try to log back in and back out because...
It's fine. Should we start with Dr. Catherine?
Now I can see them.
Dr. Zach, it's working now.
It is? Okay, perfect. Let me get... Are you able to see the presentation?
Yes.
Okay, I apologize everybody for that delay, so let me get started. I will start with just a broad outline of what our platform is. Essentially, a messenger RNA technology starts from DNA, but really uses the genetic information as a template for the vaccine that we're making. And what we do is combine that template with enzymes and buffers and nucleotides to generate the messenger molecule. And so this is all synthetic biology. It happens in an aqueous phase. And at the end, we complex it with the lipid nanoparticles that protect it, and that's our drug. And because this is starting with genetic information, it's a synthetic in vitro approach, it is actually quite fast. And that's what enabled us to start really quickly and get into the clinic. Now, here's what the actual vaccine does. We have the mRNA that encodes for the spike protein. When that gets into the cell make the spike protein on their own, and that ensures high biological fidelity because it's our own body cells that are making the protein. In the context of what we know about SARS-CoV-2, this was actually scientifically relatively straightforward as soon as the sequence was known because of the work that had been done already on SARS, MERS. We knew that this was the thing that we wanted to target. In terms of what we've done with our technology, this is actually a relatively straightforward antigen. It's just one antigen. We've done much more complicated vaccines in the past. And so we went into this with a high degree of confidence that we would get what we were looking for. And us and the NIH and the scientific community developed the tools to measure it along the way. To get to a very high level of what we know from preclinical models, I'd make the following points. First, we have been able to get really dose-dependent neutralizing antibodies in every species we've tested it. I'll show you the data in non-human primates. We see both binding antibodies and neutralizing antibodies. And what we've learned is that although sometimes only a single shot, just a prime, may be enough to protect, when we're able to give a boost a few weeks later, we get a much higher immune response and much more neutralizing antibodies. And we think neutralizing antibodies are really critical for protection. Number two, as it relates to protection, we've shown that vaccination can protect every species we've tested so far, mice, hamsters, and non-human primate. And we've shown the ability to protect from viral replication both in the lung and in the nose, and I'll show you an example of that. In some cases, we've even shown that a prime only can already protect animals from viral replication. And finally, there's been a lot of theoretical concern of disease enhancement. We have been looking at that carefully using all the available preclinical models. And at the end of the day, not only do we not see any evidence of that, the totality of data for the kind of immune response that an mRNA vaccine will get goes the other way. In other words, if we think of immune enhancement as something, certainly the respiratory immune enhancement has been associated with a Th2-like allergic immune response, we see the opposite. We see a very consistent Th1 biased response, and that's what we would expect from this platform. It's also the basis of other applications of this platform, like cancer, for example, for cancer immunology. So what we've known and what we've shown so far with this platform suggests that we can expect a very strong Th1 biased protective effect in the clinic. Let me walk you through a little bit of that clinical data. You can see here on the non-human primate, this was published in the New England Journal several weeks ago. You can see on the top left the binding antibodies. They come up fairly quickly within 2 weeks, and you give a boost. And then 2 weeks after the boost, you see further increased by about a log. And you can see the low dose in blue, the higher dose in red. And then if you look on the right, you see the neutralizing antibodies at the high dose come up even after one dose. But certainly the boost gives everybody a very nice boost. And so what we can see from this curve is there is a dose response effect. We can see binding antibodies, but the neutralizing antibodies come on strong, especially after a boost. Now, how strong is that? We can actually see in comparison to convalescent plasma on the bottom graphs. So here you see box plot data. You can see starting at the far left the binding at the low and high dose for the non-human primate, and it's compared to convalescent plasma, meaning these are actual serum from patients who have been clinically sick with COVID-19. And that's sort of the benchmark we use to figure out where are we in the dose response curve. And what we see here, if you look at neutralizing antibodies, is the 10 microgram, the low dose, already gets us to the level of convalescent plasma, no matter what kind of assay you use. At the high dose, you exceed it. And that for us was really an important point, because as you'll see in the clinic, we were from the get-go shooting not just to get to the range of convalescent plasma, although it may be enough, but to try and achieve the maximal immunogenicity we can, thinking that a higher effective neutralizing antibody should lead to both higher protection and a longer durability of effect. So how does that manifest in terms of protection? You can see here the data from the same non-human primates. There are 8 primates per group. And what we show on the left is the bronchoalveolar lavage. This is looking for subgenomic RNA, that is evidence of viral replication. And you can see at the dose of 10 microgram, 7 out of the 8 non-human primates have no replication in the lungs starting in day 2. And you can see the PBS, the placebo, on the far left. So clearly the virus is getting into their lungs, and it takes some time for their immune response to kick in. But the virus is not able to establish itself in the lungs from the get-go in the NHPs that have been immunized. What's interesting is on the right, at the high dose, we're also able to prevent almost completely abrogate viral replication in the nose. And so as you infect these animals, and they were infected with relatively high doses compared to other studies, you can see that there is complete protection in most animals at the high dose, even from viral replication in the nose. And we think this could have important implications, not just for the ability of this vaccine to prevent disease, but also the ability to ultimately prevent transmission. And so let me walk you through now the clinical development program. The program started relatively quickly. Once we had the sequence, we put it into production. The National Institutes of Health geared up to do the first in-human trial. This was always envisioned as a large, conservative, and quick development plan, in the sense that we would do first a phase 1, then a phase 2, then a phase 3. However, we would use safety from phase 1 to go into phase 2. We would use immunogenicity from phase 1 and safety from phase 2 to go into phase 3. And that was the plan. And indeed, we, together with our collaborators at the NIH, have been able to execute against this plan, such that the phase 2 started on track in May. And at the end of July, we started the phase 3. In fact, this phase 3 has now been completed its enrollment. And I'm going to walk you through 3 points of data. This is all in the public domain. It was published in 2 manuscripts in the New England Journal, first for the young adults and then for the older age groups. And I'm going to start with safety at the 100 microgram dose. The first phase 1 demonstrated the 100 microgram to be the optimal dose in terms of achieving robust immunogenicity. And we had no need to go to higher doses at 250 that may be associated with less tolerability. And so at the 100 microgram, what I'm showing you here is the adverse event profile in terms of the solicited adverse events. So when you talk about vaccines, you talk about safety and tolerability. These are 2 slightly different things. Tolerability relates to the expected solicited adverse events, those so-called flu-like symptoms that we expect a potent vaccine will have. And indeed, what you can see here is that after the second vaccination, you see more of them in correlation with mounting a strong immune response. And what you see are the expected adverse events of flu-like symptoms, fatigue, chills, headaches, myalgia, etc. You see some local, primarily pain, but you don't see much exacerbation of the pain on the second vaccination. So I think that's likely less specific to an immune response. All of these were transient, all of these were anticipated. And in terms of safety, and that is the really serious, unanticipated, and less common events, We did not see any related serious adverse events in this trial. We did not see any notable laboratory abnormalities, and the totality of our experience on this vaccine is consistent with what we've seen in our other vaccines that are in phase 1 and phase 2. I should note that this is, broadly speaking, consistent with the safety profile that we've seen, albeit yet in a blinded fashion, from the phase 2 and emerging phase 3 trials. Here are the neutralizing antibody data from the clinical trial. What you can see here is that the neutralization is at the top range of the convalescent serum. Now, there's 3 points to be made from these data. First, you can see that a prime will already get you in some subjects neutralizing antibodies, but it's really the boost that gets you to consistently high levels. So a prime boost regimen. If you look at the response, you will note that nobody's left behind. Everybody achieves a level of neutralizing antibody that is quite significant. And you see that there is no difference as it relates to the age. So whether you're an 18-year-old, a 75-year-old, or in between, people have the same level of neutralizing antibodies. And we focus on neutralizing antibodies as the relevant factor here because what we've learned, I think, is that neutralizing antibodies are the relevant part of the immune system that protects us when it comes to preventing infection. That's been true, I think, of passive transfer studies. And in fact, monoclonal antibodies that are neutralizing are now being more and more used as therapeutics. And so I think the overwhelming data suggests that it is the neutralizing antibodies that are what we should be caring about. Now, there's been a lot of discussion on T cells, and let me assure you this vaccine does induce T cells. And I'm showing you the data for the Th1/CD4 positive T cells. And I think this is important for 2 reasons. Number one, you can see, again, there's no difference in the different age groups, whether you're 18 to 55, 56 to 70, or above 71. You already see a Th1 type response at day 29 and clearly a boosting effect. And the important point here is twofold. Number one, it is the same Th1 phenotype. No allergic-like Th2 cytokines are coming up, and so no basis for concern for enhanced disease. And the second point is, while I don't think these T cells themselves are ameliorating disease, I do believe these T cells are underpinning the strength of the immune response that you see the B cells manifesting in terms of level of antibodies. And since durability of effect is going to be a function of how many antibodies you have and how many memory B cells you have, I think the ability to show the right kind of T cell support is what's giving us optimism and confidence that we should see a durable effect here, although that's, of course, to be determined. So let me spend 2 minutes on the phase 3 design. Fairly conservative design, large trial, independent data safety monitoring board, an important strata that has subjects above the age of 65 and with comorbid conditions. You'll see what it shakes out to be in a minute when I show you the demographics. A 2-year study. Primary endpoint is prevention of clinical disease. Secondary endpoints of prevention of both asymptomatic infection and severe disease. The definition of disease here is having 2 systemic symptoms or 1 respiratory symptom and a PCR test. So a conservative measure. And this is what the demographics look like in terms of the population. We actually enrolled quite a diverse population here, 63% Caucasians, 20% Hispanic or Latinx, 10% Black or African American. So roughly mimicking the census of the US and I think broadly applicable to global populations. We have an age breakdown that you can see a significant proportion are middle-aged or older, that gender distribution as expected. And in terms of comorbid conditions, we had 8,000 plus out of 30,000 subjects here that had significant comorbidities, whether it's severe obesity, diabetes, lung disease, or cardiovascular disease. And you can see the breakdown in totality is that 42% of the trial population is actually at risk of severe disease should they get infected. And so we anticipate the data when we see them to be informative for the people we truly want to protect here. If you look at the other parameters of this study, 22% were healthcare workers, and everybody here in this study should have been somebody who's somehow on the front lines, people who are at risk of infection, such that if you're like me and all you're doing all day is giving talks from the comfort of your living room, you should not be enrolled in this trial. This is really was designed for people who are at risk within their community. And that's being borne out by the cases that we see emerging. And so as we look forward to the analysis plan here, the first opportunity we will have to see whether the trial is successful is when we get 53 cases. We'd have to be at least 75% effective to pass that threshold. The trial is ultimately designed to be able to demonstrate at least a 50% threshold. The rate at which cases are occurring now means that we'll probably see this first interim sometime this month. And by the end of the year, beginning of next year, we should actually be all the way through to a primary analysis here. So in summary, on the clinical side, we've seen safety and efficacy that's not surprising, on par with what we would expect from a potent immunogenic vaccine. Nothing that leads us to undue concern so far. The data has enrolled smoothly. The trial has enrolled smoothly, completed its enrollment on the phase 3, and we're monitoring closely for when we can have data. The last minute I'll spend here is actually on something that's as important as having data, which is having vaccine available once we have data. We have leveraged the history and experience we have with this platform over the last many years to actually have a CMC supply chain that is favorable in the sense that we use existing infrastructure for the most part in most places of the world. I recognize that in the Middle East, the minus 20 Celsius degree is not a trivial undertaking. That being said, this is existing infrastructure that we all have in modern facilities, whether it's minus 20 or 2 to 8. In fact, many current vaccines on the market already deploy this. And so we anticipate the ability to use existing infrastructure for the supply chain. This is a simple vaccine, no preservative, no dilution required. The first launch will be with multi-dose vials. And we've been already ramping up production both in the United States and in Switzerland to be able to supply the US and the world with up to 1 billion doses by the end of next year. With really, I think, in the US, we'll start to have doses in the millions by the end of this year. From a global perspective, I think first quarter is when we really start ramping up in Europe. I think I'm up to my 20-minute mark, so let me thank everybody, particularly our collaborators at the National Institutes of Health, and of course the trial participants who have volunteered in the tens of thousands to join us on this journey and help determine the safety and efficacy of this potential vaccine. Thank you.
Dr. Manal, would you like me to share my screen?
Dr. Manal, you're on mute.
Okay.
And I believe you can see my screen now?
I think it's clear, yes.
Okay, thank you. Would you like me to go ahead?
I'll give the introduction. So now we'll move to our next speaker, Dr. Catherine Taylor. Dr. Catherine Taylor is the Vice President, Medical Affairs, Therapy Area Strategy for Jason EMA, Europe, Middle East, and Africa. Prior to that, she worked in Middle East as Regional Medical Director, EMA Emerging Market. Between 2016 and 2019, she was the Medical Affairs Lead in Hematology for Jason Oncology across the EMA region. Dr. Taylor holds a medical degree from King's College, London, and is a member of the UK Royal College of Physicians. She also holds a diploma in pharmaceutical medicine and has completed her UK specialty training in pharmaceutical medicine. Prior to joining Jason in 2015, she held positions in UK, in EMA, Regional and Global Medical Affairs at Pfizer and Stalis. You can start, Dr. Taylor.
Thank you very much, and thank you very much for this kind opportunity to speak at this symposium. I'm going to be sharing today with you some of our COVID vaccine efforts and clearly discussing our commitment to the patient. As Dr. Manal said, my role previously to this one was the Regional Medical Director for the region, and I'm actually still based in the United Arab Emirates, so this is an area that's very dear to my heart. So I'm going to talk about Janssen. Janssen is actually the pharmaceutical arm of Johnson & Johnson and our promise. So at Janssen, we are creating a future where disease is a thing of the past. We wish to have a world without diseases. And how will we do that? Well, I think I should talk a little bit more about the Janssen commitment. Firstly, we're committed to developing medicines and solutions that transform individual lives and, more importantly, solve unmet needs of our time. And I think we can all agree that the global pandemic of COVID is one of them. But in addition to that, we also acknowledge that quality and safety drive every step of product design and manufacturing, and that includes after the medicines reach our patients. So specifically, if we're talking about the scientific response to combat COVID-19, I think I can quote no better person than Paul Stoffels, who is our Chief Medical Officer. We have been actively engaged in fighting pandemics for over a century. We have done it before and we will do it again. And I think this very clearly states our commitment towards a vaccine for COVID-19. So if I elaborate more as to what we're committed to, well, firstly, we have substantially accelerated the timeline to deliver a vaccine for COVID-19, and I'll go into that more in the next few slides. We've worked with our partners to screen a library of compounds so that we believe we found the optimal candidates, and through rigorous research and development, we can identify a potential treatment. And then finally, it's only through collaborating with regulators, healthcare organizations, institutes, and community worldwide can we ensure that we use the research platforms, our existing science, to control and maximize our outbreak expertise to stem the public health threat. So let me go a little bit more into our innovative vaccine technology platform, our ADVAT platform. So firstly, we take the synthetically generated spike protein, the DNA encoding the spike protein. We transgenically insert it into a non-replicating adenovirus. And this is a common human adeno cold virus. This is injected into the patient. And as we expect, the release of genetic material encoding the spike protein is released, which then results into the immunogenicity that we would hope for an effective vaccine. This is a timeline of our development, and this will, I'm sure, be familiar to the audience. So firstly, we quickly responded when the first cases of COVID were emerging in the world, and vaccine development was initiated in January. By March, we announced that we had a lead vaccine candidate with 2 backups. Now, some people might say, well, that was 2 months. You know, why did it take you so long? And it's primarily through what I've alluded to before, is finding that right candidate in terms of immunogenicity, but also in terms of stability, in terms of manufacturability. We wanted to ensure that really we were bringing the best medicine that we could to the market. In July the 20th, our first in human phase 1/2A started, and we actually received our interim results from this trial in September, and I'll be sharing that with you later on in the study. Our phase 3 clinical trial is now initiated, and we're looking at the potential timing for emergency use in 2021. So I talked earlier on as to accelerating vaccine development. We have achieved all this, and we believe we will go to emergency use within 15 to 18 months. And this really proves how quickly we've got to this point, because typically vaccine development takes 5 to 7 years from phase 1 to launch. So acknowledging the global need, we've really done our best while ensuring that it's in a safe environment to do so, to bring a vaccine as quick as we can. So like Dr. Zaks from Moderna, I will quickly go through some of our data. So I'll start off first with the preclinical data. So this is where we immunized several groups of rhesus macaque monkeys And actually, we used not one candidate, but 8 in this case, because again, we really wanted to find the one that showed the most promise into our clinical trials. As you can see from week 2, all 8 of these vaccine candidates actually showed promising immunogenicity responses in terms of pseudovirus neutralizing antibody titers. However, by week 4, the bar in the green shows that the AD20, what we call the AD26-CoV2 variant, actually showed the most titer increase by nearly 6-fold. And more encouragingly, even in the convalescent phase, this showed a sustained response compared to the controlled groups and compared to convalescent humans and primates. So what I also wanted to show was that a single dose of our AD26-CoV2 variant protects both the lower and upper respiratory tracts of the SARS-CoV-2 challenged non-human primates. On the left, you can see what I've got. The chart at the top is the bronchoalveolar lavage, and the chart at the bottom is nasal, so representing upper and lower respiratory tract. And you can see in the control groups that the viral copies continue to remain very high. However, in the AD26-CoV2 variant, in the macaque monkey group, you can see that after one dose, in the lower airways, there was the viral load was completely flat, and also this showed in the upper respiratory tract near virtual protection, with only 1 out of 6 monkeys demonstrating a low-level response. So with these encouraging results, we then went into our first-in-human studies using our optimized lead candidate, the AD26-CoV2 variant. And I'll quickly go through the schema here. So this was a study that was 1,045 patients, conducted primarily in Belgium, the US, and looked at healthy adults in the 18 to 55 year age group, as well as 65 years and above. What we see here is in cohort 1, we looked at safety and immunogenicity in younger adults, and in cohort 3, which is the same cohort but in older adults. You'll see in between there is also a cohort 2, which is actually looking at the effects of duration of immune response and also the effects of boosting. But this is actually, the enrollment is due to start this month. So what then happened was that we actually received our interim analysis, and this is actually based on cohort 1, but also a small subgroup of cohort 3. And while I can't share the full data because it's actually yet to be published in a peer-reviewed journal, it's actually in the public domain. It's been shared on medRxiv, and therefore it's a little bit clunky, the format that I'm presenting, but I can quickly walk you through what the conclusions were. So in the interim analysis of the first-in-human study, we showed that a single dose of the AD26-CoV2 variant, either low or high dose, is safe, well-tolerated, and highly immunogenic. Because we found similar immunogenicity in both dose levels, we actually selected the lower dose for further clinical evaluation. We've now initiated our first phase 3 study in the US, and later on, these will include sites in Africa and South and Central America. And we will therefore, in our study, evaluate the efficacy of a single dose of the lower dose of our candidate. We're also planning a host of other studies in our clinical plan, the first of which is an additional phase 3 study, which will also assess the efficacy and durability of a 2-dose schedule. So while we believe from our preclinical data that 1 dose will be enough, we believe also in exploring the 2-dose vaccine so that we know which one is best when we bring the vaccine to the patient. I'm sorry, this might turn out a little bit small, but I also wanted to reflect a little bit on the side effect profile, and generally to say that, as I mentioned, it is well tolerated vaccine. So you can see, I hope, that any side effects are generally grade 1 and 2. And the most frequent adverse events are those that we would expect with vaccinations, such as site pain, some fatigue, headache, and myalgia. There were no grade 4 side effects reported. So I just wanted to then now take you through our phase 3 study. This is the Ensemble COV 3001 study. And again, this is a multi-center randomized double-blind placebo-controlled phase 3 study, which will evaluate the efficacy and safety of our Ad26-CoV2 variant against placebo. So you can see we're again starting off with healthy adults in greater than 18 years of age, and there will be cohorts within this subgroup which have comorbidities. And the estimated enrollment is 60,000. We will be looking at a single intramuscular dose, as I mentioned, or placebo. And we were looking at endpoints of the reduction of the number of participants with first occurrence of molecularly confirmed moderate to severe or critical COVID-19 seronegative status. I wanted to end my talk on, again, affirming that Johnson & Johnson do have a plan against coronavirus. So to recap, as soon as the novel coronavirus sequence became available in 2020, we were immediately on the case, began our efforts to research the potential vaccine candidate. I spoke already to the acceleration. We condensed what is usually a 5 to 7 year program into something much shorter, commencing our clinical trial in the second half of July, and we even brought that forward from the original date of September 2020. So we really are invested in finding a medicine as soon as possible. Partnership has been very important, and as I mentioned, we have collaborated, and in our case with BARDA, with a commitment of more than $1 billion of investment to co-fund the vaccine for research, development, and clinical testing. And our goal is to supply more than 1 billion doses globally through the course of 2021. We have begun production at risk, and we are committed to bringing an affordable vaccine to the public on a not-for-profit basis for emergency pandemic use. Thank you very much for your attention.
Thank you very much, Dr. Taylor. So we'll move to our third speaker. Unfortunately, she wasn't able to join us, but she shared her presentation. I will present Professor Maria Potazzi. Professor Maria Potazzi is an Associate Dean of the National School of Tropical Medicine, Professor of Pediatrics, and Co-Director of Texas Children's Center for Vaccine Development at Baylor College of Medicine in Houston, Texas. A global thought leader, she has received national and international awards, has more than 150 scientific papers, and participated in more than 250 conferences worldwide. She's a member of the National Academy of Science and Honduras and an emerging leader in health and medicine, scholar of National Academy of Medicine in the US. Dr. Potazzi currently serves as a co-chair of the new vaccine task force of the Lancet Commission on COVID-19. In August 2020, Forbes LATAM selected Dr. Potazzi as one of the 100 most powerful women in Central America. Dr. Potazzi obtained her bachelor's degree in microbiology and clinical chemistry from the National Autonomous University of Honduras, and a doctorate in molecular immunology and experimental pathology from the University of Florida. Her postdoctoral training in cellular biology was completed at the University of Miami and Pennsylvania. Can we share Dr. Potazzi's presentation?
Thank you very much for the introduction, Dr. Alamiri. It's a pleasure to be with you this morning. I'm going to give you a very brief presentation and tell you a little bit Children's Center for Vaccine Development and how we have been advancing yeast-derived COVID-19 recombinant protein-based vaccine and our, of course, collaboration with an India-based vaccine manufacturer, Biological E. But I wanted to first start a little bit putting it in context to remind, I think, everybody, especially the role and the importance not only, of course, of partnerships. As you know, working in collaboration, it's always more efficient and, of course, even resource savings, but especially how academic institutions play a pretty big role, and now including for COVID-19, how it has really advanced not only COVID-19 specific activities, but just in general, how institutions such as pharmaceutical companies or biotechnology companies, of course, find a value of forging these collaborations with academics. And again, it's because usually within the academic framework, we, of course, have the start of the basic science. We have a lot, of course, of innovation, and organizations look for how to kind of chase that innovation, ultimately also acquire some of the knowledge, especially specialized knowledge, as well as technologies. Occasionally, because we have already advanced studies, it could even increase in speed and agility through the, of course, development. And, talking, about vaccine development specifically, for example, our group focuses on what we call an integrated development strategy, which within an academic institutions, you can have access to how do you access potential targets, even doing some very interesting, sophisticated genetic engineering or reverse vaccinology. Nowadays, more and more, including our institution, has an advancement in process development and production and scale up, developing assays that are what we call regulatory enabling for release, or even eventually its use for stability indicating assays. As you know, institutions have a lot of experience in setting and using preclinical models, whether they're in vivo or in vitro, to assess all sorts of immunogenicity and efficacy. And many times, as you can see, even in the context of COVID-19, a framework where you can even do clinical trials. So I know that especially our history in collaborations with, as an example, the Kingdom of Saudi Arabia, we have forged this academic partnerships, but also academic linking to biotechnology companies, where we can certainly advance vaccine development. And therefore, if you then apply this to COVID-19 vaccine development, besides the fact that I'm going to tell you the story of having our own little horse in the game, our horse in the game fits within the puzzle of, what I call the three pillars of achieving success through the development of a COVID-19 vaccine, which is the fact that our group have expertise in the production aspect and creating what we call the prototype of the actual vaccine product. We also have the ability to do some evaluation and preliminary preclinical assessment of such prototype. But at the same time, we are very cognizant of the end of the line, or what's the end game, which is who we want these vaccines to be delivered to, the fact that they have to have global access. They therefore have to have suitable processes that could be affordable, adaptable, adoptable, and eventually for equitable delivery. So in fact, Texas Children's Center for Vaccine Development, which is our vaccine center, which is based in Houston, Texas. We, in fact, have already now 10 years in the Houston area in the Texas Medical Center. But before that, we actually had a prior 10 years of experience working, again, as a product development partnership based in Washington, D.C. And the moment we arrived in Houston, in fact, we came with an award, a grant that was given to us from the National Institutes of Health of the United States, which in fact was specifically to develop a coronavirus vaccine, both for SARS as well as for MERS. But, you know, the one that was advanced more rapidly was our vaccine against SARS, which was a collaboration, again, a partnership between the New York Blood Center in New York City under the leadership of virologists such as Lang Ying Du and Shibo Zhang, together with the University of Texas Medical Branch under the leadership of Kent Seng, where we brought a SARS receptor binding domain candidate from 2011 to around 2015, to the point where we were had a whole ability of technology transferring to a pilot manufacturer, which in this case was Walter Reed, and we ended up actually manufacturing a drug substance vaccine candidate. Unfortunately, as you know, I think one of the challenges we have seen throughout the years of maintaining sustainability and continuity, especially of groups like ours, which are, of course, in the nonprofit sector, not led by big pharma or biotechnology companies. It's this stop and go of getting access to funding. We therefore had to pause, a few years, our endeavors to look for coronavirus vaccine, not only candidates, but, the evaluation of such. Now, however, the lessons learned that we had between 2011 and certainly 2016 and even beyond, because we kept doing stability assessments on such prototypes, really allowed us, as you can see in the blue line, to, in less than 6 months, replicate what we had done practically in 5 years, the development of a new SARS-2 RBD vaccine candidate, which now I'm going to tell you the story of how we ended up eventually even licensing this technology to Biological E, the Indian manufacturer. So if you are interested in seeing in more depth what we did, how we were leveraging the lessons from our SARS program, we have, of course, various publications, not only on the product, on how we developed the product and the prototype, but also the preclinical evidence supporting the functional and the, of course, efficacy using a mouse model, using a mouse-adapted SARS strain. And, the philosophy of our center is, again, since we have our niche, which is really to create the prototypes vaccines, meaning we actually make the vaccines in the laboratory, mocking and already thinking of who is going to be the manufacturers that we're going to need to transfer that technology. So we adapt the processes. We have a document control. We have quality assurance, but we mock the process development activities such that we could very readily transfer even the documentation packages suitable for rapidly enabling, of course, any regulatory dossier. While at the same time, we, of course, also do all the preclinical assessments of those prototype vaccines. As an example, then, I'm going to tell you the story of how, using, again, the information from the SARS, we knew that we work primarily with the yeast expression technology, the platform. We know yeast certainly has a tendency of hyperglycosylating proteins when they're being produced by the host. So we knew from our experience from SARS that, for example, in this case, if you take the wild type sequence of the receptor binding domain of SARS-2, we knew that most likely we were going to need to do several modifications. So one modification is the deletion of the N-terminus first amino acid. And that already we knew from SARS that it was going to rapidly reduce the hyperglycosylation produced by the yeast. But at the same time, the SARS-2 construct also had an extra cysteine that was unpaired and therefore would probably interfere and create intermolecular disulfide bonds and would probably have the likelihood of creating dimerization or even aggregation of the molecule. So here is just a representation of indeed, when we try to engineer a wild type version, and one with just the first and glycosylation site deleted, or when you not only delete the end glycosylation but also mutate the C-terminus, we in fact ended up just on the pure fact of easiness of production, quality control, and eventually we even confirmed that even if we do some process development scale-up process evaluation, we compared again with the expectation of what we were seeing in our SARS experience, that we ended up seeing much higher yields. We get around 300 milligrams of the protein, recombinant protein, per liter of fermentation supernatant. And even if you look at just the not only quality of the material, the yield, the type of production purification steps that we use, that was the ideal candidate to select. We also, of course, go more deep into not only doing very simple orthogonal type of quality control assays, but we also do studies such as, comparing its biophysics by dynamic light scattering, where we again, in fact, regardless of whether it's wild type or modified protein, we see very, very similar secondary structures. Of course, as you can see, it's preferable to use the RBD219 with the both modifications because we also keep it in a monomeric form, and we now know that it is, of course, very stable. Of course, more importantly is how functional this protein is. We do a couple of studies. We do studies to see how the protein itself is able to bind to a human ACE2 receptor, which is the paddle on the left. But we also, after formulating it on aluminum hydroxide, which is our preference baseline formulation scheme, we also titrated down the binding capacity of the protein on alum, but at the same time seeing whether we see any differences in being able to, the protein after its binding or absorption to aluminum, it has its efficiency in binding to the receptor. Of course, then we do immunogenicity studies, and this is just a representation of one simple study where, again, we looked at the different binding ratios between protein and aluminum, comparing wild type, which is the red, compared to our modified protein, if it's there's any difference in its immunogenicity. As you can see, they're not statistically significant for the most part, even if you change the ratio of protein absorbed to the aluminum. But when we also do pseudovirus assays, I think we have some very clear indication that the RBD, even when it's just bound on an aluminum formulation, independent of the ratio, probably even up to ratios of 1:4, we see some very high levels of virus neutralizing antibodies. So this was kind of our preliminary data that, of course, when we got approached by Biological E, they were able to do, of course, in-depth due diligence of not only our program, our documentation, our suitability, and we are very proud and very privileged to work with an organization such as Biological E. They, of course, have secured a non-exclusive license from us. We transfer all the know-how technology, and I'm happy to report that we've already heard that Biological E have submitted their dossier to the Indian regulatory bodies. They have gotten a green light to proceed. And we're soon, most likely even as soon as next week, hear that they're going to be able to start recruiting cohorts for starting a phase 1, phase 2 clinical trial. I think for us, the big excitement is they, of course, already scaled up the process of production to the point where we know they can produce up to 1.2 billion doses of drug substance or recombinant protein candidate. They will be advancing an adjuvant system, which will include the aluminum hydroxide plus a second immunostimulant. We know that they're going to eventually advance to reach registration and licensure in India, with the intent that they already have committed to work together with, of course, COVAX, Gavi, WHO, to ensure that eventually this becomes a global access, affordable vaccine. I have to end this presentation certainly by thanking not only our team at the Texas Children's and Baylor, but of course many collaborators. These are just a few faces of those who have been in the front lines day and night, doing, again, in short of 6 months, all the work to have this ready and succeed in transferring this technology. We look forward to possibly even have more collaborations. And on top of that, of course, all our partners from our prior efforts, even groups such as Millipore Sigma, who always helps us in improve the types of processes, the process development activities that we do in our labs. PATH has been instrumental, certainly helping us working with Biological E and looking aspiration of making this vaccine a global access vaccine. IDRI, who helps doing a lot of the formulation science, and we've been working with them for almost 2 decades. And of course, thanking all our generous supporters that have been very pivotal for us to be able to advance this work. So I'm going to stop there. I thank you, and I hope that this information has been of importance. And if by any chance you're seeing this as a recorded presentation and I am not able to connect live, feel free to reach me if you have any questions. You can certainly look for me in the Baylor College of Medicine website. My email is botazi@bcm.edu, or you can tweet or reach me through Facebook or any other social media. Thank you very much.
3, 2, 1, you're live.
Welcome back. So now we'll start our discussions, and I will ask some questions. I'll start with a question to Dr. Zach. Given that the development of vaccines has been fast, how do you trust the safety and efficacy of this vaccine, other vaccines? Can you please elaborate on that?
Sure. Thank you, Manal. It's a good question. It's probably the first one I often get asked. And I think when we step back, you should look at the trials and understand why we've been able to move so fast. And there are 3 reasons we've been able to move fast. The first is having scientific platforms and technologies that allow us to start quickly. The second is we've been able to progress quickly because of terrific collaboration with regulatory agencies, and I think that's been true globally of many countries and many companies. And the third is that speed, at least in phase 3 trials, is a function of cases occurring. You need a certain amount of cases to be able to demonstrate clinical benefit. And typically in vaccines, you are trying to prevent something that occurs infrequently or rarely, and so you have to wait a long time. Well, unfortunately here, with the pandemic, the way it's being transmitted everywhere, certainly in the US where our trials are going, unfortunately, we've not had to wait a long time to see cases occurring, and that's why we anticipate that it will be quick. Now, the second part of the answer is to look at the phase 3 design. It's actually quite a conservative design. The efficacy is driven by very conservative assumptions on a 95% confidence interval that excludes 30% at the lower bound. And the conduct is one that's being overseen by independent data safety monitoring board comprised of NIH-appointed experts. So it's not me and the company that's going to tell you this thing works. It's actually an independent group of experts that are continuously looking over our shoulder for both safety and efficacy. So when the results come in, then we should all feel good that these trials were done with a very conservative stance and with independent monitoring. Now, the one thing that I think will be challenging is the speed, because cases are occurring fast, will mean that length of follow-up is going to be relatively short. So, you know, if something rare happens 2 or 3 years out, well, there's no way to know it until you're 2 or 3 years out. And if the durability of effect isn't there for 2 to 3 years, well, we won't know it until we get out 2 and 3 years. So I'd say two things to that. Number one, we, and I'm sure other sponsors, will continue to monitor the trial as well as the real-life deployment to continue to assess safety and effectiveness over time. So we're not done when this phase 3 trial gets unblinded by any means. And I think the second element is, you know, it's always any decision to deploy any medicine or vaccine is a function of benefit and risk. And the question is, in the US, when we have 1,000 people dying a day of COVID-19, you know, at what point do we believe that the benefits, once we've shown prevention of disease, would outweigh the risk of the unknowns? And so while the phase 3 trial has been designed for 24 months, it is inconceivable to me that if it works, we're going to keep everybody blinded for 24 months just to see what happens, right? I think many of these people fall into the 1A and 1B category of people at highest priority. You've seen the demographics. So I think while I'm comfortable that the trials are conservative and they're being conducted appropriately, we as a society are going to have to take a benefit-risk approach at the end of the day to using this vaccine. And I think that's not going to be up to the companies. It's going to be up to global regulators. Over.
So we'll go to Dr. Catherine. Thank you very much, Dr. Zaks. Dr. Catherine, what are the lessons from COVID-19 vaccine development that will make us prepared for the next one?
Thank you for the question. And Dr. Zaks gave such a comprehensive answer that he nearly covered the entire spectrum of vaccine development. So I need to find something he hasn't already covered. I think a couple of things that maybe I'll just choose to elaborate on. So firstly, it's something we took from previous vaccine development and we'll take into the next, which is actually, and Dr. Zaks mentioned this, is about reusing the platform, the legacy of a platform that we know can work repeatedly with different inserts is very valuable, right? So our ADVAC 26 platform, this is actually not its first use. We actually used it first in Ebola vaccine, which is actually now commercially available. And we're also continuing to use it in our Zika and HIV development program. So that's why we were able to swivel so quickly, because we already knew that we had this kind of very handy platform that we could then use the COVID insert. I think the other thing that has been mentioned already is about the interchange with regulators, right? We adapted very quickly this time, and really the partnership in early engagement with the Saudi FDA, with acceptance that we were going to perhaps use a dossier that was common. So rather than having to develop it for different countries, that the flexibility to say, let's make this quick so that we can get this approved as quickly as possible, and even to have the data package, you know, in different times, right? You know, kind of rolling submission. I think all of these things speak to a very strong will on both sides to relieve this pandemic situation. And I see Dr. Zaks nodding, so I think it's probably something that we're adopting as an industry.
Thank you very much, Dr. Taylor. So we'll take some questions from chat box. So this is for Dr. Zaks. Will the mRNA vaccine, would it be available for developing countries?
So I anticipate yes. I think that the cold chain requirements, I think they may be challenging, but they're not insurmountable. I think others have deployed with a cold chain of -20. So you get 6 months at least of storage and shipping at -20. Then it can be in the fridge today for up to a week. 12 hours in room temperature. Once you puncture the vial, 6 hours because there's no preservative. So it's the infrastructure of the cold chain that I think is the most challenging, but it's not. This is nothing new, and I think the developing world has dealt in the past and continues to deal with that kind of cold chain. So I think yes, the question becomes both supply chain and logistics and quantities over time, which is why Catherine and I were chatting before. I often say that my only two competitors in this race are the virus and the clock. I think we hope that everybody else succeeds, and we need many companies to come to the fore so that there's as many options available for humanity as we can muster as a society.
Thank you. There's another question. Does it have an impact on gene expression on long term?
Yeah, I smile. That's a common misconception. The answer is absolutely no. Messenger RNA, while it's a nucleic acid, actually doesn't work in the nucleus. And so this is a scientific truth that's accepted by regulators worldwide. And there is no ability or concern about integration. Once the messenger is in the cell, it's in the cytoplasm. It functions for about 24, 48 hours. It gets degraded into food for the cell. While it's got a modification, it's a naturally occurring modification found in transfer RNA. So once it's inside the cell, it works as it's supposed to, and then it becomes food, and it has no conceivable way of having a long-term effect. It's a transient, if you will, carbon copy of the gene that even the gene isn't there, which is the viral genome.
Thank you very much, Dr. Zach. Going back to Dr. Taylor, so what would be the side effects of Johnson & Johnson vaccine? Can you share it with us during the development of the vaccine?
Sure. I mean, I actually had a slide on that, and I'm happy to project it again if we need to. But generally, it's well tolerated. What we found in what we reported in our interim analysis is that most of the side effects were grade 1 and 2. The effects were what we would expect with most common injections. So injection site pain, some headache, fever, myalgia, but no grade 4 events. And so we would say consistent with a spectrum of a vaccine.
So another question. So could you please expand on the plans to advance the Johnson & Johnson vaccine for global access?
So at the moment, as what's ongoing at the moment is that there are a number of discussions worldwide. So I can say that there's different levels of discussions. So, for example, some countries will be choosing to have the direct one-on-one, so to negotiate what we call the bilateral agreements. Particularly for this region and in sub-Saharan Africa, our global public health organization are engaging very strongly with Gavi and the COVAX facility to discuss what is going to be the provision for the Gavi 92 countries. And then, so this is an ongoing discussion. And of course, our commitment stands that we will provide 1 billion doses in a pandemic situation. So, we are committed to sort of global equitable access. And of course, we don't know yet what that looks like, but there are a lot of people working to ensure that happens.
So this is a question for both of you. When do you think we will have an effective vaccine?
Catherine, you want me to take that first?
Well, you said you were going first, right? So I'll let you go first. I
think we're likely to have data first, so I guess I need to answer that one first. I don't think it's going to be a black and white. In other words, it's not going to be one Monday morning we wake up and everybody can go to the local CVS and get a shot and it's over. I think it's going to be a prolonged, slow evolution over time that will see different countries getting access on different timelines depending on the choices and the commitments they made. And within those countries, I think different governments are going to have different viewpoints on who gets access first while supplies come online. I think the data will come quickly and we'll have clarity on data, but that's just the first step. And I think then the attention will shift to deployment. Now, that being said, my anticipation, if you look at the projections for people's manufacturing capacity, is that by next summer, a good proportion of people should have been vaccinated. And certainly by the next winter, I'm hoping that vaccines have made a significant impact on this pandemic. Will it be completely over in every reach of the globe? Probably not. But will the current rates of morbidity and mortality significantly subside and life return to a semblance of a new normal? I think by next winter, I'm optimistic that yes. But the year is going to be tough because I think first the winter is going to be challenging until we have vaccines. And then as we get into the spring and vaccines start to become available, it's going to be complicated in terms of getting ourselves all vaccinated. That's my sense.
And Dr. Taylor?
Yeah, I mean, similar answer, but if we're talking about actually strictly about the data science, we can obviously make certain predictions. It's an event-driven study, so we need to have the statistically prescribed number of events in order to be able to read out our data. Of course, then this is very much dependent on the speed of the events happening, and of course, we use sort of modeling forecasts to choose deliberately sort of high exposure areas, right, to see if we can detect that difference faster by going to countries that have very high event rates. But ultimately, it's all down to the events. So, again, it's hard to pin it down because of that. But we're looking at data sometime in the first quarter of next year, and then again, as you've heard, each country will have their immunization strategy determined, and beyond that, it's about how the doses reach the people. But again, hopeful that we will be able to get a good number of doses out to the patients within 2021.
Okay, thank you. We hope we can get it earlier, so we can go back to our normal life. But that's still good news. So this is now a general question also. Among all the potential COVID, this is from the chat box, among all the potential COVID-19 vaccine, what defines a successful vaccine candidate? This is not going to be the only virus that we will face. And I think this is a great question, just a general question about the best way to develop an effective vaccine, best strategies as well.
I think, I'll take a stab at answering this because I think it alludes to the conversation we were having as a group earlier, right? I mean, I think there is room for more than one vaccine, and I think that each vaccine brings something different to the table, right? So there are points of differentiation, which will suit different, whether it's a 1-dose, 2-dose vaccine schedule, or the storage and supply chain conditions, or the actual platform in which it's delivered. I think there are all nuances, right? So I think it's going to be assuming that each vaccine is successful in terms of efficacy, demonstrating efficacy, then I think there will be, like the seasonal flu vaccine, a number of different options to choose from, right? And they're some will suit some populations more than others. So it's an almost impossible question to answer, I think.
So I I agree with that, which is why on this one I'm going to take a more parochial stance as I look at it from the viewpoint of the messenger RNA technology. I start with, I think, what's most important, and I think Catherine alluded to it, which is safety and efficacy. I think the silver lining for me is that this will have been a showcase for science stepping into the fold quickly and providing a benefit. I think on the back end of it, I hope we will have demonstrated that the mRNA technology is very potent in terms of the immune response and the ability to translate that immune response to prevention of disease. I mean, we've already seen the potency on the immunogenicity. Now what's ahead of us is demonstrating that that matters in terms of an effect on disease. Once we do that, I think we will have done two important things. The first is we will have set up the expectation from a regulatory and clinical sciences that if this virus mutates, and we have a SARS-CoV-3, that actually we can very rapidly react to it. And the second point for us as a company is we will have set up a manufacturing footprint that means that next time scaling up will be faster. And so I think coming out of the other side of this, there will be some really key learnings on both the science, technology, and the manufacturing that should enable us to be better prepared for the next one.
Great. So another question for you, Dr. Zach. We did hear from the CEO of Moderna earlier that he expects to get FDA authorization in December. Can you elaborate on that?
Well, I think what he said is that December is a likely time frame by which we could see positive results and then submit for emergency use authorization. And I think it's based on three things. Number one, the FDA guidance, which tells us what is the amount of data that we should expect to be able to submit. Number two, seeing the cases occur on the trial which are at expectations relative to the population where the trial is running. And so we think we're going to be on track, as I've alluded to have the first 50 cases in November, and by the end of the year, between 100 and 150 cases. And number three, the high level of immunogenicity that I've described earlier, which I think puts us in a good place to actually show efficacy at a high level in one of those first interims. So I think that's the basis for his optimism of expecting this to work, knowing that the cases are going to be there, and understanding from FDA what's required to get an emergency use authorization.
So going back to you, Dr. Taylor, so what opportunities for collaboration that are available with Johnson & Johnson for the MENA region? So how can we work together?
That's a very broad question, right? So, I mean, I don't know exactly, was it referring to scientifically or particularly regarding the vaccine? Or
vaccine production, scientifically.
So, one of the things that we're advocating for very strongly is the inclusion of the Arabic ethnicity in our upcoming clinical trials. And as a company, we've already started doing some very innovative trials in the region, in Saudi, in UAE, and we. And this is not COVID. This is in other products for myeloma, for example. So there is an absolute commitment from Janssen to partner on clinical studies in the region. In terms of COVID, as I mentioned before, we use epidemiological forecasting to determine where we will place our future studies. And if I look at the new caseload in Saudi Arabia, or as of yesterday, I think you guys have been very successful in controlling your current new case rate, and therefore at the moment, actually, you compare it to some of the much more high exposure countries in the world, especially in South America and in the US, and now Europe looks like it's getting a second wave. At the moment, the epidemiology cannot forecast, are not forecasting you as a high risk area. So, of course, we're very aware that the Arabic ethnicity is one that we need to observe, and we are working with our global team all the time to raise awareness of this. But congratulations, you guys have been very successful in controlling the disease, so that's a good thing.
Thank you very much. So this is a question for both of you. It has been said that the duration of the vaccine is short. Is this what is expected? And will we need boosters? And do we need it every year? So for Moderna vaccine and for Johnson & Johnson, it can be specific for your vaccine.
So let me I'll answer it in a couple of ways, because this one is obviously an important one. First, I think it's too early to say, is the truth, in terms of duration of effect. There have been conflicting reports as to the durability of antibodies post-infection. I think, barring a few case reports, it's pretty much uniformly true that if you've been infected, you're not going to get infected a second time. There have been some case reports of that not being true, but they've been by and large rare. So my sense is that if we achieve a high level of neutralizing antibodies, and if there's enough T-cell support to sustain that, then we should see memory, and that should take us at least through a season. The question on boosting, I think, is a relevant one, and I think we and others are ultimately going to have to test this over the long run, whether you need a booster shot at 6 or 12 months, or longer. I think people are starting to discuss that. I think, and I'll let Kathy talk about the J&J, because they clearly have an interesting test going. The final point I'd make is this is where different platforms are different. With our vaccine, we know we can boost without interfering immunity for a very simple reason, is that the body doesn't recognize our vaccine. It doesn't recognize the LNP. It doesn't recognize the mRNA. It only recognizes the protein once it's the vaccine is the mRNA is in the cells and making the protein. We've shown this with other vaccines. For example, our CMV vaccine is given with a prime boost and then another boost at 6 months, and the antibody levels keep going up. It's beautiful. And so I believe that our vaccine not only will be able to boost in the long run, but we'll also be able to boost other platforms should that be needed. Whether we need an annual shot like flu, I don't know. I don't think so, because remember, with flu, it's not the vaccine stopped working, it's the virus moved along and changed. So that's the primary reason why you get a flu shot every year. But I do think that there is a strong possibility we will need an additional booster shot at some point to get a stronger immune response, whether that's at 6 months, a year, 2 years. It's by and large going to be a problem for us in 2022 and beyond. I think the focus in 2021 will be to give everybody their prime or prime boost and start to turn the tide of the pandemic. Over.
So, I mean, from a J&J perspective, we just I don't want to sound facetious, but because of the acceleration, we actually haven't had a year, right, with the vaccine in existence. So we simply, because of the great will to accelerate, we just don't have the data at this moment in time. So, of course, this is exactly what we're doing studies to accumulate. And as you'll remember from my presentation, that in our phase 1/2A, we had a cohort that actually looked at the duration of boosting and the duration of immune response, and that's up to 2 years. And again, in phase 3, right, we will be looking at the effects of, well, our primary phase 3 is in 1 dose. We will also be looking at the effects of 2 dose, right? So we want to have this full repository of data, which we will get in time, but there's nothing more that we can do at this point than wait for time to elapse. But I'm again with Dr. Zaks on here. I think we need to be in that position where we show good efficacy after 1 dose. We achieve our phase 3 study, and we start vaccinating people to really control this pandemic.
Thank you, Dr. Taylor and Dr. Zak. So I believe you answered Dr. Zak this question during the discussion, but I will just repeat it because I believe this question will come to every geneticist. So we've seen reports that of the viruses having mutated to be more infected. Will the vaccine work against these mutations? Do you anticipate new mutations, and what will we do if the vaccine doesn't work anymore?
Yeah, let me take a first stab at that, because obviously this is not lost on anyone. Most of us are using the original strains to design our vaccines that were put into production back in the beginning of the year. We've since then seen one mutation emerge that is more infective, that's taken over the Western world. I think there's now talk of a second one. So far, the important thing to note is as follows. First, the mutations that are clinically significant in circulating are all blocked by the neutralizing antibodies that our vaccine generates. We test it when we actually test for the serum. If you look at the New England Journal, I think there's reference there of the 614 mutation. In fact, the neutralizing antibodies block it just as well or even a tad better relative to the wild type. So a mutation that causes an increase in infectivity by binding better or other mechanisms that we don't understand is not the same as a mutation that is an immunological escape. And so far, I've not seen clinically significant immunological escape mutations out there. So everything that we've tested, and we try to test against what we think is relevant, the neutralizing antibodies that we generate seem to block. Now, that's not to say that in the future we won't see a mutation that is a true immunological escape such that prior immunity no longer holds, like influenza. It's less anticipated because these beta coronaviruses actually have a proofreading enzyme, and so their rate of mutation is actually lower than other RNA viruses. But it's possible. And I think what's going to happen is if that is the case, we'll call it SARS-CoV-3 and we'll need to make a new vaccine. The good news is everything we've learned on how to quickly make a vaccine using the technologies that we're talking about today and the manufacturing footprint we will have stood up means that the next vaccine should take much shorter than the year it's taken us this one. So while this appears to be a world record, in a world where there's a new mutation and established proven technologies with an established manufacturing footprint, it should take us weeks and months, not a couple of years, to get to billions of doses. Over.
Okay, thank you very much, Dr. Zach. So the next question is, what is the advantage of Moderna vaccine compared to the other being developed? And what is the advantage of Johnson & Johnson vaccine compared to the others being developed?
So I think our advantages are threefold. Number one, we seem to be very potent, as good or better than anything else out there, which I hope will translate into a high level of protectiveness. So I think that's one potential advantage. Number two is the ability to respond quickly. And that, with an existing manufacturing footprints, I think puts us very well positioned for any future pandemic. And number three is a little bit more subtle, but I think that as I step back, this platform is more likely to work because of higher biological fidelity. In other words, I don't have to worry about protein misfolding or other untoward things happening. Once I show it works a first time, it's much more likely to work a second time. In fact, this is the 9th virus against we are able to mount neutralizing antibodies, and we've tried it against 9 viruses. So if you look at our hit rate of the ability to consistently generate neutralizing antibodies, albeit still in phase 1, it's been pretty robust for the platform as a platform. And I think that's because the cell can't make the wrong protein. It's made inside our body. So as long as we give it the right genetic information, it'll make the right protein, and the rest is up to evolution and immunology. And I think that high biological fidelity and the agility of the platform are going to be inherent advantages, not just for COVID-19, but for the future. Over.
Dr. Taylor?
Sure. I would say that we do offer some things that seem to be specific to us. So I think the most prominent is probably the 1-dose vaccine schedule. You'll see that many others are too. Now, for many, this may not be a differentiator, but you can see that particularly for us in a region where, and in a pandemic situation where you're trying to locate people, vaccinate them quickly, this will have implications for the speed of getting patients immunized, and also the amount of people who can be vaccinated in the first instance with the same number of doses. The second benefits we bring is that we have quite favorable storage and transportation conditions. So We have a 2 to 8 degree storage condition, so this is a kind of bog standard refrigeration requirements. So especially again for this region, perhaps an easier condition. And then finally, I think I mentioned before that the ADVAC 26 platform is one that is not new, right? So we can have some confidence in the fact that we have about 90,000 patients now walking around with the same platform with the Ebola insert. So who are we have continued to monitor, and we know that this is the platform that is well tolerated and has not generated any at least medium-term safety signal that we need to be aware of.
Thank you. So this is another question for Dr. Zach. Scientists have developed ultra-potent COVID-19 vaccine candidates. According to the researchers, including those from the University of Washington School of Medicine in the US, the nanoparticles vaccine produce virus-neutralizing antibodies in mice at level much greater than that is seen in people who have recovered from the disease. So I believe they would like maybe to know the side effects of using nanoparticles. That's from the chat box.
Okay. Well, if I understand correctly, if you're asking me about our vaccine, yes, we have higher levels than neutralizing antibodies than what you've seen on average, but it's within the range of what you see with people who've been sick. That's why we're confident about the immunogenicity. I don't think the nanoparticle per se is likely responsible for significant adverse events. You see some pain, but that's mostly mild, and you see that with any vaccine you give. So that's nothing new, and you don't see an exacerbation of that on the second dose, so it's probably not specific. The lipid that, if you look at the components of the lipid nanoparticle, there's one lipid that we've invented that enables it, but that lipid has a very short biological half-life of a few hours, and so I don't anticipate any adversity from it. The rest of the particle is, PEG, which is in our toothpaste, cholesterol that's in our body, etc. So there's no black magic there, and I don't have any anticipation of adversity. I think what we see for the most part in terms of the adverse event profile is related to a strong immune response, and I think that's good.
Okay. Thank you very much. If you would like, we have 1 minute left. If you would like to add anything, Dr. Taylor, Dr. Zach?
Just simply, I think that some people may consider that by putting Moderna and Johnson & Johnson on the same panel that we're pitted against each other as adversaries. I reiterate again that each vaccine has their own to bring to the table. There is some differentiation, but honestly, we just need as many vaccines as we can to treat this pandemic, and I really do trust and hope that with the benefits of great science and the greatest minds, also in collaboration with academic institutions, with governments, that we will be able to do something to control this outbreak very, very soon.
I can only say amen. Thank you for the invitation, and may the spirit of collaboration and health that this pandemic has brought out prevail beyond the pandemic for a better world afterwards.
Thank you very much. It has been a great pleasure hosting you, and hope to see you in person soon in Saudi Arabia. Thank you. Take care. Thank you very much. It has been a wonderful session. Thank you.
Thank you.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound. Where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
المملكة استضافة قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound. Where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
المملكة استضافة قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
life.
Assalamu alaikum. So it's really a great pleasure to have this first day of the COVID-19 vaccine global challenge and prospect. And really we have learned a lot from the leading scientists, and also actually we have heard good stories about what these vaccines are and how the biology around them. It was amazing. It was really amazing. And personally, I have learned a lot. And in this session, we just give you some learnings. Today, myself, Dr. Ahmed Salman, and Margarita, give you some learning. And personally, I learned, for example, as we all know, that there are 6 different vaccines in the pipeline, vaccine type of vaccines. We have the attenuated vaccines. We have the non-replicating virus vaccines, and we have DNA vaccines, we have the RNA vaccines, we have the protein vaccines. And luckily we heard actually the latest news about these vaccines today. And the first things that we have learned from Professor Bali is the immunity in the body, and is the importance actually on what we are doing with this immunity to actually help whatever treatment we are going to have, either antibody-based or maybe a vaccine-based or another therapeutics for this COVID-19s. And the things that really strike myself is about the microbiome in the gut. That is how actually they play a big role in how these vaccines and the immunity in the body is playing a role. That is actually, it will tell us that it's not only the vaccine that we need to watch and the therapies, but also what we are eating and our behavior during the day. And all the things that we have learned from the immunity is about how the antibodies and the vaccine actually can work together. So, and we can actually augment the vaccine's efficacy and its protective role if we can actually combine them with antibodies, and also on how we can use the system biology. This is for the biologists, how to use the system biology and actually ask these questions that everybody in the world, and especially in the virology about the COVID-19s. What is the immunity? We don't understand it yet. But I think the scientists like Dr. Bali and other immunologists, they're coming to us with answers, and these answers actually they are important for all of us. Now, to sum up the day, actually, I will let Dr. Ahmed Salman to give us some learnings from this lecture that we have learned. And then after that, Margarita will give us actually an overview of what will happen tomorrow. And then we close this today. So I will let Dr. Ahmed to give us the learning of the lecture that we have heard from the leading scientists. Dr. Ahmed?
Thank you. I would say that the world after COVID-19 will never be as the world before COVID-19. But wait a second, before thinking that sounds very tough. I would say think deeply about it. You might find there's lots of good sense about it, and it might have also some good points. I mean, I'm very impressed about how scientists and organizations and different bodies that basically put everything behind their back and they work together. I'm very impressed in that in a small scale, as we saw today, how scientists from different parts of the world collaborate, help each other, share the knowledge and suggestions and recommendations, and how this basically will speed basically defeating that pandemic. I'm sure about this. And that also will help reassessing the priorities of the world. I'm sure people will not just give the attention to the medical science as part of just to save lives. It will be more than that. It saves our livelihood. It's important for our economy. It's important for our basic life, social activities, mental health. So I'm sure after this tough experience, there will be lots of good side that might drive this world to a better direction. So today, there were lots of nice talks. amazing about how these scientists work day and night very hard to achieve that mutual goal about generating different vaccines and really achieving that in unprecedented speed. How this happened in that short time, this is a really record in that field of vaccination that in a standard situation takes up to 10, 15 years. So today we heard about different type of vaccine, different strategies, and each one of them has very good sides that complement the other type of vaccines, maybe some minor, I would say not perfect sides. As far as things can be produced in a large scale with a good price, other vaccine can produce in faster, can produce in safer way. So non-replicating vaccine, as example, can be applied without adjuvants. So that's going to save lots of the cost required and the stages of the manufacturing. RNA-based vaccines, it's also can produce to give a quick immune response. They might have some points about negative sides about the storage and so on, but that could be also applied because there will be always a room for than one vaccine. And I think having all these vaccines together, they definitely put us in the correct track and reaching the mutual goal for all these scientists. So I would like really to thank everyone was participating today in the organizations or in the speakers. They did an amazing job. And I will pass the microphone now to Dr. Margarita to speak.
Thank you, Dr. Salman. Today was a fantastic day. I watched it from the very beginning, all through the night until this morning. And, 10,000 attendees watched the show today and tomorrow, and from 94 countries. And we hope everybody comes back tomorrow because tomorrow's program is going to be very interesting. One of the interesting tracks that I'm really looking forward to tomorrow is 3 of the top humanitarians in the world will join us and give us insights into the challenges of distributing the vaccines worldwide to people in need. And these are 3 of the top organizations, and they've already been very involved with delivering supplies and medicines to people. So they'll be able to tell us where the need is and the logistics of getting the vaccines to those places. And I'm really looking forward to that. And we're really fortunate to have humanitarians as part of this conference, which I really want to thank Dr. Bouz-Jaleh and Dr. Salman for thinking of having humanitarians at the conference too, because it's so important that we once we have these vaccines that we can help everybody in the world. So I'm really looking forward to that. Dr. Salman, do you want to talk about any of the other speakers tomorrow?
Yeah,
give a preview.
Sure. Tomorrow, of course, will be more scientific talks as well, in addition to the humanitarian talks. So that will be a very rich day. And there will be some nice closing talk by Professor Adrian Hell, summarizing everything and telling also the learning points from the different talks and different vaccine strategies, in addition to having Professor Faith Osier giving us in the discussion highlighting very important points about how vaccines could be delivered to low-income countries, including Africa, and the distribution issues as well. So that will be a very interesting talk, in addition to all the talks and the speakers tomorrow. So we'll wait for you tomorrow to join us.
Mohammed, before you say that, I want to say one thing. For anybody who missed any of the talks today, the fantastic team in Saudi Arabia is going to take all the recordings and then share them with you. So if you've registered for the conference and you miss anything today or tomorrow, we will send you links to speeches broken down into an hour each, and you can watch them. So I just wanted, so if you missed anything, you'll be able to see it. Okay. Go ahead, Ben.
Just want to conclude that actually welcome you tomorrow, and actually from our country here in Saudi Arabia, we have special experience. Before everybody has experienced the COVID-19, we experienced the MERS-CoV, and luckily we have long experience in this. And tomorrow, one of our leading scientists, Dr. Naif Al-Harbi, young scientist, Saudi Arabia, they will actually give you the data on clinical trial of the HPV vaccines, how we've been successful first in vaccinating the camels, and then now we're vaccinating the human in phase 1, phase 2. And these are all good stories that you will hear that actually give you the happiness and the relax that actually the COVID-19 vaccines will be efficacious and the humanity will return to its norm. So we thank you all, and actually we look forward to you for tomorrow to complete the day. And thank you very much for attending this meeting today, and we look forward to meet you again tomorrow. So have a nice day if you are in the Far East, and have a nice evening if you are in the Far West. And we wish you a good day, and thank you so much.
Thank you. Thank you
so much.
On a bullet train in which we race against time towards developing a vaccine to save life and humanity. This journey includes 32 experts and 10 scientific sessions, in which we discuss 6 topics revolving around developing a successful vaccine against the COVID-19 virus. From Riyadh, Kingdom of Saudi Arabia, we welcome experts to whom the world looks with hope, and major companies that will lead the way in distributing vaccines around the world. From Health Affairs of Ministry of National Guard, King Abdullah International Medical Research Centre, KMARC, King Saud bin Abdulaziz University for Health Sciences, and the Saudi Secretariat of the Group of 20, G20. We say to everyone, let us proceed with one ambition and with one goal towards a meaningful life, and let us work together to protect the health of our fellow human beings and our future generations.